Hello. Good morning, everyone. Welcome to the second day of the third BRICS Neuroscience Symposium 2026. I'm Tahiti, Research Associate at SGBC, and it is my pleasure to serve as the MC for the first half. We will begin our day with a mindfulness practice guided by trained yoga professional and dance choreographer, Mrs. Ashika Jain. Mindfulness offers an opportunity to pause, focus our attention, and prepare ourselves for a day of learning, discussion, and collaboration. I will now invite Mrs. Jain to lead the session.
Hello. Hello. Can you all hear me? Can you all hear me? All right. I think I should be in the center. How is everyone doing? Tired? Everyone tired? Because I can see puffy eyes. Okay, so can we start the session, Tahiti? All right. Before we begin to the session, I would like you to invite for a simple vocal resonance practice by chanting Om. Okay? So we use 3 syllables, A, U, and N. I'm going to chant it once for you. If you are comfortable, you can join or simply listen. Okay? I close my eyes. You don't have to close your eyes. I'll instruct you by everything. I close my eyes. I inhale. So the sound flows one into the other. You don't have to speak like separately. No, it goes one into the other. Okay? Can we try this? 3 times. Everyone. Keep your spine erect. Try to sit erect so that you have more space in your lungs. Can we close our eyes if you're comfortable? You can take your glasses off for some time. You can keep the glasses on the table. Eyes closed. A little smile on your face. Inhale, everyone, through your abdomen if possible. Let's do that together. One more time. Inhale, everyone. Keep your eyes closed for some time. And very gently open your eyes. I'll just take 2 minutes to introduce myself. I'm Ashika Jain. I'm from Bombay and it's been 4 years I've moved here. I'm a yoga teacher, a dance choreographer, and I run my own studio here called Yoga Natyam, where I try to help people to cultivate balance, peace, presence, and well-being. So today, we don't have much time, so I'm not going to ask you to achieve anything, perform anything, or be relaxed in 20 minutes, 30 minutes, because I'm assuming that for next, like the entire day, you're going to assume, analyze, question, solve, discuss. So I request you to shift your energy or your attention from doing to just observing. Okay? Nothing to perform, nothing to fix, nothing to achieve. Just observe. Can we do that? Can we start our session? Are we all ready for the session? Thumbs up? With a little smile will be great for me. Thank you so much. Okay, so try to sit comfortable. You can take your shoes off if you're comfortable. If not, you can be yourself. Just be yourself. There is no one to judge you. For 35 minutes, 30 minutes, be yourself. Okay. Can we close our eyes, everyone? Keep your spine erect, not very stiff, but comfortable. And tell yourself in your head that for next 35 minutes, I'll be with Ashika's instructions. I'll try to be. If not, never mind. No problem. I'll try to surrender. I'm ready to surrender. Because here, I'm not here to believe anything, achieve anything. I'm just here to observe and surrender. You can rest your arms on the chair comfortably. Let it naturally fall on the chair. Keep your eyes closed for more focus, if possible. Now imagine you have traveled all the way here. Just imagine. You've entered the room, found your seat, and you sat on the chair. Observe the contact of your body on the chair. Observe the weight of your feet against the floor. Just observe, nothing is changing. You're not supposed to alter anything. Just observe the way it is. Relax your eyebrows. Your eyebrows are going away from each other. Observe the temperature of the air against your skin. Now very slowly, take your focus on your breath. We are not taking deep breaths in, we're just observing. If it is shallow, let it be shallow. If you feel your natural breath is until your stomach and your lungs, let it be that. We're just observing. Inhale, and exhale. Inhale and exhale. Inhale and exhale. Inhale and exhale. Keep your eyes closed. For next 35 minutes, 30 minutes, your eyes are closed. Now for next few moments, we are going to intentionally create tension in our body so that you can easily recognize the release tension from your body. Take your focus on your feet. We are going to create tension, so try to clench all the toes very tight. Remember, no one is observing you. No one is judging you. Be yourself. Try to clench your toes, all the toes, very tight. I want to see tension, very tight, very tight. Calf of your calves, your knees, your thighs, your glutes, everything is engaged and tightened. Very tight, little more tight, tighter. Hold this tension for a while. Tighter, and release and relax. We'll repeat this for a few times. Clench your toes, all the toes. A little more tighter, you can keep your eyes closed. Just follow my instructions. Calf muscles tight, knees are tight, back of your knee muscles, your shin, your thighs, your glutes. This time pull your stomach in. You're not holding your breath. Breath is normal. Pull your stomach in. More tighter. Let it go towards the spine, back of the spine. Your abdomen is going towards the back of the spine. More tighter. Tight. A little more tight. Tighter. Everything is creating tension. Hold this tension for a while. A little more tighter. Tighter. And relax. Let's repeat this again. Clench your toes, all the toes. Your feet is tighter. Your calf muscles are tight. Thighs, glutes, abdomen. This time make a fist of your palms, both palms. Grip it tighter. Your forearms are tight. Your biceps are engaged totally. Lift your chest slightly up. Puff your chest, your scapulas, your shoulder blades are coming close to each other. A little more tighter, people. Tighter. Remember, no one is observing you. No one is judging you. A little more tighter with your palms. Tighter with your palms. More tight. Make a fist of your palms. Tighter. Tighter. And relax. One more time. Last 2 times. Let's go. Give your best. Clench your toes, your calf muscles, your shin, your knees. Take your focus there. Your thighs, your glutes, your abdomen. Chest is a little puffed out. Shoulder blades are growing close to each other. Make a fist of your palm. Very tight. Make a fist of your palm. Forearms tight, biceps engaged, totally engaged. tighter. This time try to look towards the ceiling. If you don't have any neck problem, please go ahead. Keep your eyes closed. Look towards the ceiling. Squeeze your eyes. Your eyebrows are coming close to each other. Squeeze, look towards, feel the stretch on your neck muscles. Up, stretch it tighter. Feel the tension in your muscles. Every part of your body is active. Tighter. A little more tighter with your hands, people. Remember, no one is watching you or judging you here. Let's go. Tight, tight, tight. Look up. Feel the tension on your neck muscles. Tighter. Open your mouth. Stretch your mouth because these are the muscles we don't relax. Open your mouth as if you want to scream louder, but don't yell. Scream. Open your mouth. Open your mouth, squeeze your eyes, stretch. Yes, people, there you go. Open your mouth. There you go, people. Yes, open your mouth, everyone. Stretch. Little more. You can stretch it more. Let go, people. Let go. More. Stretch more. You should feel the tension in your face muscles. Yes, now. I can see that. Tighter. And relax. Now very slowly, again bring your attention to your breath. Observe the natural flow of your inhalation and exhalation. We are going to do box breathing. You can keep your eyes closed. Box breathing is inhale, hold, exhale, hold. I'm going to instruct, I'm going to give the counts. Try to be with my counts. Try to breathe through your abdomen because we humans breathe until our chest. Like we use only 20-30% of our lungs. Inhale, 1, 2, very slow, 3, 4. Hold your breath for 8, 7, 6, 5, 4, 3, 2, 1 and exhale slow. 3, 2, 1 and again hold your breath. Do not breathe in. 1, 2, 3 and come back to normal breathing. If you are comfortable, you can hold 8. If not, you can reduce the counts. You don't need to hold it very tight. Let's do that again. Inhale, everyone. 1, 2, 3, 4. Hold your breath. 8, 7, 6, 5, 4, 3, 2 and exhale slowly. 4, 3, 2, 1. Pull your stomach in and hold. 3, 2 and normal breathing. Let's repeat this for another 3 times. I'm going to increase the count. Inhale for 5, hold for 10, exhale for 5, and again hold for 5. If you're not comfortable, you can reduce the counts. Inhale, 1, 2, 3, 4, 5. Hold your breath for 10, 9, 8, 7, 6, 5, 4, 3, 2, 1 and exhale. 5, 4, 3, 2. Pull your stomach in and we are holding. 5, 4, 3, 2 and normal breath in.
One more round. Inhale, everyone. Try to inhale through your abdomen until your abdomen. Inhale, 1, 2. Feel your breath, 3, 4, 5, and hold, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Exhale, 5, 4, 3, 2, 1. Again, hold, 4, 3, 2, and normal breath in. Observe yourself. Now we'll scan our body, just scan and observe from top to bottom, bottom to top. Usually we do this in Shavasana, but we are going to sit and do it. Remember, observe. We are not changing anything. Let's take our focus on our toes. Just observe all your toes. Observe your feet. Observe your heels, sole of your feet, ball of your feet, the little toe, big toe. Just observe all the toes. Observe your ankles. Your calf muscles. Your shin bone, that is the front part of your calf muscles. Your knees. Back of your knees. Just observe. See if you can feel any sensation in your body. If not, do not create it. Just observe. Your thighs. Back of your thighs. Your groins, your glutes. Your abdomen, your lower belly. The entire torso, your chest, shoulders, your shoulder blades, back of your spine, mid spine, lower spine. Your fingertips. These are your nerve endings. Observe them. Your ball of the palms, heel of the palms, center of the palms. Your entire fingers, all the fingers. Your forearms. Eye of the elbow, your elbows. Your biceps, your shoulders, your triceps. Your neck. Your chin, your lips. Check your tongue. Your tongue should rest at the bottom palate of your mouth. To relax consciously. Check your eyebrows. Your eyebrows should go away from each other. Create space between those two eyebrows. Your temples, forehead, crown of the head, back of the head. Just observe. We are scanning our body. Crown of the head, back of the head, your ears, your cheeks, your nose, eyeball resting at the lower socket of your eye. Tongue resting at the bottom palate of your mouth. shoulders, the entire arms, your fingertips, your palms, your wrists, your chest, your middle abdomen, lower abdomen, groins, thighs, glutes, knees, back of the knees, your calf muscles, your shin, your ankles, your heels, your toes, ball of your feet, sole of your feet. Take a deep inhalation through your nose and exhale through your mouth. Very soft exhalation to let go. Let's do that. Inhale. Little exhalation through your mouth. Inhale. Exhale through your mouth. Inhale. Exhale through your mouth. Keep your eyes closed for another 2 minutes. You are almost done. Stay there. Just observe the state of your mind. Do you feel any change? If not, no problem. Remember, mindfulness is not about creating an awareness. It's about becoming more available to the experience already here. May this quality of attention, curiosity, presence accompany you through today's discussions and discoveries. We are going to end the session by 3 Oms again with 3 Shantis. You can follow me if you are comfortable. If not, simply listen. I inhale. Shanti, Shanti, Shanti. Observe yourself, come back to the room. Observe your body. Observe the contact of your body against the chair. Weight of your feet against the floor. Take your time, and whenever you're ready, you may gently open your eyes and be yourself. If you want, you can just create some warm energy in your palms, because these are nerve endings and we are going to use our eyes throughout the day. Just make it a little warmer. The ball of your palms, the heel of your palms, you put it on your eyes. Gently massage your eyes, anti-clockwise and clockwise. And look at your palms with a great smile on your face. I want everyone to smile and tell yourself that you're going to be aware of your breath, your emotions throughout the day. Thank you, everyone. Namaste. So we ended our session with 3 Shantis. Shanti, it comes from a Sanskrit word which means peace. Why 3 Shantis? First Shanti is for yourself, inner self, because when you are at peace, you can help others. You can't be sick and help others, right? You need to be healthy. You need to be at peace at some level to help others. Second Shanti, again peace, for your surroundings. Third Shanti is for the world. Any questions? If you have any questions or doubts, you can just mail me. My mail ID is yoganatyam, YOGANAATYAM @gmail.com. Kritika is here. You can just talk to her to get my contact details. Thank you so much. I had a wonderful session with you. Have a wonderful day.
Thank you, Mrs. Jain, for that refreshing and grounding start to the day. Before we proceed with today's scientific sessions, I would like to briefly outline this session format. Each scientific session will begin with a 15-minute featured speaker presentation, followed by 2 or 3 talks of 10 minutes each. The session will conclude with a 10-minute question-answer session. As we have a combination of in-person and virtual speakers, we kindly request all presenters to adhere to their allotted speaking times. A bell will be rung 2 minutes before the scheduled end of each talk to indicate the speaker should begin to wrap up their presentation. And lastly, we have the slide changer and the pointer here. The top button is for pointing, and the bottom ones are for changing the slides. And if that doesn't suffice, there's one pointer here as well. Thank you all for your cooperation. Without further ado, we will now proceed. Following the 6 highly engaging scientific sessions from yesterday, we will now commence the 7th scientific session of the symposium. For this session, it is my pleasure to introduce our featured speaker, Professor Michel Valdez-Sosa, followed by Professor Taize Graziel Lima de Oliveira Tutayan, and Professor James Olukayode Olupade. The session will conclude with a 10-minute question-answer. I now invite Professor Michel Sosa to begin the session.
So I thank the organizers of this meeting for allowing me to participate in this very interesting activity. And I must say, this is the conference which I will begin with the greatest peace of mind in all of my professional practice. So thank you very much for the mindfulness session. Now, I'm going to talk about near mesoscopic circuit changes underlying visual selective attention. And the mesoscopic part here is very important because there's a lot of work going on now here in India and in other countries related to this. So this is a very busy slide because there's a lot going on in neuroscience today. And we can work at the macroscopic level, hear a lot of information from fMRI about the organization of different circuits in the brain. We have a lot on the microscale. And now there's a terrible expansion, important expansion of work about the mesoscale neural circuits. And a lot of this is being done here in the CGBC. Now, it's very important to see the ties between the different levels. This one, the connection of the micro and molecular level with the mesoscopic level, I think is going to proceed at a fast speed. In fact, we've been told that after the characterization of tissues, of cells, cell counts, anatomy, now the whole field is moving towards transcriptomics. So this connection is guaranteed. However, this other connection, which is what is happening at the level of the whole brain and whole brain circuits, and its connection with the mesoscopic scale is not very clear. And one of the problems is we need an fMRI studies that are closer to the mesoscopic level. Current fMRI connectivity has moved away from the mesoscopic level. Once you average all the activity in a region of interest, you lose the details of the computations that are taking place inside that region of interest. And all these connectivity studies that come out of, for example, resting state, they're telling us interesting things about the highways through which information is going from one brain region to another. But they obscure what information is transiting along those highways. The development of very high field, above 7 Tesla MRI, is increasing the spatial resolution. Thus, we could look towards voxel-level encoding models, which will push fMRI connectivity towards the mesoscopic level of analysis. The visual system is an ideal battleground for this strategy because we know very much about the visual system. And the interface of consciousness and physiology can be clearly observed in one process, which is visual selective attention. Now, there are over 20 areas that have a specialization for visual processes in humans. I'm going to be talking today basically about V1 and V2, which are the first areas, and a tremendous amount of information is accumulated on how we process information in visual cortical areas. And here, the receptive field mapping techniques have played an important role. The classical receptive fields are well characterized in several species. We know now how different neurons respond. But the interesting development of the last decades has been the creation of encoding models where in humans we can study population receptive fields. With population receptive fields, we're essentially fitting a model in each voxel in an area responding to visual stimuli and trying to find which is the best Gaussian descriptor of activity in the visual field connected with the responses of that neuron. And this is an exhaustive process that has to test many models and is carried out for each voxel independently. Now, this is great, and with population receptive fields, we've been able to replicate many findings of the classical animal receptive field studies in psychophysics. For example, that the size of the population receptive fields grows with eccentricity away from the fovea, that it grows, the slope grows because they get bigger and bigger, as you see here in V1, V3, and V4, as you go forward from the early visual areas to the higher visual areas. But this technique is time-consuming, very noisy. It's estimation by voxel, and it's very difficult to integrate with experimental tasks. A new technique has appeared, which is the connective field mapping. With connective field mapping, what we're doing is fitting a Gaussian model to see how we can predict activity in a target voxel from the activity in V1, for example. The idea is that we test many different models over the cortical mesh of V1, sum their activity through a Gaussian envelope, and see which one is the one that predicts best the activity in the target voxel. Now, this technique is promising, but it should be better for exploring functional variation of retinotopic maps and different experimental tasks, but it's very noisy, and estimation is voxel by voxel. The visual cortex is retinotopically organized. For example, this is V1. These are different eccentricities in the visual field. You see different colors here. And the lines represent different polar angles. If you describe the visual field in polar reference system. And here would be V1 and V2. Now, these basic retinotopic maps can now be inferred from surface anatomy alone by using deep learning methods. For example, there's a new model, deep retinotopy. You just have to input the cortical surface, and then you get a good estimate of the mapping of the retinotopic areas. And the connections from V1 to V2 are organized also retinotopically. So this is you see in green V2, red V1, and the organization is like a sheet. So there's a preservation of the topology of the retinotopic organization from one area to another. The basic skeleton, thus, of voxel-based connectivity is dictated by anatomy, but it can have deformations according to function. So we see here in red, feed forward, there's a convergence as you go to higher order areas, but you have to remember that there's a feedback from higher areas, and there's also within area connections. And this gives retinotopic maps a possibility of variation beyond the basic skeleton of anatomy. Now, the question is, do retinotopic maps warp under selective visual attention? There's a lot of preliminary evidence with PRFs that suggest they do in middle and higher visual cortices, but V1, V3, the evidence is scanter. PRF mapping is very difficult to reconcile with selective attention experiments. Although connected field mapping is more amenable to selective field attention experiments, it's very noisy, and this making the measurement of warps due to mental states very difficult. The problem is that both PRF techniques and CF techniques do not use the property of topology conservation from one area to the other. So we must evolve population information, use population information to constrain the solution for individual voxels, and this would give cleaner mappings, and we could examine the warpings. So now I'm going to briefly mention a method for studying visual mapping using connected field that was developed, and for now is focused only on the center of the connected fields. The connected fields have a center and they have a size, but we're concentrating here on the centers. We've tested this method in a visual selection task, global versus local attention. So I'll first present the experimental design and then explain the method briefly. So we've developed a technique which is called rapid serial object transformation. Now, this is known to all psychologists and many cognitive psychologists. This is a Navon figure. And you can attend the overall form, let's say here's an E, or the local forms, which is E. In our rapid object transformation, what we do is we have a mask, and then we unmask or a global letter or a local letter here. And if we do this very fast, we can measure the event responses to each of these levels separately. This is not possible with the classical Navon figure because both global and local information are placed in the screen at the same time. And what we demonstrated is, in previous work, is that, and here you can see in blue when the local level is attended, in black when it's not attended, and the difference waveform is in red. And there's a selection negativity over the extrastriate cortex, larger over the left side, when you attend the local level and ignore the global level. And if you do the reverse, you get the same result, but now a selection negativity on the right side of the brain. And this is interesting because there's evidence from studies in patients that left hemisphere lesions affect more local processing, and right hemisphere lesions affect more global processing. And this is just a control that the number of mistakes increases when the subject tries to identify certain letters at both levels at the same time, but is very accurate if he concentrates on one level only. So this is a classical let's say, visual selective attention response. Now, we adapted this same method for fMRI. What we do is we present global stimuli during a block that lasts 44 seconds, and then we present global stimuli. So this allows us to have blocks of fMRI activity when the subject is concentrating on the global aspects or when he's focusing on local elements. And the method we're using to study the changes of the retinotopic maps is thin plate spline fitting. This is a method that allows you to study deformations. There can be affine transformations, some shear, but always preserving the topology of the this is, for example, an application in anthropology, right? And we use the calculation of Beltrami coefficients to preserve the topology and avoid tearing and shearing of the maps. And so we call our method, it's a Bayesian thin plate spline warp with Beltrami regularization. We basically fit a mathematical model, the Shearer model, to the skeleton of V1. And then we have as our objective the minimization of the energy necessary to bend the shape. That would be the thin spline, thin plate spline, okay, and the Beltrami coefficient. Now, here's the results of the method. This is the fit of the model, which is very high, the Shearer model. Now, this is the determination of the center of the connective fields. As you see, it's very noisy. With our method, we allow variation, but we push the points towards the prior. And this reduces the amount of noise. So when you obtain a map under these conditions from global and local, you get something like this. This is the two cases. We're seeing that there's a shift of the center of the connective fields when you go from local to global towards the upper hemisphere. And this is one case. There are variation among cases, but if you submit it to a statistical analysis, a mixed effects model, this shift is very significant. And it's basically in the left hemisphere. The right hemisphere has less change. And we have also some rotational shear. I'm not going to detail this. But the problem is, of course, the two cortices, let's say V1 and V2, are not being rewired completely. The anatomy cannot shift in such a short time. So we must find the functional changes either by feedback or by within area activity that is affecting this functional shifting of the visual field and deformation. And the conclusions are we present a Bayesian thin plate warp with Beltrami regularization to measure the transformation of these connected field retinotopic maps. Global to local attention has an upward shift. There's some rotation. There's a marked asymmetry. Left hemisphere is all the action. The right, very little. The use of very high field, 70 and more MRI will improve this estimate, and we can conclude that attention does warp functional retinotopic maps. Just two messages to our mesoscopic anatomists. More precision is possible with more mesoscopic anatomical data. We need to incorporate these priors in the estimation of the fields. And CF estimation, which I didn't talk about, this connected field estimation size, is a deconvolution problem that can only be solved with more anatomical a priori data, but the spread, lateral spread of the connections between two areas. And I finalize by saying that understanding the neural basis of visual selection, I think, is within reach and could contribute to the big problem of consciousness, understanding consciousness. So thank you.
Thank you, Professor Sosa. I now invite Professor Tutin to please deliver her talk.
Hi, everyone. Thank you for the invitation and for being here. I'm also from northeast region of Brazil. And today, I will talk about how changes in functional brain network can help us understand the altered state of consciousness. And this work was developed with my co-authors listed here on this slide from this University of Brazil. So, I want to briefly motivate the central question, the central idea of this study. We investigate clinical and altered state of consciousness, including, for example, working? Okay. So we investigate clinical and altered state of consciousness that include, for example, major depressive disorder and schizophrenia. And also we include meditative states, and we include also one group of persons that we call mediumship. And the people of this group report entering a trance, receiving spirit, entering a trance, and they have mediumistic abilities and they write automatic letters. So this act to do automatic letters we call psychography. So we also include pharmacological induced states with ketamine and DMT to understand the brain physiology of these states using functional brain network, specifically dynamic functional brain network, to do the brain characterization of these states and understand what kind of brain similarities do they have. And because we think that this approach can help us understand how psychedelic substances can be used to treat some psychiatric conditions. For example, we know that DMT has been used to treat major depression with success. So, why investigate altered state of consciousness using functional brain network? So we know that these states are a complex phenomenon, and some reports in literature are inconsistent, reporting both, for example, saying there is increase or decrease in the neural entropy, as well as integration or fragmentation of the cognitive network. So there is a gap, a lack of a comprehensive characterization that encompasses and unifies different types of altered state of consciousness that give us information about the patterns and similarities, what the fragmented approach that we have actually does not allow. So, an important aspect of this study is the variability of the EEG data set, but I think that I have a problem with my slides. So we use a public EEG data set that comes from different countries, but we also do our own EEG data collection. So it's not passing. One minute? Or pass one? It's not working. Okay. I'm waiting. Another? Okay. 1 minute, please.
Taisi, your slides are working fine here. So it's just give us 2 minutes.
Okay.
Okay?
2 minutes. Okay.
Is it in PPT or some other format? Taisi, sorry. Is it on PowerPoint or some other format?
PowerPoint.
PowerPoint, okay.
Yeah.
Just reload it.
So let's continue. Thank you. So an important aspect of this study is the EEG diversity that we used. So we worked with EEG datasets that come from public EEG datasets that come from different countries, as you can see here, but it also do our own EEG data collection. So this is just some photos from during the EEG data collection with mediumship people and meditation. And with this EEG dataset, we constructed a functional brain network using advanced methods, for example, time-varying graphs and motif synchronization. And this motif synchronization is a method that were proposed by Rosario et al. From my lab, and it captures temporal dynamics, including rises and fall EEG patterns, and it builds synchronization between brain regions over time. So let me try to explain a little better about how we constructed this functional brain network. So here in the letter A, we have the EEG signal, and we define a time window. So this time window sleeps through time, and from each time window, synchronization and adjacency matrices were built, and then the time-varying graphs were generated considering all synchronization that happened at time in the window. So finally, all these graphs are superimposed, and we have the weighted aggregated static network in the letter A. So when we have this aggregated static network, we can extract a lot of indices to evaluate the brain physiology. So you can see some results that we present from this point in these papers here. And after we did the brain characterization using functional brain network, we carry out the principal component analysis to evaluate the convergence and divergence between these states. So, but we just use only the global indices from functional brain network. So here in the panel A, we can see all functional brain network indices and how the group are clustering. But we decided to do another PCA using the 3 most representative indices of each axis. And the 3 most representative indices were probability of hub formation. These indices give us information about how the brain regions that were hub over time. The coefficient of variation of edges, the coefficient of variation of clustering, are indices that give us information about the brain topology. For example, if the number of edges, that means the number of synchronization between brain regions, changes a lot over time, it means brain stability. So if it didn't happen, it means brain stability. So the other indices, there are common indices, weighted degree, provides information about the time that 2 or more brain regions were connected over time. The edges, just the connections, or better, just the synchronization between brain regions, and the degree, the number of connections of that brain regions. So to understand better this result, we set up the biplot graph. And I just put 2 because we have no time to explore more about here. But in this graph, we can see for the probability of hub formation and the weighted degree, how the group are, the distribution of the group. And I want to draw attention to both these points. In the blue, the people during the use of DMT, and in the red, the people with major depressive disorder. So they are in the opposite position in the graph. It means that they had less brain network similarities between them. And this result didn't happen only from both of these indices, but 2 other indices too. For example, for the coefficient of variation of edges and clustering, they keep in the opposite positions in the graph. So the other groups that are in the center of graph, for example, schizophrenia, Isha meditation, Himalaya tradition, Raja Yoga from Brahma Kumaris, Gurdjieff tradition, ketamine, the people during the use of ketamine, and the people that have mediumistic abilities are in the center of this graph, but to other graphs too. And it means that they have the greater homogeneity functional brain network between them. So, in conclusion, we were able to identify similar brain characteristics. Sorry. We were able to identify common brain characteristics and also divergent characteristics between these states. And the people that have mediumistic abilities are constantly compared with schizophrenia, but the group that we worked had no associated pathological characteristics, but although we could find some common brain characteristics between them, as you can see here, cluster coefficient and probability of field formation, but of course, others divergent brain characteristics between them. So, to meditation, an interesting outcomes also, because we worked with 4 different types of meditation, and 3 of them that practice focused attention, and they had the similar brain functional brain network characteristics, as you can see here, in contrast to Isha tradition. And to DMT and major depressive disorder, I couldn't put all the results here, but we could see that the functional brain network during the use of DMT improved this functional brain network. For example, have more efficiency, more brain efficiency. In contrast to results that we found to major depressive disorder. So our results can support the use of DMT to treatments of depression. And I just want to say, we want to encourage other researchers to continue exploring this area to help us understand more alterity of consciousness, the brain physiology, and the human consciousness. Of course, collaboration, make more collaboration. And he is my team, my research, or our research team from Labios, UFBA. And of course, we have others researchers that is not on only UFBA, Federal University of Bahia. And I want to say thank you so much. It's an honor for us to stay here. And you can find me here. Thank you.
Thank you, Professor Tutin. The next speaker, Professor Olopade, is with us online. I now request Professor Olopade to begin his talk.
Okay. Thank you very much. I hope I am clearly visible. Greetings from Ibadan in Nigeria. Apologies, I was really looking forward to come to India, but the change of timing affected my schedule. So I'm to give a brief talk on environmental pollution and the brain, and what we have learned from the lab, and what we have learned from the field. The Nigerian's Niger Delta is the major place of oil production in Nigeria. Oil exploration still accounts for over 80% of the GDP. And because of that,
Sorry to interrupt, Professor. Could you please go on a slideshow mode? Yes, it's visible. You have 10 minutes.
Okay, so
I will remind you after
yes.
8 minutes. You can continue.
Okay, thank you.
Thank you.
So there's so much pressure on the oil production, both from the local and international companies, with a population of about over close to 40 million in the Niger Delta area. So there is a constant pressure on oil exploration, leading to both accidental and intentional destruction of pipelines and constant burning of crude in this congested, population-dense ecosystem. So one of the major problems of Nigerian crude oil is that a main contaminant of the crude oil is vanadium. Vanadium is atomic number 23, and Nigerian crude oil is abundant with that, and vanadium is released into the ecosystem. Now, this work in 2009 from Iowa had already shown scientific evidence that vanadium is an environmental toxicant that could predispose to Parkinson's disease due to protein kinase C delta dependent oxidative signaling mechanism. So we know that even though the study of the population in the Niger Delta and the incidence of neurodegenerative disease is quite low, because despite the fact that oil comes from that area, the poverty level in that area also remains high. So we have been studying the Niger Delta region, looking at African giant rats in the local population as a sentinel to describe what could be going on in the brain. Unsurprisingly, there is increased vanadium content in the brains of the giant rats that are living in the Niger Delta compared to other regions of the country. So we have taken a two-pronged approach. We have looked at the rats living naturally in Niger Delta, and we have also exposed rats in the lab to vanadium to look at the neurotoxic effects. So in this graph, where we'll measure on the green is showing Niger Delta, blue and red is showing two other different regions of Nigeria, and we looked at the vanadium content in the kidney. You can see it in the rats. This is the African giant rats. So these are natural sentinel rats that are ubiquitous all over the country in Nigeria. So you can see the level of vanadium in the kidney, in the bone, compared to other regions in the liver, in the brain, which is what we are interested in. So you can increase vanadium content, and expectedly in the lungs is quite high because most of these metals come into the animal system by inhalation. So this is what you have in the substantia nigra of the dopaminergic staining of TH neurons in the substantia nigra in rats in the forest region of the country, intact intact expression of the TH neurons, but you can see a pallor, a myelin, a neuronal loss in the Niger Delta region of the rats. And you can look at it at the high magnification. You can see the pallor and pycnotic presence of neurons in the substantia nigra, supporting the fact that vanadium exposure may be predisposing to Parkinson-like symptoms or Parkinson's disease with the people in this population. So now in the lab, we now just exposed the rats from the forest. Okay, these are the rats from the forest, which did not show any evidence of TH loss. We now exposed a subset to vanadium, and another subset served as control. You can see the control, and then after vanadium exposure, showing both from the field and in the lab that vanadium could be contributing to loss of dopaminergic neurons in the substantia nigra. Okay, so in doing a long-term exposure, we actually decided to expose laboratory rats for a lifetime to vanadium. So from postnatal day 30 till about 12 months, okay, just to see using a laser ablation induced corpuplasma spectrometry to see how vanadium moves in the brain over time. So these are control rats, okay, from 3 months to 18 months. And when you look at the heat map of blue being absence of vanadium to red, showing high vanadium content. So vanadium is not a natural constituent of the brain. But when we expose rats systematically 3 times a week over the 18 months period, we could actually see the changes and the increase in vanadium. Exposure was through the intraperitoneal route. So definitely vanadium easily crosses the blood-brain barrier. And after 18 months, you could see the high presence in the olfactory bulb, in the occipital cortex, in the cerebellar cortex, and in the medulla. So for animals that were exposed for 3 months and the vanadium was withdrawn, we could actually see that there was withdrawal of vanadium, but vanadium content still remained for about 15 months after the withdrawal, even though at a very low quantity. Okay, so using this same specimen that we use for this test, what is the risk factor? Animals that have been so exposed to vanadium over a 15-month period, what's the risk factor to these brains having expression of amyloid, tau, and alpha-synuclein? So showing that chronic exposure to vanadium over time could be a predisposing factor to neurodegenerative diseases. So we recently published this, showing that chronic vanadium exposure promotes aggregation of alpha-synuclein, tau, and amyloid. Okay, so let me skip all this. So these are the control animals, 6 months, 12 months, and 18 months. And we could see minimal, hardly any expression of amyloid. But in the vanadium exposed, this is the hippocampal CA3, you could see vanadium exposure from 6 months, it continues to 12 months, and continues to 18 months. So the more the animals were exposed to vanadium in the lab, you had more increased expression of amyloid beta. Okay, then this is tau in the corpus callosum. You could see, we couldn't see tau expression at 6 months, 12 months in control animals. You start to see some expression of tau at 18 months. But for the animals we were exposed to vanadium, by 6 months, we already saw tau expression in the corpus callosum increasing at 12 months to 18 months. And then looking at alpha-synuclein expression in the rats in different regions, the frontoparietal cortex, the hippocampal CA1, and the hippocampal CA3. We could see with particularly from 12 months.
Sorry to interrupt. You have 8 minutes. You're done with 8 minutes. You have 2 more minutes to wrap up. Thank you.
Thank you. Thank you very much. So you have this increased alpha-synuclein expression at 12 and 18 months. At 12 and 18 months, it already started around 6 months in the CA3. So showing from laboratory simulation that increased prolonged vanadium exposure leads to alpha-synuclein deposition. So lastly, we look, there's a big industry because of so much plastic waste, metal waste. Nigeria has started creating a lot of dump sites all over the country. And those dump sites have become like habitual areas. Big industry for unemployed people to get jobs. People virtually work the whole day on dump sites. And we decided to also use these rats as sentinel that what are the neurotoxic effects of working on dump sites over a long period of time. And then looking at the same African giant rats from the forest compared to the dump site, we could see here a loss of Purkinje neuron arborization in animals that that were obtained from the dump site compared to the forest, increased tau phosphorylation in the dump site compared to the forest, increased alpha-synuclein expression both in the cortical area and in the cerebellum. Demyelination was occurring in the brains of the rats of the dump sites compared to the control in the forest. And you could see submeningeal myelin deposition was seen in 40% of the rats in the dump site compared to those in the forest. And strikingly, at the substantia nigra, we saw tyrosine hydroxylase neuronal degeneration of the TH neurons from those on the dump site to those in the forest. I do not have enough time to show metal quantification, but you could see that the red is the dump site, increased metal depositions in the soil, in the environment, in the vegetation, in the dump site relative to the forest regions. Okay, constant environmental monitoring as it relates to brain health is crucial and must be invested into. There has to be regulation of activities that lead to heavy metal pollution, and this is imperative for mental health. With increasing dump site-related industries, the occupational health hazards should be noted, and advocacy actions to get workers better protected needs to be put in place. Thank you very much for listening. I appreciate the students who have been doing this work, and our collaborators in Göttingen and in Verona that we have been working with on neurodegeneration, and also in Canada. I also want to appreciate those who have funded my work in time past and continues to fund the work we are doing. Again, I feel so sad that I couldn't come to India for the first time, but I'm happy to be presenting online. Thank you very much.
Thank you, Professor. I would also request you to stay with us online for some more time because we have a discussion session now. And I would also request the speakers of this session, Professor Sosa and Professor Tutain, to join us on stage for the discussion. We are running a little late, so I would request the audience to please limit your questions. Hello. Are there any questions?
Hello. Yeah. I have a question, Dr. Tutain. So you said that you measured improved brain efficiency in DMT-treated major depressive disorder patients. DMT treating, right? You saw improved brain efficiency. So how did you measure that?
Yeah. So I could put all results here because we don't have time, but we could see during the use of DMT, the functional brain network increased efficiency, decrease the path length, is the path, the path that the one information passed to one part of to other part of the brain, like this, and we could see another results that during the use of DMT, the brain are more efficient with all other indices that we investigate also. But in major depressive disorder, the literature also report that the functional brain network in major depressive disorder are random, not efficient. The path length is large, and is this. So in Brazil, we have an institution from northeast of region that use, for example, DMT to treatment major depressive disorder that they can't use another drugs to treatment.
In your data, does it suggest that it makes the connections more denser, or does it make it lesser but more dynamic?
Not intensive. Let me see if I understood your question. The functional brain network is not more intensive, but more efficient. Yeah? Thank you.
This is a question for you, Professor Sosa. You had initially modeled your receptive fields using Gaussian filters. Now I'm wondering, given the nature of low-pass filtering that the visual system initially does, is Gaussian filtering, the Gaussian approximation, the right way to do this? Or should we look at, let's say, the contrast sensitivity function and see what bandwidth properties they have and model the receptive fields based on those?
Very, very good question. That's a topic of active research. Some people believe that the connected fields or the population receptive fields are best modeled by an isotropic Gaussian, but there's evidence that there could be asymmetry. The question we tackled in our work was to divide the problem in two stages. First, determination of the center of the connected field, and then work on the problem of the overall shape and the size. This is an ill-determined problem because the problem is that as receptive fields get bigger, they start overlapping more. And this leads to a problem of indetermination in the methods. That's the reason we decided to concentrate on the organization of the maps, measuring the centers, and then leave for a second stage. And we believe if we have more a priori knowledge incorporated into the estimation methods, this is the deconvolution problem. With more data, we cannot separate the contributions for different receptive fields. But I agree, that's an oversimplification considering that an isotropic Gaussian is adequate.
Thank you.
Thank you. Thank you to all the speakers. Thank you, Professor Olopade, for joining us online. That was a really interesting session. For the 8th session, it is my pleasure to introduce our featured speaker, Professor Jing Zhang, who will be joining us online, followed by Dr. Poonam Thakur and Dr. Hiya Ghosh. The session will conclude with a 10-minute question-answer session. I would now like to request Professor Jing Zhang to please deliver his talk.
Hello, can you guys hear me okay? And also with my PPT?
Yes, Professor, you are online. I would request you to please go on slideshow mode.
Great.
You have 15 minutes. I will give you a reminder after 13 minutes. You can please proceed.
Thank you very much. Hello, everybody. First, I really apologize for the fact that I cannot come here in person. I also want to thank Mohan and Richard for inviting me to participate in the presentation. So the title of my talk is Progress in the China Brain Multi-Omics Atlas Project, CB map. So why human brain? Omics and the mapping. I think I don't have really answered the question for you guys, since you all know. But major point I try to make here is that even in monkey, the size of the monkey brain is so different from a human. Not mention the complexity of the two species. And also at the moment, most study being done in rats, mice, rather than in monkey or even human. So in China, we developed a program called CB map. That's in the end of the 2023, where we start collecting brains across China from 6 major human brain banks. At this point, we have close to 2,500 brains collected. But in the first the period of study, we only include roughly 1,200 brains. In this white paper, we propose that we'll be doing all the studies, including genomics, epigenomics, transcriptome, proteome, and metabolome. At this point, we already finished over 1,000 brain with all studies I mentioned above. So to describe a little bit more about the human brain dataset. In this dataset, the age group is quite diverse. And also we have different disease types, including Alzheimer's, Parkinson's, and TDP-43, vascular dementia, and also a lot of the normal aging matched controls. The quality of this study is rather interesting, saying that the ring value or the time of the post-mortem delay is quite comparable across the globe, including the one being demonstrated most, like GTEx and ROSMAP. Our dataset, in a way, is more advantageous in terms of ages, the spread from like year 1 to over 100. That is very important for the age-related research. The first paper coming out this the CB map is the hopefully will be accepted soon by Nature Aging. We just revised the paper. In this paper, we discovered that about 1/3 the CPG correlate with aging, and among that is significantly a driving force for the development of Alzheimer's disease. This area, as you know, is very, very competitive. While our paper was under review, this paper already came out in Cell, using the quite a bit in the brain regions to describe the epigenomic alterations as a driver of the Alzheimer's disease. So it's a quite interesting area. Everybody is interested in it. In that regard, people already gone beyond the epigenome, move on to proteomics and also PTM, the post-translational modifications. The paper just came out last month from Penn's group in Cell, describing several key points here. One is that all neurodegenerative diseases are composed of multiple subtypes, and which in a way is already known, but now they have molecular signature for each of subtypes. The second point is that the different diseases have some shared common pathways, especially for microglia lysosome pathways. And they propose that in the future, moving forward, the study should probably include more of the biomarkers for subclassification and also for the engaged therapies. In a way, we are a little bit behind. We have a paper under review now, doing exactly very similar thing, but we're using roughly about 800 brains, studied the various PTM in Alzheimer's disease, Parkinson's disease, so on and so forth. And hopefully we'll have some of those publications come after soon. Now, the group, I don't think I have emphasized too much. The brain, the human brain is so different from the other organs in that each brain region responds for different things. And neurodegenerative disorders are regional specific. Like Alzheimer's will be like a cortical and a hippocampal issue, whereas Parkinson's disease, Huntington's disease will be more basal ganglia. So we published a paper last year working on more sensitive brain region for Alzheimer's disease, that's the hippocampus. I don't think we have to explain the hippocampus to this group. In this study, we compared patients with Alzheimer's disease with those of age-matched controls, and we define a single cell resolution, different regions of the hippocampus where what disease mediate changes based on the cell types, whether it's neuron, if the neuron excitatory versus inhibitory, if it's glia, is it microglia, oligodendroglia, or astroglia. And we define that based on the subregions, not as a whole of hippocampus. We have also focused on a single cell resolution, each cell type, alterations around the plaques, which is diagnostic the feature of Alzheimer's disease. Now, now I want to bring something different to share with you guys. So this will tell you where we're going. For that study, for the paper published in Neuron, we only compare 8 Alzheimer's brains with 8 healthy control brains that cost us a fortune. So if you want to do this whole brand and with a lot of brands investigated, financially it's not possible. So we try to bring in the cognitive AI. This a concept talked really well by the Jensen Huang. I don't know you guys listened to that talk early this year or not. Essentially, they try to use the he was talking about autonomous driving, but essentially use generative AI to simulate all thought possibilities, so that they don't have to do real simulation with the real what's going on the street. So I think we can do very similar thing for our the omics study, especially for the single cell omics study. Now except keep doing the different regions of omics, we also want to bring in the multimodal fusion front. So I think Richard talked about yesterday, and Professor Sosa also talked something earlier about visual cortex. So we are doing things very similar, where we try to get MRI image ex vivo. This study here showing with 7 Tesla scan of the brains before the brains were they pathologically processed. And then we try to merge the MRI image with code registration pathology in addition to those immunohistochemical studies we do normally. But the problem, apparently, not problem, it's challenges that we are facing clearly is the alignment. I think both the professors already they addressed a little bit. So it's not really fusion issue, it's the alignment. How can we align the MR image with the much more the high resolution of the pathology, not mention the molecular typing, right? So for the 7 Tesla, we already published. Now we have 14 Tesla. But even at a 14 Tesla MRI, the resolution is only 100 micrometers. But what about the pathology? That's 1 micrometer, right, or less. And for molecular, that's even more, the detailed analysis. So how to analyze them together, put them all together, that's a daunting job. That's what we're working on anyway. But I should emphasize that the pathology, not just a big pathologist, I really want to emphasize pathology, but it's true though. To really align everything together, we have to be aware the detailed histology, because histogenomics is the centerpiece of all multimodal the fusion. The other thing we try to do is try to move beyond just omics. The reason for that is we spend so much money talk about omics, whatever change, what does it mean really, right? So that's what we are working on next. So in that, I'm trying to bring you guys to the Alzheimer's, the front for the Alzheimer's diagnosis. Now, I think everybody knows that the Alzheimer's disease is a huge burden to the society. China, for instance, we have probably the biggest number of Alzheimer's patients globally. at the moment, we often say we have to do evidence-based medicine, right? We have diagnosed Alzheimer's disease not by the tables or the questionnaires. We have to use the biological evidence, including like PET imaging, MRI, or the analysis of CSF biomarkers. But as you know, whether it's PET or CSF, they're all invasive, little bit invasive, or more expensive than typical people can accept. And if you want to use PET imaging or CSF to screen patients for early diagnosis, that's just not possible. So getting a blood test is really key. Now, the good news is that last year, USA, the FDA just approved an assay, the p-tau 217 over A-beta 42 for the diagnosis of Alzheimer's disease as a sort of like screening test. But I can tell you that this assay is good for the established Alzheimer's disease, but not for early Alzheimer's disease. In addition, the assay cannot differentiate the Alzheimer's dementia versus other type of dementia precisely. I'll give you an example. If somebody with stroke, the p-tau 217 will increase. For people with ALS, amyotrophic lateral sclerosis, you're going to have increase the p-tau 217. So it's sensitive, but not really specific enough.
Professor Zhang, sorry for the
issue,
interruption. 13 minutes are up, but you still have 2 minutes to continue. Thank you.
I'll be done quickly, okay? So the way we did is try to, as I mentioned, neuron paper, we identified a whole bunch network alterations, right? But we only focus on the part that can be transported across blood-brain barrier and to be detected in the blood. We have established pipeline over the last 10 years also where we can use a blood sample where we can capture what's derived from central nervous system, whether neuronal or glial derived exosome, and then we can measure biomarkers in those patients. So in that neuron paper, we actually, after discovering different pathway alterations, we also use human samples to achieve not only Alzheimer's diagnosis, but also differential diagnosis between Alzheimer's disease and non-Alzheimer's dementia. So to basically try to summarize that, we already established a platform where we can combine the omics study to blood test. Now, for the future, I think we try to do the multimodal analysis from the MRI to pathology to omics and then to blood test. What we try to do is try to do precision diagnosis and precision therapies. Now, I try to bring you on the lung cancer treatment and diagnosis. Years ago, we only really divided lung cancer into like squamous carcinoma, adenocarcinoma, and small cell carcinoma, right? But nowadays, we don't do that anymore. And when we see lung cancer, we say, okay, what genes turn on? What genes turn off? And then we gear towards precision therapy. I think in the future, for neurodegenerative disorders, we're going to have multimodal fusion, multimodal alignment, including clinical MRI image and the pathology biomarkers, so we can subdivide the patient precisely, and then we gear towards targets for that person specifically. This is all my talk, and this is my group where we worked on the projects from various angles. Thank you very much.
Thank you, Professor Zhang. I would request you to stay with us for some more time because we have a Q&A session at the end of this session. I would now like to invite Dr. Poonam Thakur to deliver her talk.
Good morning, everyone. First of all, I would like to thank the organizing team for inviting me here and for their wonderful hospitality. So I work at ICER Thiruvananthapuram, and you can see the very, very pretty picture of our campus here, arguably one of the prettiest campus in this country. And our lab there works on Parkinson's disease. By now, you all know it's an age-associated neurodegenerative disease, majorly affects old people, and also happens because of the degeneration of neurons in this tiny region of midbrain called as substantia nigra. And when these neurons are lost, what happens? People suffer from motor disorders. So they have tremor, rigidity, difficulty walking, difficulty stopping, and they eventually become progressively worse over time. And previously, it was believed that Caucasian populations have high prevalence of the disease, but now we know in India the incidence is increasing very rapidly, and problem in India is that disease onset is at least 10 years earlier than the Caucasian populations. So people have to live much longer with the disease in our context. All right. So in our lab, we focus on multiple aspects of Parkinson's disease. We are a basic biology lab. We work with mouse. So we try to work with the animal models that can mimic the disease, and we rely on progressive models, which are based on alpha-syn overexpression directly in the brain region of interest. And using this model, we try to understand various kind of neuroprotective strategies that we can evaluate. Also, we are very interested in looking at how other comorbidities can impact the disease, because diabetes or metabolic syndromes are another very important issue prevalent in our country, and a lot of people who will be diagnosed with disease will often have these comorbidities. So how their presence is going to impact them and what changes it causes in the body is another big focus. And while the impact of metabolic disorders on neurodegenerative disease has been quite well studied in recent years, we have recently also found that brain disease without anything else can also directly cause some metabolic alterations in our body, and that is what I'm going to talk to you about today, which is the last part. All right. So alpha-syn, which is the major protein known to misfold during the disease, is very well known also that it can associate with the lipid membranes under certain conditions and form some structured, some ordered structures. And while traditionally known as a protein aggregation disease, in the recent years it is clear that these aggregates are not solely composed of proteins. They are consisting of a large number of other components such as broken organelles, fragmented membranes, lipid droplets, and so on. So therefore, in recent times, there's a view coming in that these are not proteinopathies. In fact, they should be viewed as lipidopathies. And therefore, lipids could have a very big role to play in the disease. And in fact, the failure of the therapies targeted to only protein aggregation, refolding, and so on has not worked, also pushes us to look at the other alternative explanations for the disease. And there's a lot of data from post-mortem samples and human studies that tells us that indeed there are also global level changes in the lipids coming in. However, when we look at these post-mortem samples, they are usually coming from people in very advanced stages of the disease when they have lived with the disease for 20, 30 years. What changes we observe in them may not tell us what is happening in the early stages of the disease, and what has come in there, was it something that actually was pushing the disease, or this is now an epiphenomena of the disease that has already happened in the brain. Also, in the living people, we cannot take the brain tissue out. We are only reliant on biofluids as a biomarkers. So if we take a blood sample from a patient, will that have any indication of what is happening in the brain? Can we match it? Of course, we can't do it in the humans. And so for these things, we decided to work with our animal model, where we can draw blood and the brain tissue both at the same time and try to see where the matches or dissimilarities lie. Okay, so that model, as I described earlier, that we use is a mouse model. We take this alpha-synuclein purified protein, we make it into aggregates in the test tube, and then we inject in the brain by surgery on a single side of the brain. Then we let these mouse sit over a 3 months period so that they can get the pathology all over the brain. We monitor them regularly with the behavioral testing, and all of this work is done by my PhD student, Unnati Agarwal. Okay, so when we look at these animals, we see that, okay, my legend is missing here. All the white here is control, all the gray here is PD, all the open transparent circles are males, and all the red ones are females. So what we see is that there is a mild decrease in the general locomotor activity of these animals over time, and also we see some gait deficits in these animals. So what we see here is changes in the stride frequency. That means animals are taking a lot more steps to walk the same distance, so they are becoming shorter and shorter, a very typical feature in human PD as well. Okay, and our goal here was not to create an advanced model of PD, but to mimic only the very, very early stages, so we can start to see the lipid changes that are likely to take place in this early stage. And after 3 months, we sacrificed the animals, we look at their brains. So we did the coronal sections in this substantia nigra region, as well as in the striatum region. This is the region where these neurons send their projections into, and this is the region also responsible for driving the motor phenotypes. And you can see here, marked by the arrow is the injected side, and in the PD brains, we can see a clear loss of dopaminergic neurons stained by tyrosine hydroxylase. And when we count for them, both male and female mouse show about 30-35% neurodegeneration here. On the other hand, the projections in the striatum are not yet lost at this point, again indicating that this is still an early stage of the disease. Okay, so now that the model is established, early stage is established, so we looked into the lipidomics changes that are, before I move there, we also could see very well the aggregation of alpha-synuclein protein in these brain regions, and this was also very clearly high in both male and female mouse brain. Okay, so then we looked at the lipidomics changes happening in these midbrain regions, and in interest of time, I'll not describe a lot of this heat map, but what it is showing is just pay attention that there is control females, PD females, control males, and PD males. And as you can appreciate here is that there are some lipid species that are changing in the males, but in females, there is no change in those species. On the other hand, there are some other lipid species in which female mice are showing much bigger change, but males are not showing much big change. So that means, despite the fact that their neurodegeneration was exactly the same, there are very clear differences in the kind of lipids that are impacted in male and female mouse. Okay? And then we also had a plasma or blood drawn at the same time, and then we did lipidomic analysis on that. Of course, the lipid species that exist in blood are very different from the species that exist in brain. So there is not going to be 100% match in the lipid species, but nonetheless, we could find several common signatures. But what does this mean for humans? So we also collected blood from human PD patients and did a similar metabolic analysis, and then we could find several species common. Just to rush through the data, when we did the pathway enrichment analysis, what was clear to us, the biggest common denominator across species and across the tissue samples was changes in the fatty acid biosynthesis. So now these fatty acids, what they do in the body, they get converted into diacylglycerols, which get further converted into triacylglycerols. And inside the cells, they can form a dynamic organelle called as lipid droplets. They will usually have these triacylglycerol and sterol esters sitting in the middle, surrounded by a lipid monolayer. And in the normal conditions, they will not be there quite a lot, but under stressful conditions for the cell, they will typically show up. And we started to look into, okay, if we have such dysregulation of these fatty acids, do we see any lipid droplets in the brain? And we observed in the neurons a mild upregulation of these lipid droplets. It didn't reach any statistical significance, so we were a bit disappointed why these neurons are not showing anything. But when we carefully looked at these images, we could see a lot of droplets outside of the neurons. So we started to investigate if they are by any chance in the other cells. And in the astrocytes, we could see the male mouse showed quite a lot of these lipid droplet accumulation. And also in the microglia, we could really see a lot of this accumulation, both in the male and female mouse. These microglia that we observe here, or the astrocytes or neurons, they are all coming from the brain. what does this mean for human? Since we can't access the brain, what to do? In the microglia, since we are observing a lot of lipid droplets, we all know microglia comes from the, not from the neural tube, but from the myeloid origin. So from patient blood, it is possible for us to get the myeloid origin cells. So we once again went to the patient samples, collected the macrophages, and in there also we could see a very huge upregulation of the lipid droplets in the macrophages. So this is something you can easily get from a peripheral tissue and see these changes. And in interest of time, I'll just wind up. Some of these results are available in this preprint that you can look into. And we have also tried to see if we could target this for therapeutic interventions. And with the lipid-lowering drug, we could see a very clear neuroprotection in both male and female mouse. And this is an ongoing study, and we are digging deeper into the mechanisms here. And with this, I would like to thank my entire team for putting in the work done behind this study. My collaborators from Sri Chitra Thirunal Institute of Medical Science and Technology, who helped with all of the patient work. Siddhesh Kamath from IISER Pune, who helped us with all the lipidomics work. And of course, all the funding agencies for the monetary support. Thank you.
Thank you, Dr. Thakur. I would now like to request Dr. Hiya Ghosh to please deliver her talk.
Good morning, everyone. I'd like to start by congratulating Rija and her team for putting together such a wonderful program, and also thanking her and Mohan for this opportunity to present our work. So today I'm going to talk about engram neurons and the study from my lab, which we think reveals a novel aspect into it. So what are engram neurons? These are the bunch of neurons that fire when an experience is being experienced, so during encoding. And wonderful studies have shown that if you ablate these specific neurons, that can lead to ablation of memory, and conversely, if you activate these set of neurons, then you can regain memory. However, an interesting point is that not all neurons that are firing during encoding actually participate in recall. So the question is, what is it that decides which neurons out of the engram neuron would participate in recall? Is it an entirely stochastic process, or is there a molecular logic to it? In the last 2 decades, studies have come to converge into the thinking that memory is a dynamic entity in terms of its formation, its storage, and retrieval. And very recent studies with high temporal resolution, which is in minutes to hours during learning, show that actually an engram, a memory engram, consists of several sub-ensembles, out of which only few or specific ones participate during recall. There are other studies that have shown that the pathways within the hippocampus that carry the encoding information or signal for the memory are also discrete. And more further studies show an observance that out of the engram neuron, the pattern of activity is not all the same. There is an ensemble that would exhibit more dynamic, plastic kind of activity pattern, whereas other ensemble may show a more rigid or selective pattern. So with all this, we ask, what is it that governs the retrieval, or what is it that governs assignment of neurons to distinct phases of memory? And could there indeed be a difference in the encoders and the recallers within the memory ensemble? And why is this question important? Well, apart from understanding some basic biology of memory processing, it also allows us to perhaps understand diseases of memory better. because in most cases, in most diseases of memory, it is the retrieval of information that seems to have been affected rather than the formation of memory. And so if memory stabilization and retrieval involved specialized cellular processes that could only be imparted by specialized neurons, then we indeed need to learn more about the cell biology. And so our interest in memory engram really stemmed out of a study, and as a side observation in this study, where we were using this particular CAM kinase 2-based system for genetically labeling principal neurons. And in doing so, we observed that this is the hippocampus. Oops. This is the hippocampus in the brain, and if you look carefully in the CA1 region, what we see is that not all neurons are labeled, and in fact, a significant percentage, about 30 to 40% of the CA1 neurons are not labeled by this method. So we wondered if these population of neurons could represent a specialized population of its own. We looked more carefully into different parameters. First, at the baseline level, just by doing a CFOS staining, we find that the unlabeled neurons, which are represented in blue, and we call it type B, are always more active compared to the CAM kinase alpha-driven labeling principal neurons. And then these neurons also had a higher baseline calcium compared to the A-type neuron, and additionally, more profuse dendritic arborization compared to the A-type neurons. So by multiple features, these populations seem to be different from the majorly studied principal neurons in the hippocampus, which was CAM kinase alpha positive. So the next question is, are they functionally different? So for that, we employed a fear memory paradigm. This is because contextual fear learning heavily depends on hippocampus. And in this paradigm, you take a mouse and you put it in a context to familiarize. And next day, you bring it back to the same context. And at the end of its exploration, you give it a little mild foot shock. And then it associates this context with an aversive experience. So next day when you bring it back, it's able to recall this memory and freezes even in absence of a shock. So with this, we asked, how are these two sets of neurons doing? And now that we can label it, we can see the type 1 in green and type 2 in blue. And you can see that, as expected, compared to the context, during encoding, both A and B fire more in number. So they are participating. However, during recall, we see that while A participates in similar number, so there is no significant reduction from here, and it's a significant increase from the context. In the case of B, there was a significant reduction during the time of recall, thereby suggesting that at the time of recall, it is the A type that is being preferentially recruited into the memory engram. So now we want to test if this is causally correlated. So for this, we employed this method in which you can send in a designer drug receptor into the choice of your neuron to be expressed in the choice of your neuron, and then send in a ligand to temporarily switch it off. So with this viral strategy, we decided to inhibit type A or type B either during the time of encoding or during the time of recall. And when we inhibited type A at the time of recall, as expected, we saw that the memory was lost. However, if we switched off type B, nothing happened to the memory. And conversely now, if you turn off A during encoding, you see that there is no effect on memory. However, if you turn off B during encoding, you see a memory loss. So this clearly establishes that there is a functional bifurcation within the memory engram, wherein type A cells are essential for recall and type B are essential for encoding. So now, what is it that enables or underlies the specialization of A and B that would allocate them for these specific phases? So for this, we went back and picked cells from the brain that were A-type and B-type and did transcriptome. And to our pleasant surprise, there were hundreds of genes that were significantly divergent between these two sets of neurons, and looking at the nature of the genes that are A-type specific, one could tell that the nature of the A subtype was more amenable to structural regulation, whereas that for the B was more amenable to synaptic activity. So this shows clear biological features that are different between these two subsets that participate in memory engram, but are differentially critical for the writing part and the reading part. And so with this, we kind of come upon a molecular logic for neuronal allocation in the memory engram, where structural and molecular features of individual neurons kind of determine their allocation to distinct phases. And why learning about this property is important is because this can provide more insights into the cell biology of how neurons encode versus how neuron recall, and that can in turn lead to more targeted interventions into the diseases of memory. So with that, I'd like to thank my students. This entire work was done by a very talented PhD student, Dheeraj Sangara, and also all the funding organization and all of you for listening. Thank you.
Thank you, Dr. Ghosh. I would now like to invite the speakers for this session, Dr. Ghosh, Dr. Thakur, and Professor Zhang, to join the Q&A session. In the interest of time, we'll limit the session to only 5 minutes. Thank you.
Hi. Hello. Yeah. Really enjoyed your talks. Thank you. I have a question for Hia. The fantastic two populations that you have found, now that you have transcriptomic differences between the two in the rodent, have you looked as to see whether there are two populations in the human as well?
That's exactly what that's exactly what I'm here for. We've been in talks with Richa, and it would be easier now and possible, seems like, to do that.
Okay.
Hi, I have a question for Poonam. I'm wondering from your data where you've looked at differences between the male and the female mice. I'm wondering if, we assume sex hormones will definitely have a role, but do you think other steroid hormones may also play a role in the process in microglia versus the other neurons?
Yeah, so unfortunately in the microglia we have not really seen a big difference. But we are digging deeper into the things. There are few subtleties there which are showing differences. But if the difference comes exclusively from sex hormones, that we cannot, as of now, say.
I have a question for Dr. Ghosh. Can you hear me?
Yeah,
I'm here.
Yeah.
So you're showing the right hippocampus in, I think, the dorsal rostral part.
Dorsal, yeah.
Yeah, dorsal and rostral. Did you look in the ventral too?
Not yet.
Not yet.
So most of these associations from previous studies which use the CAM kinase alpha also allocate the dorsal as the more context determining part. However, there are also studies suggesting that the entire network is important. So if you ablate the dorsal, the ventral will take part. But we haven't done the ventral studies yet.
Okay.
Can I, Poonam, this is for you. I couldn't quite get the exact lipid changes, obviously, which lipids change. But from that data, can you suggest, do they indicate some sort of specific etiology for the disease? Does it, is it enriched for certain organelles?
So what we saw was there was a decline in a lot of phospholipids, and there was increase in a lot of fatty acids. In addition, very interestingly, we could also see in the brain upregulation of glucosylceramide species. Now that is interesting because GBA mutation is well known to cause PD. We don't have a genetic model here, but despite the fact, the substrate of that shows up as very highly upregulated in the brain tissue. So these were some very strong indicators that it is indeed saying something about the real brain pathology.
My question is for here. Here, please, have you looked at if there is any kind of recurrent inhibition between these two subtypes of neurons?
So, so this is the first report, and we are definitely following up on those lines. An interesting revelation from our transcriptome is that the encoder types, which kind of drop out from the recall ensemble, they express NPS4, which is known for switching off the neurons. So they are more amenable to, or they get more inputs from inhibitory neurons. So I think there is something about that.
Great, thank you.
Hi. Hi, I have a question for Dr. Thakur. I really enjoyed your talk. So we know about the increasing relevance of the gut-brain axis in Parkinson's. So I was wondering, with the lipid droplets mechanism that you're trying to understand, do we know anything about if this is happening only in cells in the CNS, or is it happening in the periphery as well?
We have looked at liver as of now, and in liver, we are actually able to see alpha-synuclein going there, as well as increase in lipid droplets in these PD animals. So definitely, it goes all over the body. I do have some indication in kidney as well, but I don't know yet. But it seems to be going elsewhere.
Thank you.
In the interest of time, we will stop the Q&A here. If you have more questions, please, you can chat with them over the break. Thank you to all the speakers. Thank you, Professor Zhang, for joining us. We now have a short tea break. We will resume our session at 11:10, so I request everybody to assemble here by 11:05. We request all the delegates and the external participants to please head to the back of the hall for the tea, and all SGBC staff to our center, SGBC, for the snacks. of the day. And for this session, it is my pleasure to introduce our featured speaker, Dr. S. Suresh, followed by talks from Dr. Ilya Bakulin and Dr. Lee Han. The session will conclude with a 10-minute Q&A session. I now invite Dr. S. Suresh to please deliver his talk.
Good morning. Thank you very much for inviting me. Amongst scientists, I'm an end user of all the research that you do, and hopefully we should make some difference in the patient's lives. So I'm going to take you through a fetal neurosonography and how the technology has actually changed over the years and what we are able to do now. So the plan is to show the developing brain why it matters, looked at some technology, then look at normal anatomy, what we look for, brain anomalies, detection and diagnosis, advanced technologies like AI, and clinical impact and outcomes that matter. So we know that the developing brain is a remarkable transformation over 40 weeks, from neurulation to neurogenesis to guidification and connectivity. And the structure of the brain undergoes significant change over the several weeks. And as it does that, we are able to map it with ultrasound, especially with high resolution ultrasound at this point. Of course, MRI of the fetal brain is also in one of the modalities, but MRI is more difficult to do as compared to the brain ultrasound, which is freely available and accessible. Now, for imaging this system of fetal spine and brain, we use 3.5 to 5 megahertz transabdominal transducers, 2 to 9 megahertz transabdominal transducers, and 5 to 10 megahertz transvaginal transducers. When the head of the baby is down, we're able to access it transvaginally for a much higher resolution. We do get sub-millimeter resolution. Modern probes resolve structures as far as 1 to 2 mm. And we have real-time dynamic where we can observe fetal movements, breathing movements. The trick here is to identify a cranial window, either through the anterior fontanelle or the posterior fontanelle, or even axial imaging we can do through the sutures of the brain. And color Doppler, of course, helps to map the brain vessels. So when you look at the axial planes, we look at three main planes, which are called the transventricular plane, the transthalamic plane, and the transcerebellar plane. And these planes are very, very standardized, and every person doing ultrasound will necessarily do these planes and identify very specific landmarks. For example, in the transventricular plane, we look at the ventricular atrium. As you can, I don't think this will work. So you can see the atrial width is usually less than 10 millimeters, which is normal at all gestational ages. And we show the choroid plexus and the cavum septum pellucidum, and the genu of the corpus callosum is also identified at this point. Now, why we identify this? Because we can identify abnormalities by just looking at this one plane. We can suspect agents of the corpus callosum, and we can see whether the lateral ventricle is dilated or not. And in the transventricular plane, when you look at the autopsy specimen, we can find that we can look at the caudate nucleus and the parieto-occipital sulcus, and we can actually superimpose the brain so well that it will exactly fit to the anatomy at this point of time. And then we go on to the transthalamic plane, where we have the midline forks, which is labeled as 1, as you can see, and it's absent in holoprosencephaly. Cavum septum pellucidum, again agents of the corpus callosum, and then the third ventricle is a small midline structure marked 3, and the widening we suspect obstruction to the CSF. Then we have the hippocampal gyrus labeled 4, and then the lateral sulcus, which changes its shape with gestational age. That again, now we are starting to use it as an indicator for the developing brain. Then we go on to the posterior cranial fossa, where we have the transcerebellar measurements. We do the transcerebellar diameter because biometry of the fetal skull is very, very important to assess gestational age and progress also. And the transcerebellar diameter in millimeters is equal to the weeks of gestation in the second trimester. The cerebellar vermis itself, we do it by the sagittal view. It cannot be measured in the axial view. It's about more than 6 millimeters at 20 weeks. And the cisterna magna is usually anything more than 10 millimeters is considered as abnormal. The presence of the cisterna magna, some reassurance that there is no open neural tube defect in the baby, and the absence of cisterna magna is, or the cisterna magna is obliterated, then we highly suspect this baby will have an open neural tube defect with a normal cherry malformation. Now, when you look at cortical development and gyrification, the fetal brain transforms dramatically with gestational age. As you can see, the number of gyri is increasing with advancing gestational age. Less than 22 weeks, we call it as a lissencephalic brain. The brain is very smooth. And so the abnormality, the diagnosis of lissencephaly is very difficult to do before this time. But when it comes to 26 weeks, the brain, the gyration, the primary sulci should have formed. And if it does not form, then we call this as lissencephaly. By 28 to 34 weeks, we have secondary sulci, and then by 34 weeks, after 34 weeks, it's sort of near adult pattern which happens to the tertiary sulci. Now, of this, one of the important sulci we look for is the Sylvian fissure or the lateral sulcus. The lateral sulcus acts as a primary landmark for identifying migrational disorders. So at 18 weeks, it appears as a shallow depression. Then at 21 weeks, it becomes a little bit more deeper. At 25 weeks, it's still more deeper. And at 30 weeks, it transforms into a deep sulcus, as you can see here. So there you can see the lateral sulcus, which is shallow at 18 weeks, slowly becomes a little bit more deeper at 25 weeks. And then at 30 weeks, you can see like a pie-shaped structure, which is there. And ultimately, at 38 weeks, it becomes such a single line, which is there. The lateral sulcus is, this process is called as apercolization, and progressive apercolization is a macroscopic mirror for a microscopic cortical migration. And I think this is very, very important because this is where we start suspecting and start investigating the fetus more. Now, delayed apercolization, here's a classical example of a delayed apercolization. This is normal, and then here you can see as progressively as it increases, you can see how the apercolization has not happened even in the third trimester. Then let's come to the corpus callosum, which is a brain's great commissure, which is undetectable before 18-20 weeks, even though the framework for the corpus callosum is laid very, very early as 12 weeks. Now, we can identify with the corpus callosum the rostrum, the geno, the body, and the splenium. All these structures are identified, but only in the sagittal plane. And the axial plane is what we normally image. Sagittal plane, we have to do what is called as an extended neurosonogram to identify the sagittal plane. Genu forms first by 12 to 14 weeks, and the splenium and rostrum by 18-20 weeks. And it's complete by 20 weeks, which means as you can see here, this is what we call as the anterior echo complex. You zoom the brain right in the middle, you can see the midline hemisphere, the interhemispheric fissure, then you have the cavum septum pellucidum, then you have the genu of the corpus callosum is here, and this is the callosal sulcus which is there. The same thing translated into the sagittal image, you can see the entire corpus callosum, which is identified as a hypoechoic structure. And then you switch on the color Doppler, we can find the pericallosal artery and its branches, and this is very reassuring that the callosum is actually well developed. Now we can also do callosal biometry, and carefully if you look at it, the genu, body, splenium and rostrum, all of them you can measure at various gestational ages, and then in case there are any the alteration size, looking at dysgenesis, partial agenesis, other things can be identified at this point. Then here is just a video showing you in real time how does it look like. We are doing a sagittal view there. As I'm trying to go to the mid-sagittal view, you can see complete absence of the corpus callosum here. And this is the parasagittal view, switch on the color Doppler, and the callosal artery is completely absent. You can see the branches going up, but the pericallosal artery is absent. Also, the lateral ventricle by this time undergoes a shape change, which we call as the teardrop shaped ventricle, where you can see the posterior horn is dilated and the anterior horn is sort of looking as a pinched appearance. And it's a very typical appearance of agents of corpusculosum. Then we move on to neural tube defects. Now, one of the remarkable things that has happened in the recent times is the scaling down from 20, the second trimester scan, we have moved to the first trimester scan to identify neural tube defects, and over 95% of neural tube defects are identified in the first trimester. How are we doing that? Sorry for that. How are we doing that? By actually looking at in the mid-sagittal view of the brain, and what we look for, this little area is called as the intracranial lucency, otherwise the fourth ventricle, which can be identified. If that fourth ventricle is absent, as you see here, this baby has got an open neural tube defect all the way down. The second thing is look at the aqueduct of Sylvius, which is normally located here in between the cerebral peduncles, and here the aqueduct has moved forward towards the occipital bone, which we call as a crash sign. It's like a car which has crashed at the wall. And these are and the lemon sign, of course, are all markers for neural tube defects which is due. Then ventriculomegaly is probably the most common CNS anomaly detected prenatally. When the atrial width is more than 10 millimeters, as I said, between 10 and 12 it is called as mild, between 13 to 15 it's called as moderate, and severe more than 15. Now, common causes are aqueductal stenosis, agenesis of corpusculosum, neural tube defects, congenital infections. Isolated mild ventriculomegaly has a 10% chromosomal risk, and karyotyping and data anomaly is detected, and if it remains isolated and does not dilate more, then over 95% of these babies will have a normal outcome. Then we look at cortical malformations. I said disrupted neuronal migration and organization, where one of the malformations we recognize is lissencephaly, which is agyria or pachygyria as we call it, absent sulci beyond 28 weeks. So here you can see there are no sulci. This baby is 29 weeks old and a smooth brain with thickened cortex. And then we have clefting disorders of the brain, as we can see. There's a cleft which goes and enters to the lateral ventricle on the brain, which is a full thickness cleft in the brain parenchyma. We have open lip and closed lip like, of course, associated seizures and cerebral palsy. Then we have holoprosencephaly. Again, holoprosencephaly is a condition while agents of corpusculosum can be diagnosed late. Holoprosencephaly can be diagnosed as early as 12 weeks because the frontal cleavage happens before 12 weeks. And if this baby the cleavage has not happened, then you have a single monoventricle, which is classical of holoprosencephaly. And excessive small gyri, as you can see here, polymicrogyria, is difficult to detect by ultrasound alone. We really have to look for it, and MRI may be needed for characterization. Then we go on to some advanced imaging techniques beyond the standard B-mode sweep, like 3D neurosonography, 4D ultrasound, Doppler studies for middle cerebral artery, brain sparing, Niger circle of Willis visualization, cerebrovascular resistance monitoring, and fetal MRI. Usually it's an adjunct to ultrasound, not a replacement. It's superior for cortical detail after 22 weeks and delineates complex malformations, and T1, T2 weighted brain sequences are usually seen. So here are some examples of very early first trimester fetal brain. You can see the remarkable amount of detail we're able to get. And this is what we call as a multiplanar view. After acquiring a 3D volume, we do the multiplanar view, and we use a technique called volume contrast imaging, where you can get exquisite detail of the fetal brain, and we can walk through the brain at various things. Then, of course, this is the 3D sweep which we can get, and this is a mid-sagittal view and showing you the MRI, showing the vermis of the cerebellum. And then we move on to malformations of the vascular malformations. Here is a vein of Gall malformation, which color Doppler helps. And lastly, I'm going to tell you something about AI and technological where we get automated biometry, anomaly detection, cortical maturity, and also telediagnosis possible. So here's I'm doing a manual imaging of the CNS system, and there I'm doing an automated CNS. I think the moment I press the button, the machine recognizes it as the brain, and then just need to update, and it takes a sweep, and within 1 second it gives you all the planes, it auto measures, and you can also auto-comment and do the annotations as well. So the time taken is only 52 seconds as compared to 2 minutes 22 seconds. And then lastly, the clinical impact and takeaways. More than 95% NTD detection, 80 to 90% major brain anomaly detection rate, 30% of structural anomalies are CNS, and 20x earliest reliable neurosonogram. So fetal neurosonogram is a powerful tool to visualize developing brain structures. Systematic approach, ventricles, posterior fossa, cortex, and corpus callosum. And then gyrification is a dynamic marker of cortical maturation. Delayed sulcation is diagnostically significant. Early detection enables timely counseling, in utero intervention, and optimized delivery planning. And AI and 3D techniques represent next frontier collaborative opportunity for clinicians and engineers. Thank you very much for your patient listening. Thank you for the opportunity.
Thank you, Dr. Suresh. I now invite Dr. Ilya Bakulin to please deliver his talk.
Good day, dear colleagues. It is a very great honor for me to attend this conference and make presentation. I would like to talk about approaches to personalized transcranial magnetic stimulation. I work at the Russian Center of Neurology and Neuroscience. It is the largest institute in Russia in relation to clinical and experimental neuroscience. Specifically, I work at the non-invasive neuromodulation lab. The main scientific activities are related to the development of a new diagnostic or prognostic non-invasive neurophysiological biomarkers of neurosystem diseases such as stroke, amyotrophic lateral sclerosis, chronic disorders of consciousness, and so on, and provide functional brain mapping. We are developing and exploring new therapeutic neuromodulation protocols based on fMRI-guided TMS, accelerated TMS, or quadrupole stimulation, and so on. The history of the use of transcranial magnetic stimulation in clinical and research practice date to back in 1985, and since then significant progress has been made. TMS is based on magnetic induction principles, brief and rapidly changing high-intensity electrical current. It generates powerful magnetic field that penetrates the cranium and is capable of inducing electrical current in excitable tissues. Applied onto the cortical areas, it is possible to provide neurophysiological or behavioral effects depending on contributing functions of stimulated area. For example, when applied onto the primary motor cortex, TMS can activate corticospinal tract and inducing twitches in muscles. TMS has not only local but distant effect conveyed through connectivity linking different brain regions of the same networks. The direct neural response to TMS pulse is very complex, involving a mixture of neurons. The propensity of the axons to fire an action potential in response to transcranial magnetic stimulation depend on their geometry, myelination, and spatial relation to the induced electrical field, as well as functional state of neurons. There are two main approaches of TMS application in clinical and research practice. The online approach applies TMS protocol to measure its immediate effect on brain function or activity. The online protocols applies to measure cortex excitability or plasticity, to provide the functional brain mapping, and define new diagnostic or prognostic biomarkers. The offline approach applies TMS protocols, so-called repetitive TMS, that induce neuroplasticity, inhibit or facilitate the stimulated brain regions. The ability of repetitive TMS protocols to non-invasively modulate brain activity has been translated in clinical practice to new neuromodulatory treatment in several nervous system diseases. According to evidence-based guidelines on therapeutic use of repetitive TMS, the level A definite efficacy was reached for depression, neuropathic pain, and motor stroke, and level B and C for many other diseases such as multiple sclerosis, Parkinson disease, and so on. But the use of TMS in real clinical practice is very limited by the high variability of its neurophysiological and effects and clinical efficacy. The big problem remains the stimulation of the brain as a black box, which partly explain the neurophysiological variability of TMS effects. So there are many approaches to more precision, personalized stimulation. One of the most promising approaches is a more accurate targeting of stimulation coil using neuronavigation instead external scalp landmarks. Using neuronavigation, we can see online the location of coil in relation to stimulated target. Another approach use functional neuroimaging data such as resting state fMRI or task fMRI to provide precise personalized target for stimulation. For example, in depression, the efficacy of TMS applied over left dorsolateral prefrontal cortex was shown in a large number of studies, but effect is moderate and variable. Big problems are related to the choice of stimulation target. Even using neuronavigation, it is not clear which area within left dorsolateral prefrontal cortex should be stimulated. In a series of articles, it was shown that effects of stimulation of different sites within left dorsolateral prefrontal cortex is related to the connectivity of each site with subgenual cingulated cortex. So functional connectivity may provide a precise approach to selection of target of stimulation in depression. We compare the standard approach based on neuroanatomical data and personalized approach, including the identification of target within the left DLPFC with maximum negative correlation, maximum negative functional connectivity with the subgenual cingulated cortex. We observed that both approaches have significant antidepressive effects, but personalized targeting can accelerate once the clinical effect of TMS. Another approach involves the use of task fMRI. For example, in the patients with vascular cognitive impairments, we use cognitive task fMRI to identify a target within left dorsolateral prefrontal cortex, and we observed the significant cognitive effects of this protocol. One of the promising ways to increase the effect of TMS is used combined protocols based on metaplasticity. The term metaplasticity refers to any to any change in the degree or any change in the direction or degree of synaptic plasticity based on prior neural activity. According to the metaplasticity concept, the threshold for activity-dependent plasticity is dynamic and is a function of activity, of prior activity of postsynaptic neurons. How can we use this approach in TMS study? We can use a combined protocol which consists two or more blocks, and this block can interact, and these combined protocols may have a more strong effect than single block, and at the same time, combined protocols may have reverse effects. So the big question is how to design optimal accelerated protocols capable of promotion therapeutically beneficial metaplasticity. It is depend on a time interval between blocks, and a long interval, 16 minutes, may have a maximum effect. Stanford neuromodulation therapy use this approach. It is a protocol that includes 10 sessions of ITBS per day and a personalized choice of targeting within dorsal prefrontal cortex, and clinical trials were shown a very big antidepressive effects of these protocols with the remission rate 79%. As far we know, we were the first outside the Stanford who test these protocols, and we also observed very big antidepressive effects of these protocols. Interesting approach includes a combination of TMS with cognitive activity, but we've shown that only TMS without cognitive training have significant cognitive effects, and cognitive load during TMS need to be careful monitoring. Now, one of the interesting approach is a connectivity-guided TMS. For example, in patients with Alzheimer disease, it was shown that stimulation applied over the precorneus, an area directly connected with hippocampus, may have a serious effect. Now we are doing two projects with connectivity-guided multi-target TMS. One of project includes a stimulation target selection based of connectivity profile, connectivity pattern, for example, with hippocampus. In another project, we selecting the target within dorsolateral prefrontal cortex and posterior parietal cortex as a part of frontoparietal control networks, and it is a multi-target stimulation of the hubs of these networks. In conclusion, I would like to say that all new approaches need a careful empirical verification in experimental and clinical studies, but these approaches and investigation in this field may significantly increase the clinical efficacy of TMS. Thank you.
Thank you, Dr. Bakulin. Our next speaker is Dr. Lee Han, who has joined us online. I now invite Dr. Lee Han to please deliver his talk.
So, can you hear me?
Yes, you are audible. You have 10 minutes to present your talk. Thank you.
Okay. First, thank organization to invite me to present my work here, and also apologize cannot be on site. And today I will take a talk about the application of cellular multi-omic sequence technology in brain science. I'm Lei Han from BGI Research. And first, as we know that genetic variants can cause phenotypes, but the gap between the variants and the phenotypes is not resolved completely. We need to understand these relationships in the context of the epigenetic modification and in the cellular context. Fortunately, in last 2 decades, there are a lot of cutting-edge single-cell multi-omic technologies have been developed, and the technology has been widely applied in biomedicine, and including brain science. A lot of papers were published in top journals in brain science, and our institute also developed a lot of cutting-edge single-cell multi-omic sequence technologies in our institute from the 2010, from single-cell exome sequencing to the single-cell transcriptomic, epigenetic, and spatial transcriptomics. And next, I will first introduce about our high throughut and cost-effective single-cell RNA sequence using a droplet-based high throughput single-cell sequence transcriptomic technology. And we use this technology and in non-human primates, and contracted the first non-human primate single-cell transcriptomic atlas. And then we recently developed another platform using chip-based single-cell sequencing. And the comparison of the 2 different single-cell transcriptomic platform, we found that in this platform, we can capture large size, soma size cells, not only in the brain, but also in the other tissues. And because it's based on droplet-based single-cell transcriptomic sequencing, it can only capture a small soma size. And this platform can non-biased capture the cell type. And then we also developed simultaneously capture the single-cell transcriptomic and single-cell epigenome sequence in the same cells. This can be precisely see the epigenetic modulation in one cell, and this was the performance of this technology in mouse brains, and we can clearly see the different cortical cells in both gene expression and chromatin accessibility, suggests the good performance of our technology. And the next, we also developed a spatial transcriptomic technology named STILL-seq. This was a cover paper in 2022 in Cells. And we can using chip-based platform to capture the over the single cell resolution and capture the every cell location of the every location distribution of the cell and transcripts in the brain size or in the embryonic mouse. Because the spot we captured transcript was so close, distributed next to each other, and the distance between 2 spots were only 500 nanometers, so we can capture single cell level information using this chip. And also our the vision field was also large. This was sagittal brain, monkey brain sections, and the size is over 5 times 5 centimeters. But we can capture more large brain tissues. The largest dimension was 13 diameter times 13 diameters, 13 centimeters. And in last year, we also developed another version of STILL-seq V2. The STILL-seq V1 can only capture the fresh frozen tissues. And in StillSeq V2, we can capture transcripts in FFPE fixed tissues. So we applied a different strategy to capture the transcripts. The V1 can only capture the poly A tails transcript. And in V2, we applied a random primer and can capture the not poly A tailed transcripts. So it can also applied in fresh frozen tissues and FFPE tissues. And also, we developed a spatial ATAG sequencing, and this was a demonstration of this technology applied in embryonic mouse. And we can see a lot of epigenetic characters can be captured in liver, brain, jaw tooth, and the spinal cord. And this is our working flow or working paradigms. And we first developed home-developed sequencing technologies, and we can generate data in a large platform. And then we develop a bioinformatics algorithm, and then we do some data mining in our data. And using this paradigm, we collaborate with a lot of institutes, and in last year, we published a series of papers in cell, neuron, and developmental cells. And the two papers was in the cover paper of cell and neurons. This was the first demonstration of our brain project in using our technology. This was the 3D mouse brain, spatial transcriptomic mouse brain, and the color represents one cell type. And this was layer 6 glutamatergic neurons in neocortex. And specific site in cerebellum. So using this spatial transcriptomic technology, StillSeq, and we can capture every single molecule and cell types in the mouse brain. Sorry. And this was also we using the StillSeq and single cell transcriptomics in marmoset, and we have a comparison with macaque and mouse. We and find a new cognitive cells in primate. And when integrating with the data of the MRI, functional MRI, we can see the correlation. We can see the correlation of the functional MRI and the molecular gradient in marmoset cerebellum. And this work was published this year also in Science, and also in marmoset cortex. And we found a molecular gradient in the cortex of the marmoset from the anterior ventral to dorsal posterior. There was a gradient, a molecular gradient between these two directions. And this can be also manifested by the functional MRI. And we also performed similar examinations in macaque cortex. and we can clearly see the different layers of the macaque cortex, and also we detect some visual cortex specific distribution of visual cortex features in macaque visual cortex. Beside we conducted in the animals, we also focused on. We also performed a series of the experiments in human. We performed in the adult human and do a cross-species comparison with macaque, marmoset, and mouse in the whole brain level. And this work was still ongoing. And we also performed this in embryonic brain and in whole brain scales. Also, we use clinical human cortical tissues from the surgery, and we profile the molecular changes or cell type changes in human cortex agents, and we found a specific cell type SST inhibitory neuron. The density of this neuron was decreased during the age. And also a recent paper in Nature also shows that in AD patient, the SST neuron was also have the same tendency. And this was we also do some research in human disease in AD. This work has been presented by Professor Zhang in last session, and I will not go to details. And the last project of our human brain project was in axolotl brains, and this animal was famous for the capability of the regeneration. And also in the brain, we do a surgery first in the adult stage, and then we observe the whole process of the recovery stage of the cortical regeneration. And we found that the specific molecular and cellular program during this process, and we see that a lot of radio gray cells can migrate to the cutting edge of the surgery edge of the tissue, and then proliferation and the differentiation, and then in the end we can see the left and the surgery part were completely the same cell composition from between the two parts. And from during that process, we can see a lot of microglia was activated, and the radioglial cell will migrate, and we find but not all the radioglial cells can be activated. Only a part of radioglial cell was activated and differentiate into the neurons and other cell types. And the summary, and we will continue to use these multi-omic, even the multi-model data like brain image and proteomics and metabolic omics together to deciphering our brain in three directions. One is brain evolution. The second is brain development, and the third is brain disease. And I should take my acknowledgement to my colleagues and collaborators and the supporting fundings and the platforms in BGI. And thank you for your attention.
Thank you, Dr. Han. I would also like to request you to stay with us and join the Q&A session. And I would also like to invite Dr. Bakulin and Dr. Suresh to join the Q&A session.
This is to Dr. Suresh. You have been involved, I have a couple of comments. You have been involved in this work for several decades. Are you seeing less and less neural tube defects? That is first thing. After that, I'll ask the second point.
Yeah. I think the question is whether we are seeing less and less neural tube defects. Yes, we are seeing less and less neural tube defects of live birth of neural tube defects. But more and more are detected very early in gestation, and most of them terminate pregnancy. If you ask me whether the occurrence of neural tube defects has reduced, actually no. Because even the implementation of prevention with folic acid and B12, because in India you require B12 plus folic acid, which very few people take. Only B12 folic acid is given by most people, but a combination of folic acid and B12 is the only thing that will prevent in India by the research which we have done many years ago.
The second point, the development of GNU of the corpus callosum, it's a very interesting observation. But if you take evolution of human brain, visual cortex and optic radiation gets myelinated first. The white matter fibers in frontal lobes, they actually, myelination continues till adolescence.
Yes.
That's why children are not able to make ethical and moral decisions because myelination is incomplete. It's very surprising that instead of splenium, which is close to the optic radiation, why GNU is getting myelinated first? I mean, why we're able to make out first?
I think this is a question to be answered by researchers. I am only seeing what I'm seeing and presenting to you.
Hi. Hi. Hello. Am I audible? This question is to Dr. Bakulin. So in one of your slides, you said, great talk, by the way. So you said you identified specific sites within DLPFC that showed maximum negative connectivity with a specific part of ACCU. So what was the outcome of that study? So was that, were you trying to alleviate MDD, depression? Was it, and how, between individuals, those specific sites that you detect within DLPFC, is there a lot of variation?
In depression, we use personalized stimulation of area within DLPFC with maximum negative functional connectivity with subgenual cingulated cortex, because the antidepressive effects depends on the how close the site of stimulation to the area with negative connectivity, and reduce this negative connectivity associated with improvement in symptoms of depression. In another disease, we can select target in different ways. For example, for cognitive impairments, we can select an area that are part of cognitive hubs and so on. It is a specific approach to depression based on resting fMRI analysis.
My question is just a follow-up to what she asked to you, Dr. Bakulin. So if you're looking at personalized TMS, what do you think is the longest duration of effect that could last and actually show you an fMRI change once you've given all of those sessions?
It is an interesting question. It is need to be investigation in future, I think. It is not an empirical data about the stability of target, of course, because fMRI is a noisy method, and regarding to the change of location of stimulation site per session during the course of ITMS.
Would you also expect, so I am a clinician scientist working with developing brain, so would you also expect developing brain to in turn impact the way the response to RTMS would be, apart from the disorder per se?
Okay, I think I expect.
In the interest of time, we will rest the Q&A session here. Thank you to all the speakers. Thank you, Dr. Hahn, for joining us. We will now proceed to the 9th scientific session. It is my pleasure to introduce our featured speaker, Professor Paul Manger, followed by Professor Soumya Iyengar, Professor Thomas Gregor Isaac, and Dr. Shrikant R. Bharadwaj. I now introduce and invite Professor Paul Robert Manger to please deliver his talk.
Hi there, it's great to be here in Chennai again. I always enjoy it. Waiting for the slides. Okay, so I'm sort of posing a question rather than giving lots of data at first. Are elephants smarter than people? So when I was shooting the breeze with Mohan last week, he sat back, crossed his legs, put his arms down, and he said, of course they are. Ask any Indian. They will tell you that elephants are smarter than people. So I'm asking Indians, if you think elephants are smarter than people, please put your hand up really quick. Okay, so he was talking pretty generally for the population. So let's see what we can do with this. Oops. That way for forward. Okay, so if we look at the basic kind of things that most people understand, the elephant has a 5-kilogram brain, the human a 1.4-kilogram brain. So the elephants have a much bigger brain. The elephants have 257 billion neurons, 3 times as many neurons as humans have. But only a third of the number of neurons in the cerebral cortex as compared to humans. But elephants have the largest and most complex cerebellum, and that goes down to sort of the level of neuronal complexity as well. It's much higher than what you find in humans. But the hippocampus of the elephant and the hippocampus of the human are kind of about the same. So if we're trying to answer the question, are elephants smarter than humans, the data is a bit backwards and forwards. We're not really sure about it. So we have to take a deep dive into some comparative research. So the first thing we want to look at is we went in and said, what are the parts of the brain doing? And especially between the thalamus and the cortex. So we divided the thalamus and the cortex up into groups and territories that are linked together. And as you can see, we have the anterior group. Its function is effect, and it's connected with the cingulate cortex. Medial dorsal group, which is the executive functions. Motor, somatosensory, visual group into 2, the extrastriate and the primary visual cortex, and then the associative parts, and then the auditory group. So we looked at this across 9 different mammals, and we measured the volumes of these from published atlases. So very easy, straightforward work, just measuring volumes, plotted them against each other, and what we found is this beautiful correlation between the volume of the thalamic group and the volume of the cortical territory that it was connected to. This means if we know the size of a thalamic group, we can predict the volume of the cortical territory that will be in any mammal. So because of that, we decided, okay, let's look at some more mammals. So we looked at 76 more species, not just 1 or 2, quite a few, and across the whole range of the mammalian radiation, from monotremes, the platypus and echidnas, through to the apes and humans, and pretty much as we could in between. I'm not going to show you all the data because that's a huge study that's we're sort of working on to publish, but I want to show you some of the really important parts of this in relation to the question. So if we look at the size of the volume of the dorsal thalamus and we plot that against the volume of the anterior group, we see across most mammals there's a beautiful regression, all strongly statistically significant and so on, but we find that primates have relatively small anterior groups. They're not as emotional as other animals. That's the bottom line. Primates aren't as emotional as other animals, but then if we look at the African elephant data, we find that it's sort of similar to the other primates. So that's kind of nice. Elephants and primates aren't emotional. If we look at the medial dorsal nucleus, which is for the executive cortex, we find that primates fall above the regression for other mammals. We have more executive cortex than other mammals in primates. That makes us a little bit more intelligent. And interestingly, the African elephant is doing the same thing. They have more prefrontal cortex than other mammals, but not all large brain mammals have big prefrontal cortex. Whales and dolphins fall down low. And I've said for many years that whales and dolphins are not that smart, and this is just another data point proving that. Now if we look at the association cortex, and it's really the executive and the association cortex that are really important in terms of understanding different animal cognition, we find that across all the mammals, except those that are blind or have other specializations, everyone's kind of the same. The relative size of the association cortex is going to be similar. And when I say association cortex, I mean extrastriate visual cortex, parietal association, and temporal association. So these are the areas that are doing a lot of different things. If we look just at visual, we see primates have, as the brain gets bigger, they have a relatively smaller lateral geniculate nucleus, which means that primary visual cortex is going to be relatively smaller, and along with that, the African elephant does the same thing. So that's all nice and good. The elephant is looking a lot like a primate brain in many senses. So let's look at it in a little more detail again. As we said earlier, brain volume doesn't really tell us all that much. Elephants are big, they have a big brain, about 5 kilograms, and the whales and dolphins have big brains as well. That's correlated with dorsal thalamic volume, but when we get to executive cortex volume, or the prefrontal cortex, humans are around a third of what we find in elephants. Elephants have a prefrontal cortex that's 3 times larger than what humans have. Whales and dolphins, on the other hand, have a tiny prefrontal cortex. If we look at association cortex, again, we're finding the same thing. The elephants have about 3 times the association cortex of humans, but humans and whales and dolphins have a similar amount of association cortex. We can go further. We can say how many neurons are there. This is kind of an important point. How many neurons are there, because they're the ones that are actually doing all the functional work. Humans have between 1 and almost up to 3 billion neurons in the prefrontal cortex. Elephants fall between 1 and 1.2 billion neurons. The whales and dolphins around 30 or 50 million neurons. There's a big difference there. If we look in association cortex, again, humans have a much larger number of neurons in the association cortex. Elephants falling right on the lower end of the range of what we find in humans, and whales and dolphins, again, very, very low. So we know that we can see humans, despite having a smaller size of the association and the executive cortex, they have more neurons than elephants do. But elephants are rivaling them. They're on the lower end of the range of what we see in humans. If we look at the individual neurons, how complex are the individual neurons? If we look at total dendritic length, the elephants are the winners there. Humans and whales and dolphins are about the same. That's in the executive cortex. In the association or the dendritic cortex, again, the elephants the association cortex, the dendritic length of the neurons, is again much larger in the elephants than in humans and whales and dolphins, which are quite similar. If we look at dendritic segments, this is the amount of branching of the different dendrites. Elephants and humans are up high, and the whales and dolphins are down low. A similar thing you find in the association cortex, where elephants have a great deal more branching than we find in humans and whales and dolphins. And when we look at numbers of spines, spines are important, that's where things connect up. Elephants are up high, whales and dolphins sort of down towards the bottom end of elephants, humans down low in prefrontal cortex. That doesn't make sense, right? We're the smartest animal. But then if we look at the association cortex, we see the same thing again. So even when we take a deep dive into this comparative neuroanatomy, there's some things that are really kind of puzzling in a bit of a way. So if we look at this in a sort of a broader sense, the large brain mammals, humans have the highest number of neurons. That's something we have. That's quite clear. And we have a high neuronal complexity. That sort of in the association and prefrontal cortex underlies our cognitive capacities. Elephants rival the humans in terms of the lower numbers of what we find in humans, but they have a much higher neuronal complexity. Whales and dolphins, the numbers are well below humans and elephants, and they have a lower neuronal complexity. So if we look at Google staining, we see a pyramidal cell in the prefrontal cortex of the human. You see the branches and the number of spines. The elephant, I had to zoom this out a little bit. So if you imagine that the cell bodies are the same size, you can see the extensive dendritic length on the elephants, and you see the much higher numbers of spines on the dendrites. If we look at the whales and dolphins, this is from a bottlenose dolphin, the number of dendrites is lower, the branching is lower, and the number of spines are lower. So it sort of puts it into a bit better of a context. Elephants do compare favorably with humans. They're actually, they have a much larger brain but fewer cortical neurons, but the numbers are sort of similar when you put the densities of neurons and other things in place. And when you calculate it all up, they're on the lower end of the range of what we see in humans. The complexity of the cortical neurons is similar, although elephants definitely have the edge over humans in that sense. And this complexity is really important because it talks about local processing within a neuron prior to any action potential being generated. And it also tells you how much information a particular neuron can process at any one time. And the elephants are kind of the winners in this in a comparative sense. And there's many thalamic features that are shared between elephants and humans that we don't see in other mammals. So we sort of come to the conclusion that elephants are the most likely non-human animal to have a language. And I want to show you, hopefully this will work. I'm trying to play you some sounds. I played these at the last BRICS meeting, but we had different people. Could you guys play this first sound first? Can everyone hear that? All right. That's the infrasonic rumbling of an elephant. It doesn't sound like too much. It just sounds like a bit of a rumble. But when we up the frequency of it, and we change the frequency, not changing anything, and we play the second sound here, please, guys. That's how it sounds to another elephant. And if we play the third one, if you don't mind. You can see it's quite different to the other ones. So there's something really interesting going on with the infrasound of elephants that makes me think that they're really likely to have a language. And we were working on a series of about 400 vocalizations from elephants. And can we go forward one more slide, please? Okay. No, I need you guys, yeah, there. No, forward, hang on. One more forward. Slide, one more, the next slide. Yeah. So we only have about 400 elephant vocalizations, but we're getting some more. And we got the AI to divide these vocalizations up into little frequency bouts. I'm almost done. And then look at the occurrence of them. And we're finding what is called Zipf's law. And this is a very common feature of language. And we need to do a lot more of this to actually be certain that these guys would need a language. But in order to do a lot more, we need about 500,000 or 600,000 elephant infrasounds to really pull it out and determine if they have a language. So I want to thank all the guys that I've been working with on this. Nelly and Paolo came out from Jose's lab in Brazil and worked with me on all these thalamic quantification, and you guys all know Jose, one of my former students, Spock, a good colleague, Nigel, Mads from the Copenhagen Zoo in Denmark that gets me lots of brains, Adil, you know, and my friend Zoltan from Oxford. Thank you.
Thank you, Professor Paul. I would now like to invite Professor Soumya Iyengar to please present a talk.
Good afternoon, everybody. I'd like to start by thanking Richa and, of course, Mohan for inviting me to this wonderful symposium to present my work on wiring the human auditory cortex during this sensitive period. So, I'm sorry, which is the laser? Yeah. So the people in my lab who contributed to this work were Advind, Anjali, who's here, and Utkasha Singh, a former PhD student, Alok, and a team of really wonderful technicians who helped me collect the brains, the human brains for this work. And a number of really wonderful collaborators without whose help I wouldn't have been able to do this work, because we are not associated, NBRC, my institute is not associated with any hospital. So people from NIMHANS, from the Brain Bank in Bangalore, PGI Chandigarh, and the Guru Tegh Bahadur Hospital, Army Base Hospital in Delhi, and All India Institute of Medical Sciences. And funding for this work came from my institute, National Brain Research Center, grants from DBT, and ICMR. Oops. Okay. So why study the development of the human auditory cortex? Hearing loss is the fourth highest cause of disability globally. And there's a prevalence of about 6.3% in India, which means about 63 million people. And this number is predicted to increase to over 900 million people by 2050, according to statistics given by the WHO. Now, cochlear implants generally provided at about 18 postnatal months can improve speech and language acquisition, but not all children are equally benefited by these. So a very detailed knowledge of what the auditory cortex looks like in humans early in development would be helpful in planning timely interventions. So I'm not going into details of the brainstem pathways for how sound reaches the brain, but ultimately information reaches the medial geniculate nucleus in the thalamus, and from there to the primary auditory cortex, which is present on the Heschel's gyrus, and the Heschel's gyrus is tucked inside the lateral sulcus. This primary auditory cortex, which is labeled as TC here, is surrounded by auditory association areas. Sorry, this is not moving. So this is the developmental timeline of hearing and speech in humans. Hearing actually begins very early in development. At 20 gestation weeks, fetuses are capable of responding to sounds played to the mother's abdomen, and these are all based on ultrasound studies. So by birth, it is not surprising that babies actually have the ability of recognizing their mother's voices as compared to another woman speaking the same passage. It's also interesting that babies which are born near the airports can actually recognize the sounds of planes, or at least respond to them. The sensitive period for learning, auditory learning, happens between this period from birth up to about 4 years, when babies start initially babbling and then gradually start absorbing and learning from their parents. And so there is a period of auditory motor integration when they start to learn their languages and speech. By 7 years, the ability to pick up a second language as well as the native language sort of tapers off. And this learning is supposed to be more or less complete by about this point in time. Now, what we found very strange was that earlier studies, this is when I first started my work, was that although hearing begins as early as 20 gestation weeks, the auditory cortex was supposed to mature comparatively late, that is by 12 to 14 years after birth. And this is based on earlier studies on cytoarchitecture, myelination, synapse formation, and particularly the appearance of neurofilaments. Now, neurofilaments are intermediate filaments, and they're used as a marker for maturation of the cortex. And they also cause an increase in axonal conduction. So you can use these, the appearance of these, to judge how mature an area is. So these are earlier studies that I was telling you about from a group that is Moore's group. What she had shown was that at 22 gestation weeks, you can see that the entire cortex, and this is a section of the cortex, these neurofilament positive axons are present only in layer 1, and there seems to be nothing below that. Of course, at this point, it's the cortical plate. 37 gestation weeks, only layer 1. 4.5 months postnatally, only layer 1 has axons, and the layers below that are devoid of any kind of axons positive for neurofilaments. Now, at 1 year after birth, you can see that besides layer 1, there's a plexus slowly increasing inside. And this is a plexus which is covering layers 4 down to 5 and 6. And this gradually increases so that by about 5 years of birth, after birth, there is this increase in axons throughout layers 2 and 3 as well, but still fairly sparse. This is complete and becomes adult-like, according to those earlier studies, only by about 11 to 12 years after birth. And yet, the fetus and not only the fetus, the young children are fully capable of various kinds of auditory functions. So they respond to changes in the frequency of tones. This was at 28 gestation weeks based on ultrasound studies. Third trimester fetuses can respond to segmented speech, prosody, and pitch. And there is also activation of the primary auditory cortex while listening to speech. This is an MRI study as early as 3 postnatal months. So all of these, then, in that case, according to these earlier studies, would be attributed mainly to layer 1 axons, the reticular activating system, which does connect to layer 1, and subcortical areas. So at the time, since we had some brain tissue available from younger ages, we decided to repeat these experiments using the neurofilament proteins. So this is immunohistochemistry for neurofilaments. Here what we found was, in our case, in our study, at 25 gestation weeks itself, of course, we see a very thick layer 1 full of axons, but we could also see these little arrows point to axons growing up through lower layers, that is layers 2 and 3, and connecting up to layer 1. This is at 25 gestation weeks. At 9 postnatal months, which is here, so within 1 year after birth, we actually see a very thick adult-like plexus of axons throughout the auditory cortex. So from our studies, what we found is that the entire wall of the auditory cortex is populated by axons coming in from various sources, and they are already there at this point. So in case anyone is not convinced by the IHC, we have also done neurofilament mRNA. So we looked for, we did RT-PCR. We found neurofilaments present, both at various stages, all the way from 15 gestation weeks onwards. And we've also seen, tried this out with Western blot, which works. We have neurofilament protein from 15 gestation weeks on to 36 gestation weeks, 36 years, of course. And this is based on the fresh tissue that we obtained from the NIMHANS brain bank. So after we looked at all these neurofilaments which were already present and did populate the auditory cortex, we decided to look at what was the identity of these neurofilaments and of these synapses that had grown into the cortex. We decided to first look at excitatory inputs, and these were basically what we focused on was the cortico-cortical, intrinsic cortical connections and thalamocortical connections. For our studies, what we did was we looked at the vesicular expression of vesicular glutamate transporters. Vesicular glutamate transporters are important for the storage and release of glutamate, and they are presynaptic markers. So there are two kinds of vesicular glutamate transporters that we used. We used VGLUT1, the expression of VGLUT1 for marking cortico-cortical connections, and VGLUT2, which are mainly present in layer 4 for thalamocortical connections. So both of these were present in the developing auditory cortex. Here is a summary of the results. What we found was that cortico-cortical synapses are present and they increase in the supragranular layers. They're mainly present in layers 2 and 3. There is an increase in their number by childhood, and then this decreases. I'm sorry. Then there is a decrease. And with thalamocortical synapses, there's an increase at adolescence, and then there is a decrease thereafter, and this is in layer 4, where mainly thalamocortical axons are present. What we have also seen, or at least correlated with these findings, is that when we see this decrease between VGLUT1 synapses, cortico-cortical synapses between childhood and teenage, this actually goes along really well with the critical period of language. The age for acquiring a second language decreases beyond 7 years. This is kind of correlating very nicely with our cortico-cortical connections. With thalamocortical connections, there are certain long latency auditory evoked potentials, P1 and N1, which continue to mature gradually into adolescence, which is what we can correlate this with. And this also correlates well with an improvement in the perception of speech in a noisy environment. So just going to skip over this, but basically the next thing that we did was to obviously look at inhibitory connections. This data is preliminary, so I'm just going to jump through, but basically this comes from your inhibitory interneurons, that is parvalbumin, somatostatin, and others. Here's a first look at this. So we have very few connections that is in the fetal, during the fetal stages. Here we have stained for VGAT and gephyrin, which labels inhibitory synapses. And then after that, during the postnatal period, the number of these synapses actually increases in the auditory cortex. So this is a confocal study which is still ongoing. I will skip over this, but since this is still an ongoing study. But what we have found so far is that excitatory connections, that is cortico-cortical synapses, are pruned between childhood and adolescence, whereas thalamocortical synapses are pruned between adolescence and adulthood. Our preliminary data does show that inhibitory synapses increase in layers 2 and 3 increase in density, whereas those in 5 and 6 appear to decrease, but we are still adding data to this. And these findings are associated, the anatomical findings are associated with the sensitive period for speech learning. Thank you.
Thank you, Professor Anger. I will now invite Dr. Professor Thomas Gregor Isaac to deliver his talk.
Hello. Yeah. Good afternoon. So, first of all, I would like to thank the organizers, Dr. Mohan and Dr. Richa, and the SGBC team for giving this honor. I would like to talk to you about something about risk factors for cognitive impairment among elderly in the community. Right now, we've been looking at the molecular aspects of it, and we have gone to the systems level. But what does it actually translate to in the community? And that is from the lens of a clinician I would like to cover. So if you actually see, I would like to look at the background of dementia in India, the community-based cohorts that we have at the Center for Brain Research at the Indian Institute of Science campus, the early identification of dementia and pre-dementia syndromes, non-modifiable risk factors, modifiable risk factors, and what would be the protective factors. So if you actually see, this aging and dementia is a big problem. It's going to be an epidemic. And more than 153 million people are age 60 years and above in India. And this is going to increase to more than 320 million by 2050. So that's where we have to understand the need for having a national dementia policy. There are organizations like Viovikas and Dementia India Alliance, which have stepped up to kind of fill the gap and develop policy considerations, but also look at that in little more wide lens. So how do we study this? So we have to look at utilizing the strength of longitudinal community-based aging cohorts. And for that, we have two cohorts at the Center for Brain Research, which coordinates these activities. First one is the Tata Longitudinal Study of Aging, started in 2015, supported by the Tata Trust, wherein we have recruited more than 1,000. Now it is about 1,500 participants, and we are following them up every year. Then we also have, because you cannot actually represent everything based on the urban cohort, we also have to have a rural cohort, because that would represent the significance with respect to the Indian community. So we have a 10,000 rich community cohort at Srinivaspura, which is about 100 kilometers from Center for Brain Research, wherein we have recruited more than 10,000 individuals, aged 45 years and ago. And we are following them up. This started with the support of the Pratiksha Trust. So we have two different cohorts. One is the urban cohort, then we have a rural cohort, and they are heterogeneous by nature. The urban cohort looks something like this hall. We have people from different parts of the world who have come and settled in Bangalore, and they would be having more of tertiary occupation, higher literacy rates, more migration, more multilinguals, more genetic heterogeneity. Whereas the rural cohort, Srinivaspura, is a place known traditionally for mango cultivation. So there are people who are staying there for 3 generations and more, lesser migration, more mono and bilinguals, and less genetic heterogeneity. So these are well-characterized cohorts. So what we do? So we do a detailed evaluation, clinical assessment, collecting the socio-demographic data. Then we call them to our center, and then we do a detailed clinical evaluation. This is done by clinicians in-house, which also would include sleep and activity monitoring. In addition, there's a detailed neurophenotyping of the cognitive functions by neuropsychological batteries. With the blood biochemistry, and blood is also taken and stored for blood biomarker analysis. We had heard before about the utility of phospho-tau 217, so that's something of interest to the institute. Then we also look at genetic analysis, brain imaging. We have eye biomarkers. We also look at gait. We also look at pulmonary functions, cardiovascular risk factors, and audiology. And these assessments are repeated in a longitudinal fashion. For the urban study, it's once every year, and for the rural study, it's once every 2 years. So what have we learned? I will try to give a glimpse of what we have learned. It may not be comprehensive though. So we have looked at the pre-dementia stages. The earliest pre-dementia stage is what is known as SCD, or subjective cognitive decline, wherein the cognitive functions are normal, but there's a subjective feeling that you are not as good at performing cognitive assessments like before. And then you have the mild cognitive impairment, where again the functions are retained, but the cognitive functions are affected. And we found that there is a higher prevalence in the rural population, which is seen worldwide. And there is something, if you have SCD and you are also positive for ApoE4, these individuals are known as SCD+, and they are very important from the biomarker perspective, and they perform poorly on memory and language-related tasks. So mind you, these are normal people without dementia, and we are following them up with the end point of seeing when they develop, if at all, they develop dementia, and whether we can bring in meaningful interventions to prevent that. We also have looked at heart rate variability. These are from the routine 10-second ECGs, and we have identified that the heart rate variability is also affected early in those with mild cognitive impairment. neuroimaging biomarkers of special interest. We have looked at the hippocampal subfields and identified CA3 region of hippocampus. This is perhaps one of the first studies from India and maybe the second study globally, which looked at the subfields and CA3 region of the hippocampus, as well as amygdala-related volume changes were actually seen in those with mild cognitive impairment. And we also try to look at developing standardized tests. The ACE test is being widely used, but actually if we have a language and memory assessment done in the ACE, that itself can be a bedside tool to differentiate mild cognitive impairment. Now the non-modifiable risk factors, definitely we know that dementia increases with age. So the age is something which is quite important, and the Indian population is aging, so that's where it's more and more important to understand. The current age-adjusted prevalence is 7.4% according to the longitudinal aging study in India. And we also know that the women are more susceptible to developing dementia, and that's something which we have been trying to identify why it is. ApoE4, unlike in other populations, doesn't have to be having a direct role in our population, but rather it interacts with other comorbidities and can worsen the cognitive impairment. There's also modifiable risk factors. Lancet Commission's 2024 has brought out 14 risk factors which are modifiable and divided them into early life, midlife, and late life. So that's where it's quite important. So if you actually see, the midlife risk factors are cardiovascular and metabolic in nature. And many a time, diabetes, hypertension, dyslipidemia develop in the Indian population at least a decade or two before what happens in the Caucasian population. So that's about metabolic syndrome. That's what we are studying, which are a cluster of these symptoms. Then late life, we also look at social isolation, air pollution, as well as visual loss. So that's something which also are potentially modifiable. Now, education is a protective factor because it also has a direct implication on neurocognitive assessment. And we have identified that higher level of education is associated with better auditory cognition. So that's something which is quite interesting to know. And in our population, about a quarter of them have prevalence of auditory dysfunction. So that's not a small number. And rural dwelling, lower socioeconomic status, high cardiovascular burden, and bilingualism. This all could alter the association between education and dementia. So that's something which we could identify. Bilingualism is protective in nature. Education is protective in nature. But high cardiovascular burden and metabolic factors do have a deleterious role. Now, coming to hearing loss, I have already explained that. LDL cholesterol has gained a lot of attention. It's higher in the urban population than the rural population. You can see the prevalence, quite high, 1/3 of the population having high LDL cholesterol. And mind you, this was in spite of being on management, on treatment. And females had a higher risk of having multiple lipid abnormalities in both urban and rural. And this could be because we have a lot of people in the postmenopausal age group. The other midlife risk factors are depression and traumatic brain injury. So we have identified these as significant risk factors. Physical inactivity definitely has a role. And in healthy population itself, physical inactivity contributed to cognitive decline or poorer cognitive performance. Excessive alcohol consumption has been widely studied. Hypertension and diabetes mellitus and insulin resistance. These were actually quite important to understand. The general prevalence of insulin resistance is 56.3%. 1 in 2 of the population have insulin resistance. And this has been structurally as well as functionally associated with poor cognition. Social isolation and air pollution are very important, especially air pollution because there's a polluting technology. We use fossil fuels, which was actually seen more in the rural cohort, and they're found to have significant atrophy with respect to the hippocampal volumes. Social connectedness, definitely important. Lesser sleep and higher cardiovascular risk score and hypothyroidism is something which has not been explored as risk factors in the Lancet commissions, but do have treatable roles. So high homocysteine is something which is quite important to know. Vitamin D deficiency and cognitively unimpaired women with early menopause, they appear to have poorer cognitive performance. So this was something which was also important. Now, when I've told you about all risk factors and dismal facts, there are some protective factors maybe in the Indian context quite important. That is bilingualism or multilingualism, which appears to be protective. The people who are trained in Carnatic music but are actively practicing it. So music has a neuroplastic effect and a metaplastic effect that has been found to be of benefit in our cohort participants. So with this, I would like to just bring in an idea which our institute or our center is trying to look on, and that is to identify dementia quite early and help the clinicians to ultimately give or provide or develop meaningful interventions, which would be early identification of biomarkers, identifying us at-risk groups. For that, we are using computational methods, reducing the modifiable risk factors, enhancing protective factors, and improving cognitive decline. With which, I thank all of you for your patient listening. Thank you.
Thank you, Professor Isaac. I will now invite our final speaker for this session, Dr. Shrikant R. Bharadwaj, to please present his talk.
I feel like a bit of an imposter standing here because I am the only person who claims to do visual neuroscience, not really touching the brain at all. None of the previous, I'm not going to use any of the methods used by the previous speakers to actually study the brain. I am a vision psychophysicist, and I run the visual optics and psychophysics lab at the L.V. Prasad Eye Institute. For me, the eye is a light capturing receptacle. It captures light, converts that into electrical signal, sends it off to the brain, and it's the brain that sort of reconstructs the whole visual scene. So the interest for me in neuroscience is if the front end of the eye is degraded because of a variety of reasons. You can see a picture of the Taj Mahal as seen by somebody with optical defects. What sort of brain processing happens there? What sort of decisions are the brain taking in order to solve information coming in this way? So I use a combination of optical modeling, statistical processing of data, and psychophysics to try and understand what the brain is doing. Bottom line, I fantasize about how the brain solves the problem. All of you in this room actually try and solve that problem of what the brain is doing. Right? So I'm going to continue on that thought process. And across the different activities that the lab does, I'm going to just focus on one part of it today, which is development of a technology that I am involved in to measure pupil responses of the eye with potential use cases for emergency rooms, concussion traumas, first care responders, and things like that. And I'll show you some data that hopefully will convince you that this is actually a good tool. I also have a prototype of the device with me, so if anyone's interested in looking at this, using it and trying it out, you're more than welcome to do that. So, of course, all the activities of the lab is done by the students and a lot of collaborators who are involved in this. Funding comes from both government sources and also from the optical industry to support my lab's activities. So why the pupil? We know that the pupil light reflex, the pupil responds to two things. One, to increases and decreases in light levels. It also responds to when you're looking at near or when you're looking far away. So of this, I am interested in looking only at the pupil light reflex. And it's so because it's largely regulated by brainstem function. It's an extremely sensitive marker for brainstem function. There you go. And what do we do to evaluate the pupil? Pupil is essentially a circle, right? It's a black hole in the eye. So you actually use fairly straightforward image processing techniques to detect what the pupil is, what the shape of the pupil, the size of the pupil, how reactive the pupil to light, and how reactive is it to near targets. We also, if you are in ophthalmology, we've got two eyes. So we are also interested in looking at how the two eyes pupils actually are very similarly responding, or are they dissimilarly responding. From a neurocritical care perspective, there are four important parameters that gets monitored for a patient in an ICU. Of course, the vitals. After that, it will be intracranial pressure monitoring, Glasgow coma scales, and assessment of the pupil reflex. Of this, ICP monitoring is a lot more cumbersome. Technology needed for doing ICP monitoring is not necessarily available in every ICU out there. Glasgow coma scales are qualitative in nature. So that leaves us primarily with the pupil light reflex that can be easily objectively measured, and you can actually quantify that. Now, how do we do this? Here is an example of the pupil light reflex. If I can play that video. what you will see is light beam. This is an infrared image of the pupil responses. Oops. There's a little video if that can be played. Oops. So if you hover this thing, so there will be, oh yeah, there it is, okay. So you will actually see that this is an infrared image. The pupils are nice and nicely seen. And when you shine a flash of light in front of the eye, you see this brisk constriction of the pupil responses. In this case, what I'm doing is flashing the light on one eye, and I'm seeing what we call as the direct reflex of that eye, and I am also monitoring the consensual reflex, what happens to the other eye's pupil when I am stimulating one. Ideally speaking, the responses should be brisk, it should be equal in the two eyes, and of course the pupil should be circular in shape. Now, how does this happen in a ICU setting? These are nurse practitioners typically monitoring the pupil on an hourly basis. They use a qualitative flashlight to actually look at these measurements. This measurement unfortunately lacks objectivity and it has high intersubject variability. And here is a card from one of the ICUs that we photographed, and if there's not enough resolution for you to see the pupils actually being monitored on an hourly basis in this patient, and you actually see that the value of the pupil is exactly the same to the second decimal. So now what this means is either the nurse practitioner is super fantastic at measuring the pupil size to the second decimal resolution, or they are simply just copying the values over and over and over again. So on one end, we say that the pupil is the only objective marker, and on the other end, this is what the reality in the ICUs is, right? So for us, the goal, therefore, has been to develop an cost-effective, portable, and an easy-to-use pupilometer that could now be used for objective analysis of these patients who are in neurocritical care and for first care responders. So our journey with the pupil, I come from an eye hospital, so our journey with the pupil started for ophthalmic applications. We were interested in looking at the pupil responses for patients with neuro-ophthalmic pathologies. So we developed a handheld device, and the blown-up photograph of the device is actually seen there. It does very similar things to what I showed in the video. Flashes the light, and it actually measures the size and the various analytics of the pupils. And we did get fantastic responses in controls. As you will expect, the constriction of the right and the left eye are exactly the same. You see data going right along the 1:1 line. In individuals with right eye deficiencies, you see their responses are lesser than the left eye, and vice versa in individuals with the left eye. So when this device was put together and we wanted to go and market this device from our innovation center, we ran into a major problem of who our competitor is, right? And we kept claiming that we don't have any competitors in this market, and turned out that the competitor was a 10-rupee flashlight that people were using. So now how do you make clinicians switch over to a 2 lakh worth equipment from a 10-rupee flashlight? So, having a single device for measuring only the pupils amongst a plethora of other devices was not really a way to go from a market perspective. So we decided to move on and modularize the pupil measurements. So we separated out the hardware from the software, used commercial VR environments, and applied the same software into now Pico Neo headsets. You can apply it to multiple other virtual reality headsets and stuff. Now, in the current state of affairs, this kind of device is actually being used for measurements of ophthalmic applications. So that brings me to the actual pupil measurements for neurological applications. This is the current prototype of our device. It's a monocular application device. You simply put the device in front of the patient's eye, and you start measuring various analytics of the pupil. It gives you both static and dynamic responses, and it's a report that gets generated. You can now compile that report into a single index that will now tell us whether the pupil's normal, abnormal, or somewhere in between, right? So normative data has been collected, compared against individuals with pathologies, and you do see nice ROC curves that you can now use as a distinguishing marker for what is normal from what is abnormal responses. I want to show some examples of longitudinal analysis of data that we collected from patients in the ICU. The top are instances where patients actually passed on while they were in the ICU setting, and you can see that in almost all of them the pupil responses are actually going down. So 0 on our scale is essentially dead, and you are now tending in that direction in cases of mortality. The one positive case that I'm very happy about is this 18-year-old child with meningitis was there for a month in the ICU. When he got admitted into the ICU, his pupil responses were flat. 0, remember, is dead. So this patient had very, very critical pupil behavior. Went through standard of care by the neurologist, and somewhere in between the pupils started recovering, better responses. And at the time that he actually was discharged from the ICU, now robust pupil responses. His term in the ICU was, of course, not dictated by our pupil device, right? So, but what we are able to see is the pupil device is now an objective marker for looking at how the patient is actually recovering from these conditions. So currently, that's our market. This is our competitor that we are now pegging against. Our cost, we believe, is significantly lower, our data does compare for all the static parameters, does compare well with the device. And our immediate use cases is cross-sectional measurements in emergency care, longitudinal assessments in critical care, and we also want to extend this to conditions of trauma emergency wards, first care responders, concussion traumas, and a whole range of other applications. You can see the data that we are now collecting from ICUs of various places. We are now open for clinical trials on this. So if there are interested parties wanting to use this in the ICUs and test this, we are very happy to partner with you. So thank you very much, and that's all that I had. Thank you.
Thank you, Dr. Bharadwaj. We now have a short 5-minute Q&A session, and I would like to request the speakers from this session, Professor Manger, Professor Iyengar, Professor Isaac, and Dr. Bharadwaj, to please join the discussion.
I have a question for
Dr. Isaac, interesting piece of work. I want to understand Carnatic music induces acoustic stimulus, and you have noted improvement in the visuospatial skills. How do you explain vision and audition?
Yeah, sir. Thank you for that question. So music is very underexplored right now in the pure scientific way. So what we did is in our urban cohorts, we had people who were into music, who were trained from childhood. And we also understood that those who were actively performing music were also there. So we looked at their functional and structural imaging. And the structural imaging, it is noted that music has a lot of impact on the right parietal cortex especially, compared to the left parietal cortex, and it just go through the language areas. So the connectivity actually showed significant improvement in the salience network connectivity. So we have to do further details to further studies to analyze it. The numbers of people in our cohort who were musicians were about 15. It's not a big number. So we want to actually do sub-network connectivity analysis to see that. But definitely we saw that the right parietal and the dorsal stream activation was significantly present in musicians. Now how music is encoded, is it like language? No. So it doesn't follow the dominant or the language pathway, but there could be other mechanisms which needs to be explored at system level. Yeah. Thank you, sir.
A question for Paul. I was wondering, the large cerebellum that elephants have, is there a particular functional advantage that you note or can think of, and in terms of neuron number as well, relative, given their limited number of neurons in the cortex, considering the size of that cortex, is there a sense of the distribution in terms of function?
Right, well in the cerebellum they have around 220 billion neurons, way more than everyone else. the individual neurons are larger, they're more complex. Clearly, there's going to be a lot of involvement with trunk movement and other things, but in terms of producing vocalizations, it can be very important. So in the language sense, the cerebellum, and we know in humans the cerebellum's involved in language a lot. And there's other things in the brains of the elephants that all point towards the potential for language.
Hi, I have a question for Dr. Bhardwaj. I was wondering if the device can be connected to, or can be synchronized rather, with other devices like continuous EEG monitoring, and also if we can continuously monitor the pupil for, attention or other state-dependent observations.
The connectivity part of your question is easier. Right now, there is built-in Bluetooth connectivity in the device that you can connect to any Bluetooth printer. That's what we are interested in. But you could potentially connect it to other devices that are used. The second question is a little bit more complex because of two reasons. One is you need to have a wearable device that can continuously monitor the pupil. And the second is for most patients in the ICU, their eyes are closed. So either this device has to be in the form of a contact lens or something like that that's now put inside the eye, or there must be an ability for us to look at the pupils through the eyelids, neither of which is a trivial problem to solve. There are a couple of literature on looking at closed eye pupillometry, but that I feel is more of an AI gimmick than reality at this point of time. So yeah, the second is a tricky proposition here.
My question is for Paul. Disclaimer, this is not in favor of dolphins, but dolphin clicks also follow Zipf's law. So is this, when you say Zipf's law, are you saying really efficient communication rather than language?
Well, it's just one of the indications. And the studies are showing that in whales and dolphins, they're a little bit dodgy. When you actually look into what they're doing, they've got like 30 or 40 vocalizations. We've at least got 400, and we're going to do another 3,000 we have in the database now. So that's, we're upping the game quite a bit. So you have to read a little bit beyond the headlines for whales and dolphins. Okay, no problem though.
In the interest of time, we will cut short our Q&A for now. Please feel free to reach out to them during the breaks. We will have a lunch break now. I request all delegates and external participants to proceed to the back of the hall. All SGBC staff are requested to proceed to first floor for lunch. We will resume our next session at 1:40, so please, everybody is requested to assemble back by 1:35 p.m. sharp. And in the interest of time, for today, we are cutting short the panel discussion. So we will have a shorter panel discussion now. Thank you.
Everyone, welcome back. Sorry for the rush, and I hope you enjoy the afternoon spread. My name is Kritika, Project Research Associate at SGBC, and I will be the host for the second session. Moving on to the scientific session 11, we have a featured speaker by Professor Yang Soon, Professor Xingming Zhao, Professor George M. Varghese, and Professor Vijay K. Raghavan, which is followed by Q&A 5 minutes. Hi, Professor Soon, can you hear us?
Yes.
Okay.
Can you hear me?
Yeah, we can hear you as well. So your talk is for 15 minutes. After 13 minutes, I'm going to remind you, and then you will still have 2 minutes for your talk.
Okay, good afternoon, everyone. It's a great pleasure to present in the BRICS conference. So today I'm going to give you update about the mesoscopic mapping of mouse and the mechanical brain. So by mesoscopic mapping, I mean to map the brain at a cellular resolution or micrometer resolution. So we have a better understanding the brain-wide connectome or cell types of the primate brains. So previously we have released the cell atlas for the macaque cortex, as well as cross-species single spatial transcriptomics of the cerebellum. And recently, actually, the July of last year, researchers from more than 30 research institutes collaboratively released about 10 results, mostly on the primate brains. So today I will give you just a brief introduction of two work that has been done in the International Consortium of Primate Brain. So the first one is organizing principles of macaque colostrum. As you know, that colostrum has been speculated that is critical for consciousness. So, but the connectivity pattern of colostrum and the cell type of colostrum for the macaque brain has not been well understood. And the colostrum is this odd-shaped structure, and so to get a sense of the cell types within the colostrum, two approaches has been employed. One is the stereoscopic spatial transcriptomic approach. The other is single nucleus RNA sequencing. So you get tissue from the colostrum and do the sequencing for a macaque, marmoset, also mice. And the question is, so we got the very nice UMAP of this monkey colostrum cell types, and the question is how this is comparing to other brain structures nearby. For example, insula, amygdala. So we found that the cell type is more closely similar to insula, but is quite different from other brain structures. So the distribution of these cell types exhibit either medial lateral preference, or there's this dorsal ventral preference distribution. As you can see that on the bottom, there are neurons exhibit preference localization at the ventral part of the claustrum, but there is also some types exhibit evenly distributed patterns. And the more important question is how claustrum is connected with other brain structures, for example, other cortical areas. To address this question, we have injected retrograde tracers in 5 distinct locations of claustrum. You can see that in the cortex, there are distinct cortical areas sent to different some region of claustrum, and this demonstrates that there is a topographic projection pattern from the cortical area to the claustrum. And the other thing is that most cortical areas send projections to claustrum, and that is also true for subcortical areas. Except for cerebellum, all the subcortical structures send projection to claustrum as well. So this shows that how different subcortical areas send to this send projection to these 5 subregions of claustrum. You can see that the claustrum, the different subregion of claustrum receive distinct input from the subcortical areas. The claustrum also sent pretty wide projection to the other brain areas. This just show that claustrums send projections to all the cortical areas, and interestingly, the different part of the claustrum send projection to distinct areas of the cortex. Here show some examples. For example, the V1 projecting claustrum neurons exhibit the ventral preferred location, and you can cluster those neurons based on their projection patterns. And this is also true for the projection to subcortical regions. This show the neurons that project to distinct subcortical regions. For example, amygdala exhibits the ventral preferred location as well. You can see that each of these examples, the neurons exhibit some preference, either ventral or dorsal. So this study has revealed that claustrum act as a central hub for brain connectivity. So this likely provides a structural basis for understanding the mechanism of consciousness. There's a project, we mapped the single neuron projectome for prefrontal cortex. So we know that prefrontal cortex has been very important for decision making, for working memory, and the other higher cognitive functions. Previously, we have mapped the projectome for mouse prefrontal cortex. In this study, we mapped a single cell projectome for the macaque brain prefrontal cortex. So this is a wire injection of virus to label the single neuron morphology, and then followed by whole brain imaging. And we also developed this automated algorithm to reconstruct the full morphology of each prefrontal neuron for the macaque. So for imaging, we use this fluorescence micro-optical section topography in Suzhou. There's a Suzhou Institute for Brain Mathematics. They have this setup that can use to image the macaque brain and which is further used for reconstruction the morphology. Here, just show the example of in the right example of one macaque brain that we reconstructed about 500 neurons for the prefrontal cortical neurons, and so we compared the macaque brain and the mouse brain for their morphology of the prefrontal cortex. So there are several interesting findings. Here I just show one of them. So you can see on the left, this is the morphology of few macaque prefrontal cortical neurons, and on the right is the mouse prefrontal cortical neurons. One thing you can see that is quite counterintuitive, because we all think that the macaque brain would be more complex, but actually you can see that the morphology of prefrontal cortex neurons in mice is more complex than the macaque prefrontal cortex. So the complexity of the macaque brain is probably not at a single neuron level, could be at a connectivity level. And there are also the terminal arborization is also smaller in the macaque brain as well. So we are continue to map the other brain regions for primate for the macaque brain, for example, visual cortex, hippocampus, motor cortex, insular cortex. So we are still in the process. Later on, we will release those data. We are still analyzing them. For the mouse brain, we previously we have released the prefrontal cortex, hippocampus, hypothalamus, and recently we are still working on the whole cortical areas, thalamus, amygdala, and other brain areas. We are covering mostly cortical, mostly brain regions of the mouse. For example, last year we have released the single neuron projectomes across all cortical areas. In this study, we have found that cortical-cortical connectivity exhibits a nested modular organization, and also we have done the correlative analysis to see whether the connectivity pattern has anything to do with our gene expression and the neural activity. And we found that there is indeed some correlation between the connectivity and the gene expression and the neural activity. And the question is how we are going to use those information for functional study. Here, just to show you one example. So for amygdala, we have mapped distinct the projectome of distinct cell types. For example, one cell type is that the neurons express opioid mu opioid receptors, and by mapping their single cell projectome, we can see that based on their morphology, we can classify them into 4 subtypes. Each of them exhibit distinct pattern. Some are more local. Some show the long range projection, and these neurons we tested how they are involved in modulation of nociception, because previously we have known it has been known that, for example, paraabductor gray or parabronchial nucleus has been quite important for the processing of the modulation of nociception. So by taking
Sorry, you have just 2 more minutes for your talk. Sorry for the interruption.
Okay. So by employing this knowledge gained from the single projectome, we have designed the behavior experiments to target at their individual branches to different brain areas. For example, we target the branch to parabronchial nucleus, and by either chemical genetics or optogenetics, we found that this branch, by targeting this branch, it has significant role in modulation of nociception. But if you target the other branch, for example, the branch to the VTA or the branch to PEG, it either has no effect or has opposite effect. The subtype 2 project to BST has no effect. So this just demonstrates that by the single cell projection, we can further to analyze the functional role of different branch. This summarizes the study that's examine the functional role of mu opioid receptor positive neurons in the modulation of nociception. So all those data are available online in the database. So you are all welcome to use those data and in your research. All right, I'll just stop here. Thank you for your attention.
Thank you, Professor Soon. Now we have Professor Xingming Zhao, again an online talk. Hello, can you hear us?
Hi, just a moment. What? Okay. No idea what. What? I just can't share my screen. Sorry. Oh my God. Okay. Can you see it?
Yes, we can see your screen. Yeah.
Okay. Thank you.
Yeah. You have 10 minutes for your talk. I'll remind you when 2 more minutes will be left. Yeah.
Okay. Thank you. First, I'd like to thank the organizer to give me the chance to present our work here. Because I have only 10 minutes, so I changed the title of my talk to gut microbiome-driven exploration of Alzheimer's disease. So, in knowing that the gut microbiome play an important role in Alzheimer's disease through the gut-brain axis. However, we don't know what happened during the progression of the disorder. So, for this purpose, we have constructed Alzheimer's disease cohort, and we have enrolled about 500 subjects. And all these 500 subjects cover the normal control, subjective cognition suspicion, subjective cognition decline, mild cognition impairment, and dementia. So actually, it covers all the stages of the of Alzheimer's disease. So based on this cohort, we have collected the metagenomic samples, the metagenomic data, and we want to know what happened to the gut microbiome during the progression of the Alzheimer's disease. Generally, we will talk about the gut microbiome. We refer to the bacteria. But based on our data, we found that except for the bacteria, archaea and fungi and also virus also also changed a lot during the progression of the disorder. What is interesting is that we found that the microbiome aberration actually present a stage-specific pattern. Some microbiota will change their abundance in the early state, while some other species will change their abundance in later stages. So they show very specific, state-specific patterns. We also want to know whether the species we found also are common to other brain disorders. So we have collected data for Parkinson disease, schizophrenia, bipolar, and also autism. For these different disorders, we found that there are about 31 species. They are common. They show common and consistent changes to these different brain disorders. So it means that there are some microbiota, they are commonly related to the brain disorder. And actually, we want to know whether there are some microbiota species that can serve as biomarkers for the diagnosis of Alzheimer's disease. So we have investigated the different combinations between different species of the microbiome to see whether they are useful for diagnosis of the Alzheimer's disease. We found that for MCI or AD, the microbiome species we have identified can achieve about 80% accuracy when diagnose the Alzheimer's disease. After training the model, we transfer the model to the external data set from Japan, which is also a data set for Alzheimer's disease. We found that our classifier can achieve about 75% of accuracy. And we transfer another data set with 16S sequencing technology, the performance is even better. We can achieve about 80% of accuracy. So after all this computational validation, next we want to do some experiment to validate whether the microbiome species we have identified are really related to Alzheimer's disease. So we have conducted an in vitro intestinal simulated experiment. And in this experiment, we use the intestinal simulate to simulate the gut condition. And we want to know whether the microbiome species we have identified from each specific stage can live or can live for a long time in the gut condition. So with this experiment, fortunately, we found that for the stage-specific microbiome species we have identified, actually there are still a lot of species can live in for a long time in the gut simulator. So we think that this species can live in the specific stages of the Alzheimer's disease. So they are really related to the disorder. And next, we conducted a mouse experiment to see whether the gut microbiome can really affect or related to the disorder. So here we consider 3 groups of patients. One group is normal control. And another group is MCI, and the third group is dementia. For each group, we have 3 subjects. And for each subject, we will transplant their stool samples to the mouse. And for each donor, his stool samples will be transplanted to 3 mouse. So in total, we will have 27 mouse in this experiment. So after the fecal microbiome transplantation experiments for about 1 month, we conducted the behavior experiment. Here we conducted 2 behavior experiments. One is one maze, and the other one is novel object recognition. So we want to know, after transplanting the stool sample to the mouse, whether the behavior of the mouse will change or not.
Professor Zhao, sorry to interrupt. You have 2 more minutes.
Okay. Oh. So here is the experiment. So compared to the mice with stool sample transplant from the normal control, we notified that the mice with stool sample transplant from the Alzheimer's disease, their cognition ability decreased significantly. And compared to the control and the dementia group, we notified that the MCI group also present significant decrease of the cognition ability. So with this very simple experiments, we can see that the gut microbiome is really related to the Alzheimer's disease. And the second conclusion is that the gut microbiome shows a stage-specific pattern during the progression of the Alzheimer's disease. Sorry, I don't have enough time. Actually, we also conduct experiments for the host and gut microbiome integration analysis, but I think I don't have enough time to present here. So I will go to the conclusion. So the first conclusion is that the gut microbiome shows stage-specific alteration pattern in Alzheimer's disease. And the second conclusion is that the gut microbiome can serve as biomarkers for diagnosis of Alzheimer's disease. And I'd like to thank the members from my group and also the support of some funding. Okay, thank you.
Thank you, Professor Zhao. I invite now Professor George M. Varghese for his talk.
Good afternoon, ladies and gentlemen. Since most of you are neuroscientists working at cellular and molecular level, I thought I will excite you with how does many of your work translate into real life in impacting patient lives. I will talk about 3 aspects. One, looking at rare deadly brain infection, a prompt diagnosis using advanced technology, molecular technology, and the other diagnostic modalities and management strategies improving the outcome. I would also give you some glimpse of research and innovation in some of the brain infections which we see commonly. And thirdly, from tragedy to transformation, the very fact that you are here today, I will talk about that as well. This is a story of a 49-year-old farmer from Tamil Nadu. He was not known to have any comorbid illnesses. he came with very acute illness, 3 days of fever and headache. He also developed altered sensorium and one episode of generalized tonic-clonic seizures on the day of admission. He also had some cough for 3 days. There was a history of binge drinking with his friends a week before, and he has been a regular consumer of alcohol. And I would tell you why that is important in a few minutes. Now, he was febrile, he was tachycardic, his blood pressure was borderline. He was comatose with a GCS of 8/15. He had signs of meningeal irritation, and his plantars were extensor bilaterally. It means that for a clinician, all these are features of meningoencephalitis, a very acute illness in a middle-aged man, 3 days of illness, very catastrophic disease. Now, when you deal with an acute meningoencephalitis, there are several causes which goes through a clinician's mind. And you look at some of the basic investigations and some of the crucial investigations which will help you decide, are we dealing with illnesses like HSV encephalitis or something else, is looking at the CSF. So when you look at the CSF, you can see that there are a lot of cells in the CSF, 5,000 cells. Normally, you should hardly see any cells in the CSF, a lot of RBCs. And you can see that the protein is elevated, sugar is reasonably not very low. And you can see the picture below. The CSF is hemorrhagic. So it usually is either a traumatic tap or, when you do a CSF analysis, when you do the LP, accidentally you puncture a small blood vessel and there will be bleeding. How you would differentiate that traumatic tap from hemorrhagic CSF is that you take serial, you look at the serial tubes, and if it clears up, you know that it is a traumatic tap, and you can see that all are of the same intensity, and that is a real emergency condition where meningoencephalitis with hemorrhagic CSF causes are very few, and you look at the number of cells, it's more like a bacterial infection and not like a viral illness. And for an astute clinician, you look at the chest X-ray, there is mild widening of the mediastinum, telling you that there is a rare cause. So our junior doctor suddenly called up, and I said, run to the lab with the CSF, and you can see that his gram stain was positive for gram-positive bacilli. PCR was confirmed for Bacillus anthracis, and the blood and CSF culture grew Bacillus anthracis, which means that this is somebody with inhalational anthrax, and that's why the widened mediastinum, and going on to develop acute meningoencephalitis with anthrax. And what do we do? So unless you make a very prompt diagnosis, you will not be able to save these patients' lives. So there are 3 key drugs. 2 of those are bactericidal agents, and the third one, which is either linezolid or clindamycin, to mop up the toxin which is produced by the bacteria, and that is the standard of care. And this is what is based on the evidence. This is probably the best treatment strategy, and this patient walked home. An astute clinician thinking about a rare condition with advanced state-of-the-art diagnostic capabilities and appropriate therapy, improving patient outcomes. There are several other things which we look at. As you know, TB is one of the common problems in our country. When people develop TB meningitis, some of them go on to develop tuberculomas, these ring-enhancing lesions in the brain, and they go on to have seizures, raised intracranial features. You give usually you give anti-tuberculous therapy for 9 to 12 months. They will continue to have these tuberculomas persisting even at the end of 2 years or so. People didn't know how exactly to treat that. With anti-tuberculous therapy alone, things were not improving. Eventually, we realized that it's actually the inflammatory response by the host which is causing more of the problem. And this was looking at dampening that inflammatory response, improving the outcome of patients with tuberculomas. And again, I won't go into the details. The invasive mucormycosis, another dreaded problem, very commonly seen in our country in patients with uncontrolled diabetes, going on to develop invasive disease, orbital apex syndrome, and invasion into the brain. This was a major problem during the COVID outbreak. The steroid use fueling the uncontrolled diabetes, as well as the steroid causing immunocompromised state, resulting in invasive mucormycosis. And that again, standardizing the treatment strategies, reducing the mortality rate from 40% down to 12% was this study. This is the story of a young man in the picture from Chennai. He was bitten by a rabid pup in the campus. He was pampering the pup. Eventually, he contracted rabies 3 months later. He came with hydrophobia, aerophobia, confirmed to have rabies encephalitis in CMC. Eventually, he died. And that was the time when there was a lot of excitement about Milwaukee Protocol, induced coma, potentially, could be saving people with rabies encephalitis. But we did try Milwaukee Protocol in few of the patients and found that it was futile. And however, the biggest encouraging thing which came out of this exercise was one day when Mohan was asking me, George, if you have the whole brain sectioned into thin slices, what will you study? And that is where it began. We actually, Mohan said we can develop a technology and image those, reconstruct the brain. And my answer to his question was, I would look at rabies where almost 100% fatality. I'll wind up, and Professor Vijay Raghavan believed in us when we said that, Mohan said that we will develop the technology and we will look at that. And then eventually Chris Gopalakrishnan funding the center, and that's why you're here today. Story has not improved very much. This is actually, it's a video, but I will just skip that. Can you, yeah, sorry, can you just put the last slide, the conclusion slide? That was a patient who came, and that video, if you really would like to see somebody in having hydrophobia, can you click on that video, please? That picture, there's a video at the bottom. He also did not make it. Eventually, I mean, we tried something called fosaprepitin, which again did not work. And the last slide, the conclusion slide, please. So just to summarize, every missed diagnosis is a missed opportunity. Rare brain infections demand vigilance, innovation, and prompt action. Patient tragedies are not endpoints, they are the beginning of research questions that transforms care. Turn the burden of infections into a global laboratory and collaboration for life-saving innovation. Rabies is one such problem where we still need answers to many of the problems it causes and the cause of death. Thank you very much for your patient listening.
Thank you, Professor Varghese. Now I invite Professor K. Vijayaraghavan for his talk.
Yeah. Good afternoon, everybody. It's a great pleasure, as always, to be here. And it's very thrilling to see the Sudha Gopalakrishnan Brain Center going from strength to strength each time I visit. It's absolutely stunning. So congratulations to everyone involved. And congratulations to bringing together all the speakers today. It's really very fascinating to hear diverse talks across multiple organisms and cell types. So it's a pleasure to be part of this Neuroscience Symposium, and this brings together model systems, scales, countries, with themes ranging from neurodevelopment and molecular neuroscience to neurotechnology and computational neuroscience. And one of the problems with neurobiology in particular, but with any complex biology in general, is what conclusions can we draw from all our studies. We are, for example, if you take a simple bacterial cell, we can figure out every single gene expression at any given time. We know how bacterial motors work, but still we are unable to make predictions about what the bacteria will do when it gets multiple kinds of sensory inputs. So if that's a challenge, is it meaningful for our address tissues, for example, which have 86 billion neurons? But my conclusion will be it's not. It's a sensible enterprise. And to understand the human nervous system in general and the brain in particular, and to treat it well when needed, we require information, data gathering, knowledge, understanding, and application. And all of this depends on technology. But none of this is a tidy sequence. They occur in parallel, in feedback, in mutual correction. So our quest is actually much like the brain itself, the way it functions. Now, Sidney Brenner once remarked that progress in biology depends on the interplay of techniques, discoveries, and new ideas, and said that it's probably in that order of decreasing importance. In other words, he thought techniques were more important than discoveries, which are more important than new ideas. We're living through an age of extraordinary technique, but technique by itself doesn't guarantee understanding. Progress comes when technology is in conversation with discovery, and discovery is guided by ideas, and this is our challenge. Now, this is particularly true in neuroscience because the challenge is intrinsically, almost by definition, multi-scale. What any cell does, what a circuit does, what an animal does is a consequence of interactions across many levels: genes, gene regulation, ion channels, synapses, neurotransmitters, excitability, connectivity, developmental history, environmental context, and behavior. And none of these. No understanding of any one of these is sufficient. Now, in some systems, and this is a system which our lab works on, there are thousands of people all over the world working on, this is the fruit fly Drosophila. In some model organisms such as Drosophila, we have a level of access that would once have been unimaginable. In principle, we can identify gene expression patterns in particular cells over time. We can follow developmental trajectories in great detail. We can study cellular physiology and behavior. We can perturb or activate or inactivate defined neurons while looking at behavioral outcomes. And we have a complete connectivity of every single neuron in the fly brain. There are about close to 200,000 neurons in the entire fly, and we know how they're all connected to each other. This is incredible, an incredible amount of information. Now, this forces a sobering question. What is our predictive power in this case when we have all this information? Can we say how a freely moving fly will respond to a complex multimodal sensory world? Can we predict from a small group of neighboring precursors during development of multiple kinds of cells which are very similar or identical to each other, how each of them will go into different developmental contexts? Can we move from a complete or a near description of information to a compact logic of how development takes place and how function emerges from that development, interacting with the cellular and outside environment? And the answer, at least for now, is pretty badly or only partially. It's still impressive, and we're becoming better and better at describing these systems in extraordinary detail. But sometimes it feels as though the more we know, the less easily we can extract the governing principles. Our description has been improving incredibly, but our explanations are advancing more slowly. Now, this is not a failure. It's the natural consequence of entering a domain, and biology is such, where complexity is not decorative. It can be at some times, like peacock feathers, but in general, complexity is fundamental to biology. Let me make the point in another angle. Consider the broad genomic landscape. E. coli, the bacteria, has about 4,000 genes, 4,400 genes. The worm C. elegans has about 20,000 protein coding genes. The fruit fly has about 17,700 genes. The fly can fly, the worm cannot fly. And we have about 20,000 protein coding genes. That number has come down substantially from earlier estimates. So the numbers of protein coding genes are relatively small and similar in various multicellular organisms. Some other organisms have many more, but really gene number, protein coding gene number, is not a measure of complexity in behavior. So gene number doesn't matter. What matters perhaps is gene regulation, and that gene regulation complexity is relatively poorly known. We got the basic principles studying bacteriophage and bacteria, but the complexities of eukaryotic gene regulation in our kind of cells, are much more difficult to understand and predict. Doing that during development is even more challenging, and doing that in terms of looking at behavioral outcomes is much more complex, even though the tools have become better and better and our information gathering is even better. So, even when we obtain an even more detailed atlas of gene expression, we're relatively at an early stage in understanding how patterns of gene expression map onto cellular states and how these cellular states change with time during development and what happens. And this is something, again, which is an area of intense research. We are becoming relatively good at taking large volumes of data, moving from one cellular state to another cellular state, but doing this over development is becoming is much more complex, and that's the next challenge. So this leads to the next challenge in terms of how development, so we moved from gene expression, how development and morphogenesis takes place, and across biology, we have a great simplifying feature, which is a shared toolkit. Cell contact, I've done 2 minutes more. Yeah. Cell contact, short-range signaling, long-range signaling, all of these are shared toolkits across biology. And this toolkit is reused again and again in different contexts during evolution and different tissues in our bodies, and yet the outcomes and impacts are very, very different. So this is because context matters. Now, gene regulation and developmental biology have been extraordinarily positive because our collection of data and our information and our interventions are very close to the outcomes we are measuring. And therefore, we don't have to make great leaps. But when we try to understand behavior of neural circuits or behavior of animals such as us, we're making great leaps from molecular and cellular data collection to the outcomes. And that remains a big challenge. Yet, it's amazing how valuable these bridges and these leaps have been and how much we've learned. So how do we bridge this gap? We have big data, we have machine learning, we have AI. These are all indispensable tools, and one should not underestimate their value. They will help us see structure and higher dimensional data. They will help us to classify, compress, and detect patterns. But they will not on their own provide the kinds of hypotheses that biology ultimately requires. They may suggest candidates, they may sharpen possibilities. They may help us navigate complexity, but exploration still requires ideas that need to be tested, simplified, broken, and rebuilt. So we must do two things at once. We must continue to build powerful technologies and scale them, but we must also search for simplifying features, principles of nervous systems that are abstract enough to unify, yet concrete enough to test. And these principles will come from molecular cell biology, using the neuron as a fundamental unit, looking at small circuits, looking at model organisms and testing their predictions in humans, and of course from technologies with scale measurement, which is what you're doing here at the Sudha Gopalakrishnan Center. If I were to compress all this into one thought, it is this: neuroscience is not short on data, but it's short of powerful conceptual bridges, and that's the task before us, to use all this information across evolution, across cell types, to see what new conceptual bridges we can create. Thank you.
I request all the speakers for this session to kindly come on the stage so that we can have a quick Q&A. Moving ahead with the panel discussion for today, the topic is neuroscience landscape across BRICS and extended BRICS nation. I invite all the panelists here, Professor Daniel Gomez-Fillo, Mrs. Natalia, Professor K. Vijay Raghavan, Professor Mo Ming Poo, Professor Manga, Dr. Leena Ali Ibrahim, Professor Mohammad Reza. And also the moderator for the session is Dr. Joshua Davamis. Please.
Okay. Just going to reset the clock quickly, back to 30 minutes. I was prepared for shortening everything, so we'll have a bit more time for discussion and hopefully we can address some audience questions as well, which would be great. So my name is Josh de Vimus. I'm representing South Africa, and I'm happy to moderate this fantastic landscape across the BRICS and BRICS Plus nations panel discussion. I'd just like to quickly introduce our panelists. You know most of them by now. We have Prof. Daniel Gomez de Almeida Filho from Brazil. We have Miss Natalia Podsonaya from Russia. We have Prof. K. Vijay Raghavan, who just spoke, from India. Prof. Muming Pu online from China. Prof. Paul Robert Manga from South Africa. Dr. Lina Ali Ibrahim from Saudi Arabia. And finally, Prof. Mohammed Reza Abul Ghassemi Dehakani. I apologize if I've mispronounced any names. So just to start off with this, I think in 1 or 2 sentences, maybe up to a minute for each speaker, what is the single most exciting neuroscientific development happening in your country right now? And what makes your national research environment distinctive? Maybe we can start with Brazil in order of BRICS, or just down the line.
Hello, can you hear me? Okay. Good afternoon, everyone. It's a pleasure to be here representing my country, Brazil, and with so many people presenting so many different and exciting research. And let me see how can I answer your question. So it's very hard for us to say one specific research because first, we are a very big country. And second, because a lot of people in different countries are doing quite of the same things. We have a lot of collaboration and so on. What I can say that is more exciting about neuroscience in general is that we are devising now to initiate a Brazilian neuroscience and neurotechnology initiative. And it's something that we are planning to do, to launch at the end of the year. It's going to be something that is devised to put forward neuroscience and neurotechnology as a new frontier, or at least the next frontier of endeavor of research and technology that we need to have. So essentially, the idea is that we need to bring closer together the humans and the machines in nowadays world. And the best way to do that is to understand our main tool of communication, which is the brain, and make it more viable to communicate better with the machine. So that's the whole idea.
Thank you. Dr. Lina?
Yeah, hi everyone. So I'm going to speak in general about the Middle East and North Africa region. To me, it seems like there's a lot of emphasis on computational neuroscience as well as neuro-AI. And I feel like there's a lot of emphasis on integrating AI technologies and trying to understand the brain, not only at the molecular level, but as well as all the way from the molecular to the behavioral level. And there's also a lot of emphasis on trying to do this with human subjects and human patients, especially given the very high genetic disease prevalence. There is a big need to try to understand how those genetic variants affect brain development, cognition, and there's a lot of emphasis on trying to integrate AI to try to understand those things.
Prof. Reza?
Okay, thank you. About the most exciting things for me in the neuroscience, I can divide it in different parts. In one terms, as a philosophical view and fundamental perspective, neuroscience helped me to understand myself and quality of experiences, which is really important for me. But in the terms of technology and understanding the world, I think because neuroscience trying to understand a capability of the brain for representation of intelligence is very important for future technology. So similar to you, neuro AI and this kind of stuff is important for me and is most exciting to continue my research.
Good afternoon, everyone. I'm very pleased to be here. Russia was the host country for the second symposium in 2024. And right now you see that this event is expanding and there are more neuroscientists who participate in the symposium, and it's great. If respond to the question, I would say that in Russia, we, maybe as all neuroscientists, feel the key frontiers for neuroscience that are fostered by, of course, health reasons, longevity reasons, talent educations and development, and artificial intelligence development. So the new frontier technologies will be something in the intersection of that. For example, brain-computer interfaces, it's already the technologies that we explore on some or so extents. For example, we all live with our mobiles, with our smartphones, and it's part of us, even we know how to communicate with them. In these terms, BCIs has already exist, and the next frontier will be in molecular machines that will allow us not only cure the neurodiseases, but also communicate on another level. So I make my stand on this technology, molecular machines in neuroscience.
Thank you very much. So what's the most exciting development in neuroscience in India recently? Don't mean to flatter our hosts, but I think the work being done here at IIT Madras is absolutely stunning. It takes technology to looking at the human brain in a manner which was really asking for it to be done, and the way they have grasped it and looked at cellular phenotypes, connections is terrific. I hope this, and I'm sure it will go on to looking at multiple aspects using the tools of imaging, antibody staining, RNA sequencing to understand how complexity in the human brain comes about. Related to this is another venture, also supported by Chris Gopalakrishnan of the Center for Brain Research, is looking at human cohorts and looking at markers of cognitive decline. So these two kinds of studies combined together, I think, will be transformative. It's also a message to those of us who study neuroscience at the molecular and cellular level to be ambitious and take those important steps into connecting with other organisms and take on ambitious new ventures. And someone who's really done that brilliantly is on the screen, that's Moving Poo, who understood synaptic development, axon pathfinding, and took that to now develop a global program on studying primate brains. That's the kind of integrative work we need to partner with across the globe, across India, and get going. Thank you.
Yeah. Prof. Moving Poo, do you want to go ahead?
Yes. Can you hear me?
Yes.
Yes. Okay. So I would like to mention two things that's very exciting in China. The first thing is the mesoscopic brain mapping work at the molecular cellular levels, as just been mentioned. We have new technologies now available to do transcriptomic studies at cellular level, single cell transcriptomics, proteomics, as well as subcellular transcriptomics. Now we know that neuron is very special cells. It has extensive dendrites and axons. How does the transcripts express in different parts of the neuron at different synapses? is a very interesting issue to understand, because each neuron makes connection with many different heterogeneous cell types. So their synapses are different. So by studying cellular and subcellular transcriptomics and proteomics, now we can have better understanding of how the neural circuit works. And this work certainly progressing from model organisms now to primates, to macaques, and even eventually to humans. That would be a long-term goal for not only people here in China, but all over the world. We all want to know the human brain, how does it work at a cellular, molecular, and circuit level. And the work in Madras that we're going to hear more this afternoon, this Brainstem Atlas is amazing. So that's a cellular understanding of brain structure which are not available previously. So I think this is a direction that requires international collaboration, efforts from neuroscientists around the globe to address the molecular, cellular, genetic basis of the nervous system. That would certainly lead to the cure of human brain diseases for sure in the long run. And the other of interest is the brain-machine interface. Now, we know this interface is very important. We are increasingly interact with machines, and the technology of reading brain signals and stimulating brain activities by electrodes has advancing in a rapid rate, rapidly. Invasive technology of multi-channel wireless communication that would lead to much more efficient interaction between brain and the machine. And so there is active work now ongoing in China, which is now in many hospitals into clinical use, using invasive miniaturized multi-electrode arrays, soft electrodes that can produce prolonged long-term recording from patients. Now, this is now used to decode signals from the motor cortex for helping the paralysis patients to control external machines and exoskeleton. And this, I think, it will be used increasingly in the next 5-10 years, increasingly in a medical setting, not only for controlling external device, but using external signals to modulate the human brain, helping in the visual prosthetics. We have very good auditory prosthetics for hearing aids. Now we need to have a way of direct stimulation of the brain, a visual cortex for very rapid, precise interface with the visual cortex with the external visual informations. So invasive brain-machine interface, as well as non-invasive neuromodulation tools are going to be very useful in physical therapy of the brain diseases. Now, we're all waiting for magic drugs for brain diseases, and that has been slow coming. But one of the likely much faster approach of curing the brain is probably the non-invasive physical neuromodulation, as well as the invasive modulation. Good example is the deep brain stimulation that's being widely used. So these two areas, the mesoscopic brain mapping, knowing more about circuits on molecular cell level, and the modulation and reading the brain signals by brain-machine interface. I think this is a new areas of great interest now.
Thank you.
Progressing rapidly in China.
Thank you very much, Prof. So neurotechnologies, neuro-AR, mesoscopic scale, I think there's some exciting things going on. Prof. Manga, from South Africa's point of view?
In South Africa, the community of neuroscientists is very small, and we're quite isolated at the end of the planet there. The most exciting thing happening in neuroscience in South Africa is being part of this group. That's the best thing that's happened in South Africa in the 2.5 decades I've been there.
Thank you. I agree. So we have another 15 minutes. I'm going to kind of swing to a slightly different track question, a little bit more on the controversial side. I'm not going to direct it at anyone, but I'd like you to volunteer if you have a response. So as emerging neuroscience powers, do the BRICS nations have an opportunity to set their own ethical and regulatory standards on issues like neurotech, data privacy, animal research, rather than adopting frameworks that have been designed elsewhere? And should there be a shared BRICS approach, or is a diversity of national standards actually more of a strength? Anyone want to tackle that?
If I may begin with some hot topics that everybody is concerned, how neuroscience would be helping to set the guardrails and governance of AI development. We all know that AI is invading every field. It's helping every field in a positive sense. But one can think that AI, with the rapid development, with the imminent arrival of the artificial general intelligence, which can supersede the human intelligence very soon. How are we facing this new species on Earth? I think there's much discussion in advanced countries, in Europe, in the United States, some in China as well, in the field of AI, about AI governance. Many discussions, international discussion, has not resulted in a consensus on how to govern the AI development so that the AI could be more aligned to human needs rather than impose perils, ill aspects to the society. So I think neuroscience can make a great contribution in this area because we know what intelligence is about. We can monitor intelligence of the AI by our understanding of human and animal intelligence. And by knowing how to monitor intelligence and how to perhaps even intervene in the intelligence development during the appearance of AI, especially physical system with embodied AI intelligence, right? With a general AGI, a general artificial general intelligence, which are, if not well regulated, it would be really a problem for the society. So I think the BRICS country neuroscientists should have an active discussion about AI governance in the future. When we certainly want to promote AI development. In the meantime, the neuroscientists should help the AI community to set a guardrail. For example, if the AI intelligence is embodied into a physical system like a humanoid robots, now how do we know that these robots will not hurt humans? Do they have empathy? Do they have feelings? Do they have theory of mind? Do they have self-consciousness? All these are the subjects of neuroscience for years. We study this. We know how to monitor this aspect of intelligence, which will help the community to really set a goal. When we develop high intelligence, especially in the physical systems, we have to set guardrails. Now, if this is not done, we'll be in trouble. The society will be in trouble.
Thank you.
I don't see anything coming out of the Europe or the United States in terms of governance because they are dominated by commercial interests. They want to get there first, and then the most powerful machine will come out very soon. And how that's going to impact the entire world, we are not clear, right? So I think this is a great issue. I just want to make this point.
Thank you. I agree completely. I think it should be a part of the BRICS roundtable in terms of neuroscience. Prof, did you want to take a crack at it? Expand on that or deal with any other things I asked about?
Yeah, I would think that I think AI is also something very important for us to discuss because this is totally new for humanity as a whole. Like since the past, I would say 3,000 years, we don't have a species that threatens humanity as a whole, in sense of being more intelligent or no. We don't know yet, but we don't even sometimes, it's hard for us to define what intelligence is, if they are more intelligent than us. Sometimes we think, okay, it makes a lot of mistakes, it has some crazy abstractions and so on. Okay, but they don't need to be perfect. They just need to be better than us. So we need to understand better what we are and how our brain works, and understand how can we deal better with this type of technology. But I think the other question that you did is that you made is regarding how should we tackle this problem across the BRICS countries. And I think this is also very important because since we have now a lot of different tools to make high throughput analysis of everything, sometimes we forget the basics to make fundamental questions. Sometimes we just, okay, I have this tool, let's do a lot of this analysis and so on, but we forget to do very specific and fundamental questions. If you look at in the evidence, neuroscience is one of the fields that has the lowest rate of reproducibility. So we are putting a lot of research output, but a lot of them is not reliable. So I think if we really want to go forward with humanity, we need to put our hands together as countries and understand what are the fundamental questions, how should we tackle these questions, what would be the role of Brazil, what would be the role of Russia, what would be the role of India, what would be the role of China, and let's face the problem together and not like everybody going crazy about publishing more and more and more and more. So that's the idea. So if we want to work together on this, we have to discuss the ethics as a whole, not as separate countries. We need to discuss how should we tackle these problems and the ethics about this as a whole.
Yeah, true, very good point. And maybe generative super AI could be our mediator in the future, but absolutely. We have a few minutes, about 7 minutes left. I'd like to open the panel to the audience. If you have a question around the landscape of neuroscience across BRICS or something related, please raise your hand, and also if you have a direct speaker you'd like to address. Any questions from the audience on this broad topic? Well, we've covered so much today. I think in Prof. Vijay's talk just before, yes, he really mentioned this extremity or this extremeness of neuroscience data, but no clear pathway of how to handle these questions. So, yes.
This is to anybody can answer. As a developing country, what can we learn from each other to develop neuroscience and technology in India? Being now in India, that's my question. And maybe Professor Vijay Raghavan, if you can answer. Even though I said anyone, but yeah.
Thank you. You know, there are two ends of the spectrum. In terms of molecular and cellular neuroscience, the growth of those areas in India, and I include physiology, has taken place, I would say, since the 1960s, very modestly, and has grown substantially now, because the labs which established themselves have their academic progeny going on to other places. So overall, I would say, through global interactions with America, Europe in particular, also with Japan, the growth of various kinds of laboratory-based neuroscience has become significant in India, particularly in the last 10 years, as a consequence of previous efforts. At the other end of the spectrum are areas related to human endeavors, such as mental health and human neuroscience. That has been relatively fragmented or concentrated. I mean, we have one National Institute for Mental Health and Neuroscience, and the rest of the neuroscience research at the high-end hospital environment is more directly patient-driven and not broader research program-driven. The National Brain Research Center is rather small. The All India Institute of Medical Sciences have got diverse programs. So we really need to expand that end of neuroscience and integrate it better with the molecular and cellular neuroscience. The third component, of course, is using AI and technology. That is, again, disconnected with all these others. We have extraordinary institutions in a city put together, but those institutions need to talk to each other despite the challenges of commuting and collaboration and so on. And if that's done, I think wonderful things can rapidly happen and can scale up, because all the building blocks are there. They need to come together.
May I add, maybe not just in perspective of India, just for collaboration between our BRICS countries. I will give you example. Right now, we are working, during the last two years, we are working on the big collaboration projects between all our countries, so-called flagship project, to test the mental health, to use different approaches to look at these problems that right now is not solved yet. And we collaborate with Brazil, with China, even with South Africa, and with Iran, and we use different approaches. Those countries like China, who can take data from a lot of patients, they will provide this data more than all other countries. We use different approaches from Russia as metabolomics and lipidomics, and BCIs from Brazil, to just study this disease and how to detect biomarkers of the disease and how to predict if it will be or no, how to find the markers for the better therapies, the exposure for therapies, and so on. And for example, we agreed with South Africa that can use our methodology to test it in clinical. So we exchange not only knowledge, we exchange experience, we exchange our findings, and that's why we teach in each other. And we combine our intelligence, we combine our resources, we combine our infrastructure, how we can use it. So from this collaboration, we will all compete. We will all have successful results, and we will, so there is a reason for collaboration.
Yeah, absolutely, yeah. I think 4 years in, it seems that a lot of collaboration has happened and there's been extreme production in this community. Does anyone want to speak to perhaps some practical barriers that there are between BRIC nations that are beyond the neuroscience, or that we could work together to improve on? You know, the fact that we don't have all members at all meetings at all times is obviously out of control. But you know, as we go forward with these, there might be solutions that we can push towards that allow this to be a smoother interchange. Are there any specific practical barriers, data sharing, funding mechanisms, language, brain drain, or just non-necessities between BRIC nations or within your nation that you could speak to? Anyone? Yeah, please go ahead.
Hi, yes. One thing is that, as you mentioned, there's a lot of variability in the data that is produced, right? And I mean, it's not just country-to-country variability, it's lab-to-lab variability and so on. And so one thing, if we want all our data to be useful for the entire world, and especially the BRICS nations, perhaps standardization of specific protocols as well as other kinds of methodologies across the board, similar to what is happening, for example, in the International Brain Laboratory, which is a consortium of multiple laboratories that have standardized behavior testing in mice. And so then they can actually test for different brain regions that are involved in specific behaviors and so on. And so since there are so many people working on comparative anatomy, all these large-scale efforts for looking at human brain development, primate brain development, and so on, I think a more robust way of sharing protocols, sharing a standardization of protocols, as well as sharing data amongst the different BRICS nations and extra BRICS nations, I think would be very valuable.
Thank you. We have time up? How much time do we have? Finished. Okay. Well, unfortunately, that is time, but I'd just like to thank all our speakers. I realize there are different questions, but we should do things differently. Right. So thank you all for your voices, your opinions, and thank you for listening.
Hello. Now we'll just take a 5 minutes break to set up the stage for our next segment. Hello. Yeah. Okay, good afternoon, everyone, once again. We now move to the highlight of this afternoon, that is the release of the Human Brainstem Atlas. For this, I invite the director of IIT Madras, Professor V. Kamakoti, to please be on the stage.
So friends, a very, very good afternoon to all of you. And again, welcome to this most important function. Friends, I always take pride that IIT Madras, we are really exploring a lot of things. But this particular exploration is something that really puts IIT Madras in the frontiers of the most complex creations that this world has witnessed, the human brain. So I take this opportunity to congratulate the entire Brain Center team. Mohan Shankar and his team, please rise up, and a very great round of applause. I just want to take this opportunity to talk about how some very interesting things that happened in the development of this Brain Center. Friends, this Brain Center was completely conceived and implemented during my predecessor's directorship, Professor Bhaskar, who is here. Sir, please get up. Please give him a great round of applause. In an institution of national importance, the main important thing is the head of the institution must be convinced about that project. And once the head is convinced, the speed at which the project gets implemented is phenomenal. And we really thank Professor Bhaskar. When everybody told that we are going to take a brain, Mohan came and told that we are going to take a brain and going to slice it at micron level and trying to study the structural property, hardly few believed him. And one was Professor Bhaskar. And then there are two great individuals, Professor Vijay Raghavan. Sir, please stand up, please stand up. He was the principal scientific advisor to the Honorable Prime Minister at that point of time. And he fully supported, his office completely supported this project. And the legacy continues. We have a PSA, Professor Sood, who will be addressing you virtually today. He's in the PMO and he'll be addressing you today. And then I call him as the Kamadhenu of IIT Madras. Kamadhenu is a cow which grants any wish that you want. We worship cow in our country, and we go and worship the cow, it grants any boon. That's our scriptures. The Kamadhenu for IIT Madras is Chris Gopalakrishnan. Sir, please get up. Whenever we have shortage of fund, this is a very large sink of income, right? So VLSI, we also call it VLSI. So a lot of money gets spent, and whenever we are short of money, Chris has been always a phone call away. So thank you very much, sir. And we again want your full support, and without your support, it's not going to take forward. So that's how we have the Sudha Gopalakrishnan Center of Brain Research. Friends, this all started like this during the COVID time. When the institute was shut down, Mohan somehow sneaked some people inside, and this brain center was basically built at that point of time. And today, we are very happy. Last year, for the first time in the history of the world, we released Dharini, which is the first 3 trimesters brain sliced at 50 microns, if I am correct, 50 microns, and studied the structural properties. Friends, each brain today, the storage of just the structural property, the structural connections between the regions, today is going towards 2 petabytes. And we have seen smaller brains. And if we actually graduate to large brains, that is, brains that are at the age of 50, 60 above, probably we may touch even 3 petabytes. So NVIDIA, the GPU manufacturer today, the most important company in the world, and Amazon, which actually I think Amazon representatives were here. If they are here, please get up. Or you're there? Oh, yeah, okay. Junjunwala is here. We really can pose challenge, and if at all somebody is going to pose challenge to the GPU world and the storage world, it will be our brain center. So that's why we always say be our collaborator rather than a vendor. That means that should reflect in your pricing also. Right. So with this, last year Dharini was a great success. We also have brains that are affected by rabies, brains that have dementia, Alzheimer's, very good brains. So we now have a way by which we could at least now come to a stage where we can say what happens because of something and what happens to the basic structure of the brain. And a lot of things that they call as neuroplasticity, etc. Now this gives us a platform to understand and simulate and see what could happen. So this is a very, very important first step in understanding this complex brain. Friends, when the supercomputer Blue Gene bet Kasparov obviously was very unhappy about it, but when you look at the parameters, the human brain is the size of a hand and it consumes 20 watts, while the Blue Gene was as big as this particular hall and consumed megawatts of power. So there's a lot more things that we need to understand about our brain, about the retaining capacity, about the storage that we have, and about how do we inference, how do we train it. So many things are still left unanswered when we probe into this great field of artificial intelligence. I am very, very sure that this brain research one day will give some very interesting insights into how we can do AI in a much greener way by consuming less energy and being very fast. And there could be several models. Already we are seeing some things. The neuromorphic computing, which comes out of spiking neural networks, today is at least for the inferencing part, is giving 1000x improvement in energy and also similar orders of magnitude improvement in speed. So we have a very big journey in front of us, and this is just a starting point. And today I'm very thankful to Professor Vijay Raghavan, Chris Gopalakrishnan, and all of you assembled here, and also Professor Sood, who will be joining us online, that we be part of this very important announcement of giving the first ever small but very crucial insight into our stem, brain stem, which we call it as anchor. We are working very fast, swift, at the same time very efficient. As I'm very frugal, by spending the least amount of money, getting the maximum results, less for more, for releasing 100 brain data in the next 1 year to come, which essentially means we'll be consuming 200 petabytes of memory. And this data, when we release, I think this will be a very major step in the understanding of brain and in the development of fields like neurology, etc. So friends, thank you again very, very much for being part of this very exciting afternoon. And let us all work together. This is not something that we alone can do. Let us all join hands. We are ready to collaborate. And as Bharat, as Viksit Bharat, as our Honorable Prime Minister Narendra Modi often says, Viksit Bharat will be an open Bharat. We are releasing this. We released the Dharini. We are releasing Anchor for the public. It's an open source. And we do not want to hide. We do not want anything. All research that are for human good should be available in open. We don't want to claim anything here. We want to work together, and this is an open call. Friends, in the same direction, I'm also very happy to tell you that we have released data related to leukemia, we have released data related to breast cancer, and all these data, the genome database for cancer, breast cancer, and leukemia is now available in public. And what we also found that there are very India-specific, Bharat-specific data, Bharat-specific genome types. And that also explains the medicines that are developed with the genome types we see in the other parts of the world, which we are using here today. That explains why our mortality rate in breast cancer is as high as 35%. Now, this also opens up a very big arena for the pharma industry to work closely together with us to see how we can cure some of these deadly diseases and make the human life as enjoyable and as healthy as possible. So thank you very much, and wishing the team again more and more success. And as an institute, we are all together, Mohan, to support you. Thank you.
Thank you, Professor Kamakoti. Now I invite Professor Mona Sankar to please take the stage.
Thank you very much. Thank you, Kama, for killing my talk. Pretty much everything has been delivered. But before we actually, go ahead and do the so-called formal release of the brainstem, I just wanted to recollect. This is the same place where we had the inauguration of the center. So, I still remember that view, and that has actually gone up like 3x, and it has become truly global, right? And that is essentially the way the center has also evolved. So I'll just give you, in one way, I'm giving you the state of the center as it started, because many of the people who were there in the inauguration are here. So give you a sense of how it started. It was instigated and inspired by Chris, somewhere in 2017. And Chris may not remember this, but my director Bhaskar, Chris, and we were together. And he sort of said, I'm paraphrasing here, but pretty accurate. But the last part of it was truly, quite visionary, I would say, because we did not have too much work in human neuroscience in India or IIT Madras. But the way he said, if we can do something which today is not possible, like what Vijay had said during his thing, if we can develop a tool that can really open up something significantly, I will support it. Right? So thank you, sir, for not only inspiring us, but sort of being a pillar to the center today on which it rests. Now, the inspiration is one thing, but then you have to take it forward. I went to Bhaskar and said, hey, how do we do this? Right? And, a lot of people ask, how are you able to do so many things? Do you have a boss? My boss is sitting here. But actually, that's perhaps the nature of the institute. We said, I don't know how to do this. So he actually sat down with me, and I still carry that piece of paper. We pretty much derived how a human brain imaging system in a large scale will work out. And suddenly we realized this is really large scale. Not only it's petabytes across kilobytes. Right? So you need a system, both engineering and computing, that can resolve at kilobyte, at micron, but do petabyte. So, then the PSC office came into the picture, and if you look at the proposal title, that itself tells you, we were not so confident that we'll actually be able to do brain mapping at this level. So we said, at least we will propose to build this technology. And so it started, but really what happened was, everybody said no. Right? And Vijay recently made a public comment, so, with your permission, Vijay, I'm quoting it. It's one thing to be ambitious, but somebody has to actually also support that ambition, and that happened through Vijay. Then COVID came. Right? So in fact, the proposal sanction came in February 2020, and within 3 weeks, I get the lockdown order from pretty much the same agency. Then what we decided is, anyway, this is such a high-risk project, so why don't we take more risk? So this entire technology platform was built during COVID times. I must say, as he said, politely sneaking people in. I've sneaked a lot. And then Richa joined us. Right? And Richa and Jairaj, my colleague, built out a platform, and what is today a world-leading platform, of imaging whole human brains at cellular resolution at a very high throughput. And today we are able to do almost a throughput of 1 adult brain per month. And we are engineering this and scaling this in the next few months to probably 4 or 5 months. We'll be able to do 1 adult brain throughput per week. And so you can imagine the triple use and the quadruple use of that. And what we did not realize is, probably we didn't expect it to succeed at that level. Suddenly it is now producing gigapixel images, teravoxel volumes, and petabytes of data. So it turns out the hardest problem that we had to solve is to build a computing platform, not just to do analysis, that comes much later, even to deal with the gigapixel image. And I will say this to many youngsters here, how will you rotate a gigapixel image? Because if you don't rotate anything, you can't do anything. Right? It's a hard computer science problem. And suddenly we realized we have to build out almost what Google Maps had done way back in 2000s. And sitting here in an academic institution, is it even possible? But then came Kirti and Shiva, who were somewhere here, and we have built something which today is probably one of the most advanced computing platforms that can take in gigapixel images every minute almost, and can process various things. I won't go into the details, but resolve at cellular level, which is about half a micron, which is about few kilobytes. So with that, the first data, and we were always clear, and Kama mentioned this, we were always clear, and Chris and I were absolutely clear on this, that we will make all these data and resources fully public. And the first set of release that happened in the form of Dharani, which we picked it because that's where the massive change happens between 12 weeks, 14 weeks, all the way to 24 weeks. And it was released, and then the global visibility started to happen. There is a very nice, kind editorial by the editor of Journal of Comparative Neurology, which said, India's gotten a seat at the table. Now, then we started to get even more ambitious, Vijay, saying, if we can do this, why don't we do this across the entire human lifespan? Right? It's a big engineering challenge. And we were not unaware of the fact that this will require a huge amount of funding. Right? And I will come to it. And part of this celebration of people who are here is how we got there. And we are pretty much halfway through, I must say. Right? Give us another 18 to 24 months. We will have pretty much, it comes to around 120, 130 brains, about 30 categories of brains. And we want equal representation of genders, which is missing severely when it comes to brain data. And here we are. And there are 3 things which I wanted to mention, which is, the center may be at IIT Madras. It's actually truly a global center, right? In terms of capabilities, in terms of its attitude, in terms of people. As you can imagine, I have listed some of the fields here which are working together. This would not have been possible even if we formally all worked together. But the way it happened is because it was so informal. People drove it, then institutions followed. Okay? And today, this map needs some more updation, especially after this gathering. Today, we have close to almost 40, 50 global collaborators, some of who are listed here, and who are working with us not as a division of labor type of collaboration, but a collaboration almost at an intellectual level where every day we are solving problems together. And that is in this particular anchor that we are releasing today. It is truly a multinational effort. In fact, many of our global collaborators spend physical time here. Sometimes I cannot distinguish whether Paul is South African or, he's practically become Indian. And, he loves Tharmani guest house food more than his home food, perhaps. Okay? Now, I want to make another important point here. Okay? Many times when we start these kind of projects, obviously the government has to take the first risk. And after that, the government says, see, I have funded you, now I have to fund different people and other people. But this is one of those projects which truly has exemplified what is possible if we get private and philanthropy to support very, very hard science, but that will move the needle significantly. So close to 20 private and philanthropic organizations have funded us. I always tell this to people, one thing is money, but what I really salute you, and it's very hard for me to describe this, I can get emotional, the courage to take this kind of risk where things might have failed. There are so many points of failure. Just to give you a sense, there are close to around 140 parameters that have to get right in order for you to deliver this kind of maps. Right? And everybody is listed here in sort of chronological order. And my deepest gratitude, not just for the people who supported this, but for their organizations that had a philosophy that we will support this kind of advanced science. So I will stop there. Now, what is the larger purpose that sort of drives us? Right? Of course, we are releasing these results. We make scientific discoveries and findings. The thing that probably unites all of us, including the people here, is that there is an acknowledgement here that in this field, and which I mentioned this, you need to have quite a transformative brain science in order for you to make applications work. This cannot happen in some other fields where you get some incremental thing and you may be able to build tools. This will require significant advancement in science, and that's what we are after. And we will walk this path. We are absolutely committed, and we really want you to support us in every possible way in this journey. Thank you very much.
Thank you, Professor Mohan. We always believe that an atlas of this magnitude is not the work of one mind. It is the work of many dedicated hands and brilliant minds. I would now like to invite the team behind the Brainstem Atlas to please come forward. I would also call upon stage Shri Chris Gopalakrishnan and Professor V. Kamakoti to officially release the Brainstem Atlas. Sorry, before that, we'll just release the video for the Brainstem.
The brainstem that connects the cerebrum and the spinal cord is one of the most crucial parts of the human brain. The brainstem includes control centers for vital function. IITM's Sudha Gopalakrishnan Brain Center is releasing the most comprehensive multimodal 3D atlases of the human brainstem to date, spanning from the prenatal period to childhood to adult brains.
So the larger mission of the center is to map human brains at cellular resolution. But not just regions of the brain, but the whole brain. They're doing it at about half a micron. The whole human brain. Visualizing it, which means we'll be able to see it, so digitally converting this to an image. Then overlaying all this image to create a true 3D structure of the human brain, which you can see as if you're inside the human brain.
These are the most detailed and comprehensive maps of the human brainstem made available publicly in digital form. These atlases encompass more than 200 brainstem nuclei and fiber tracks, built from hundreds of serial sections. To resolve distinct neurochemical cell types, 8 complementary immunostains were overlaid across more than 500 sections, enhancing the detailed mapping. Identifying the specific cell populations affected in brainstem lesions will be critical for clinical applications. The 3 specimens analysed that are from different age groups were acquired with due consent and institutional approvals.
The challenges were in optimizing the protocol for a larger tissue versus a smaller tissue. A lot of our adult brains, what we are processing at this center, are diseased brain. Again, that requires a different optimization.
Histological processing, digitization, quality control, and atlas annotation were done at the Brain Center in IIT Madras. All 3 specimens were fixed in 4% paraformaldehyde and subsequently cryoprotected with graded sucrose solutions to minimize freezing artifacts. Prior to cryoprotection, 3D surface scanning of each brain was performed to generate a custom cryo mold that was utilized to freeze the specimens in an OCT medium at -80 degrees Celsius.
So the first one is purely the methodological one, how you cut the brainstem. The second is that the brainstem contains a lot of fiber tracts, a lot of pathways, if you want, and many nuclei are hidden inside of these fiber tracts. The third is that the brainstem contains many nuclear formations or neuronal formations, more than 200. And many of these are very, very small in the human, and one has to have a lot of patience looking for them. The fourth one is that for humans at least, the brainstem is not so well studied. The human brainstem is not so well studied. So there is little, let's say, literature on the human brainstem nuclei.
Cryosectioning was carried out using the tape transfer technique and cover slipped using custom-developed robotic cover slippers. To establish a 3D reference volume, the exposed block face was imaged during sectioning using the block face imaging technique. The staining of the serial tissue sections consisted of Nissl, hematoxylin and eosin, myelin, and 8 different immunohistochemical markers. Brainstem sections were manually annotated to generate high-quality atlases. The annotations were made using a custom in-house online annotation tool, and the brain structures were divided into 5 general classes. The difficulty with the brainstem is there's so many different parts to it. We know what a lot of those things do, but between different animals and in humans, they can vary quite a bit. So knowing all those different parts, it's like one of the most complex Rubik's cubes. The process begins with MR imaging to establish the global anatomical reference, followed by block-face imaging that captures the physical geometry of the tissue block during the sectioning process.
Raw data is actually 2D. It is serial sections of 2D, but then we reconstruct the 3D. Our own teams are putting it together in 3D by performing 3D registration reconstruction. Once we do that, we also register it to the MRI so that we get a proper 3-dimensional reference space through which we can compare across brains also. Once we form that, we actually utilize our specialized tools, some of which we actually collaborated with companies like NVIDIA to custom build for our use cases. So once the data is ready, then we use these tools and then we make it available online.
To make these high-quality atlases practically accessible for researchers globally, we have developed a web-based low-footprint viewer. By establishing precise spatial correspondence across these modalities, the viewer enables a seamless transition from gross brain structures in the MR images to features at the cellular level. This unified coordinate system allows navigation across adjacent histological sections, from Nissl to the 8 immunohistochemical stains for all the annotated sections simultaneously. The online viewer offers synchronized side-by-side panes that align with identical spatial coordinates, enabling users to concurrently zoom, pan, and examine high-resolution cellular insets of Nissl and immunohistochemical stains while preserving macroscopic content through co-registered MR images and block face images. Cross-referencing these complementary visual cues enables researchers to confidently identify anatomical borders of complex neuronal parts and validate macro to micro registration across imaging modalities. Using this detailed primary data, we have developed a 3D brainstem foundational model across brains using Oak Ridge National Laboratory's supercomputing facility. This model of the human brainstem is the world's first cell-level 3D brain model. Key findings include the identification of protoplasmic commissure dendrites of the hypoglossal nucleus, previously only identified in the African wild dog, and the distribution of specialized catecholaminergic neurons in the human brainstem. To our knowledge, this is the first time that the protoplasmic commissure dendrites of the hypoglossal nucleus have been identified in the human brain. The pretectal nuclei in the 25 gestational week fetal brainstem are also described in detail for the first time.
In addition to neurotypical brains from fetus to old age, we are focusing on diseased brains. And today we are releasing Anchor, which is detailed map of the brainstem of humans across lifespan. And this is a very important milestone in this mission of mapping 100 human brains.
Future developments of Anchor will include extended sets of immunohistochemical stains and applying in situ hybridization techniques as well as transcriptomic maps. We envision that these maps and atlases will have significant implications for neuroscience and neuromedicine.
I now call upon the brilliant team behind all this massive work, and also Sri Chris Gopalakrishnan and Professor V. Kamakoti to officially release the Brainstem Atlas. Thank you.
Thank you. It is somewhat emotional to me for various reasons, so you'll have to pardon me on some things. I don't know how many of you noticed that there are many unsung heroes and heroines in this, but there's one set which you probably never realized, right? Which is, where did this brain come from, right? Yeah, I sneaked people in, but let me assure you that we didn't get the brains from them, right? This is post-mortem human brains, and a lot of people don't realize that the clinical community and the clinical ecosystem, which goes way back from our HTIZ days, came forward and said, we will help you do this. Now, you may wonder why it is so. They get nothing out of this, right? Generally, when you work on a biomedical technology or a solution, usually you have a direct and immediate benefit to the patients. But something like this is truly a selfless exercise where they really have to work very hard. And I don't want to go to the details. Getting a high-quality post-mortem extraction of any brain structure is extremely challenging. In fact, the thing which I didn't have time to mention is, after I spoke with Bhaskar, the person who gave me the confidence is somebody who's sitting very coolly there, George Verghese of CMC Vellore. You will see him briefly. He said, we will get you the brains. I said, it has never been done before. Nobody knows how to extract a high-quality, one full brain, right? Not like chopped pieces. And not only did he develop the protocol during the infection days of COVID, it today is considered the method of extracting and acquiring human brains. And it is, in fact, we teach this protocol to various institutions in the US right now. So, it's one thing for us to do this work at IIT Madras, which is fine. I consider that as our responsibility. It's another thing for these clinicians to really do this and always stay behind the scenes. So I want to call upon Dr. George Burgess and his team and Dr. Suresh of Mediscan. Right. Yeah. So I have a team which exactly tells me the next word even before I say it. Dr. Rajeshwaran of Ramchandra, Dr. Vinod of KMC could not be here. Right? So I want the entire clinical team that is here to come to the stage, please. Because this is perhaps the only time you see their faces in any of the work or wherever. Right? So I want to invite Kama and Chris to come, and it's hard to give anything of token of respect or gratitude, but at least as a memory in time that we would like to honor them.
With the Atlas now formally released, we are delighted to hear from two distinguished voices in global neuroscience who have witnessed this journey. I invite Professor Shubha Tole to share her reflections.
Well, the hippocampus is required for spatial navigation, and I clearly demonstrated that I do not have one. It's an honor and a privilege to be a neuroscientist at a time like this. And it's a particular privilege to be an Indian neuroscientist at a time like this, from a generation that literally had to leave the country to seek PhD training abroad because research training in this particular field was not well developed at the time, to now see a thriving, flourishing, visionary program that puts India literally, to borrow words, at the seat of the international table and leads the way in examining the human brain and studying its intricacies at a depth that could not have been imagined. To see the confluence of people from engineering, from neuroscience, from medicine, how seamlessly everybody has come together. we see it as one atlas, but clearly it reveals an integration that is unprecedented and so rewarding to see, particularly in our community where researchers and clinicians sort of lived two separate lives, two separate training programs, and to see everybody meet like this at a venture unprecedented in its conception and its execution. I share and celebrate everybody's joy and enthusiasm and privilege to be a witness to this. Happily, I offer whatever support I can offer in my capacity as a neuroscientist. I encourage young people, the next generation, to take inspiration from this and carry the baton forward. Not everybody necessarily has to become a neuroscientist or a clinician. Please, some of you, go forth and set up fantastic careers in something that may not even be related to science, but then connect via philanthropy so that you can create initiatives like this. We can all take forward humankind's progress in exploring what each of us individually does best. And I say humankind because another thing that the center has shown very clearly is that national boundaries are pretty much just little squiggles drawn on a globe by happenstance and circumstance. But collectively to be part of this international effort is something that the young should take inspiration from. And I look forward to seeing where this goes next.
Thank you, Professor Tole. I have Professor Mo Ming Pu online to share his reflections and thoughts as well.
Yes.
Yes, we can hear you.
I'm very happy and honored to participate in this ceremony, and I'm particularly impressed with such a rapid progress of the brain center in Madras about this Human Brain Atlas. It is the beginning of a long journey. I think brainstem is very important to the human body. It's a connection between the brain and the spinal cord and all our motor movements. It also controls a very important aspect of our physiology, how we become awake, how we get into sleep, how we breathe. They're all controlled by nucleus centers in the brainstem. I think the choice of release the first atlas in the brainstem is a very important, a very wise choice in this grand effort. I recall maybe decades ago, I had an opportunity to meet Professor Vijay, who is present here in Bangalore. I remember discussing his vision about the brain science, neuroscience in India. And I think he will be very happy now as one of the person behind the scene to promote this effort, to have this rapid development of brain initiative in India. In fact, I also very much amazed by the perfect marriage of basic neuroscientists, clinical neuroscientists, and the engineering and computing talents in India that make this achievement possible. And I think this is a good example of interdisciplinary work that we all drive for in brain science. And I think this beginning we'll be seeing more coming out of the platform here, and I hope to soon to be able to visit the Madras Center to see how this wonderful work is carried out. And also very pleased to see that this is probably the first best example so far about BRICS country neuroscientists collaboration. In fact, we begin to discuss a BRICS country neuroscientist consortium a few years ago, 3 or 4 years ago, and this is the first thing we can show, in fact, in this consortium. So I want to congratulate in particular Mohan and Richa and Paul Menger from South Africa, whose joint effort make this achievement possible. So I would like to encourage young people, as our previous speaker mentioned, the young people who are interested in brain science and neuroscience to join forces in the coming years, because this is an area for the 21st century. We not only to solve the problem of biology in understanding the brain, but also the very important social problems of brain disease intervention, diagnosis and intervention. All depends on our understanding of the brain at the molecular, cellular, and circuit level. And this effort of brain studies, which is coming soon, about the 100 brain mission at Madras, is a very important effort. And I'm looking forward to future collaboration involving not only India and South Africa, but with China and Russia and Brazil and other BRICS country scientists to work together. Because this is a big, huge endeavor of neuroscience. And I think today we have witnessed a very important step in this direction. So I congratulate the achievement, and I also hope to promote further collaboration within the BRICS scientists toward a common goal. And thank you.
Thank you, Professor Moving, for being with us here. The Sudha Gopal Brain Krishnan Center exists today because of the extraordinary generosity, trust, and vision of one person. It's my privilege to invite Sri Chris Gopal Krishnan, co-founder of Infosys, distinguished alumnus of this very institution, and the founding patron of our center, to address us.
First of all, congratulations, Mohan, Richa, the entire team on the successful release of Anchor maps. You know, this is what it's all about for us. You know, the result of all your hard work. Our support is a small thing, you know, the hard work that you people are doing, you know, the effort that has gone in conceptualization, creation of this center, a lot has gone in, and thank you very much. Really appreciate the work that you have done, and I hope you know, this will make an important contribution to neuroscience in the world. Second, let me thank all the participants who have come from across the BRIC nations, across the world, to participate in this. Your participation, your being here on the occasion of the release is a huge encouragement for, I believe, all of us, huge encouragement for this center, that you are all here when this significant event has happened for this center. A lot was talked about, how, this is a step in understanding the brain, understanding the science behind it, neuroscience, understanding how this is going to help us with neurodegenerative diseases, etc. There is also talk about standardization, harmonization, data sharing, protocols, etc. All of that, I think, must happen, and an event like this, I think it's very important to actually collaborate on that. I always have believed that international collaboration, us all coming together to work on problems facing all of us as society, is the way to solve these problems. In India, we have this awareness of affordable science, affordable technology, because ultimately technology has to serve society, has to serve people, and accessibility, affordability, inclusiveness, ethics, all of these things are very important. And I hope that this proves that point. This demonstrates that we can come together to work on major problems facing humanity. Now, Professor Kamakodi mentioned something, and that's going to prompt me to put a new challenge in front of the team, because understanding the brain is what created the entire field of, or trying to understand the brain is what created the entire field of computer science. Two people who created the fundamental theorems behind computer science are von Neumann and George Boole. George Boole wondered about the brain and said, brain has to be a logical machine. And he created Boolean algebra. Okay? Thinking that we think, in Boolean bits and bytes. And von Neumann thought that there are different parts of the brain with different functions. There is a part of the brain which does computation. There is another part of the brain that does the programming, etc. And he came out with the von Neumann architecture. Today we know that both of them were wrong. So two wrongs created the entire field of computer science. But that's how it is actually. Similarly, the entire AI and machine learning came out of us wondering about neural networks and how learning happens, etc. We still don't know how learning happens. We still, I believe, don't know exactly how brain stores information, etc. But the thing is, nature over millions of years have created this amazing machine. Kama mentioned that it's 20 watts of power, 2 pounds in weight, right? So I actually looked up, the problem that the world is facing today. The power cost of running AI. 1 gigawatt, it requires multiple gigawatts. 1 gigawatt of data center to be set up is estimated to cost $38 billion. 1 gigawatt of power is estimated to cost $38 billion. The current estimate of simulating the brain, how much power does it take? It's 2.7 gigawatts. And to set up a computer center equivalent to 1 human brain, the cost is $103 billion. That is 2.7 into this. $103 billion. Somebody has given us all this brain free of cost. We all are born with it, actually. Though we don't understand it. Another interesting data point. The current scientific estimates of the human brain storage capacity ranges widely, but it's estimated to be around 2.5 petabytes. Mohan, correct me if I'm wrong. 2.0 petabytes, and the storage, Amazon people are here, is estimated to be $0.10. So 10 cents per gigabyte per month is what is the cost of storage. So at that cost of storage, the cost of our brain is about $3 million per year. The storage cost of one human brain is 3 million. Again, we have got it free of cost. But really what I want to say is that this is an opportunity for us. Of course, we are looking at understanding the brain and trying to find solutions for neurodegenerative diseases, brain disorders, etc. But similarly, we have to think about how can we create better models of computing also by understanding the brain. Can we collapse the cost, maybe not to 20 watts, but at least few hundred watts if possible. And somebody mentioned about molecular computing, biological computing, etc. These are all possibilities. And this is what I hope together we can explore. And this is what I hope this center will evolve to in IIT Madras, to become a full-fledged center which looks at brain and looks at brain-inspired science, which will, I believe, solve many of society's problems on computing, affordable computing, society's problems on neurodegenerative diseases and things like that. I want to thank all the people who have come forward to donate to this center. We in India have to support research, have to support science, have to be an active creator of technology rather than just consumer of technology. We have to become very active in creating technology, and we have an important role here. Government only can do so much. I think private philanthropies, private sector, industry, all will have to come forward and support world-class research. And the names that came up on the screen is like a who is who. I hope that more people come forward. We need to raise our research expenditure to 3% of GDP at least. Right now it's 0.7% of GDP. So I'm very glad that these people have come forward to support this center, but support not just this center, but any research initiative that you feel is worth doing, which you can support. I also want to thank all the technology providers. Your work with this center, I can, on behalf of IIT Madras, on behalf of the people, Mohan and team, can say that will allow them to think big. Whatever cost that can be offset, we would like to think of this as your contribution to the society through this center. So please support this center in any way you can. We need all the support that we can get. In fact, if the center becomes so successful, all the investment that is going into data center is going to actually go waste. Right? So think about that, but you can make it happen. Finally, all the people who are participating today, thank you. You need to keep inspiring these people, supporting these people, give them confidence that we are all behind them. That's what we can do. They will do the science. What we can do is that we are behind them, supportive of all the work that they're doing. This is true for all scientists anywhere, actually. Society must support scientists, and I want to thank all of you for coming today. Thank you very much.
Thank you so much. We are honored to present a special address by Principal Scientific Advisor to the Government of India, Professor Ajay Kumar Sood. He couldn't be here physically, so he has recorded a talk for us.
A very good afternoon to all. Dignitaries on the dais, Mr. Chris Gopalakrishnan, Professor V. Kamakoti, Professor Mohan Sankar Siprakasam, international faculty from the BRICS country, Brazil, Russia, China, South Africa, Saudi Arabia, Iran, and Cuba, Indian faculty from the leading life sciences and medical institutions, CSR leaders from industries, philanthropists, and the faculty and research staff from the IIT Madras Brain Center. It is my pleasure to join you, though in virtual mode, on this very important and momentous occasion of the release of the world's most detailed 3D atlas of the human brainstem. I am happy to see yet another milestone achieved by the IIT Brain Center after it had released Dharani, the largest publicly accessible digital data set of the human fetal brain last year. An important feature of the IITM Brain Center is that it has been a multi-institutional, multinational, as well as a multidisciplinary endeavor with the support of the government, industry, and philanthropy. The center has become an exemplar for research and innovation through collaboration and a diversified support system for long-term sustainability. IITM launched its cutting-edge brain center in 2022 with seed support from my office till January 2025, which was actually initiated during the tenure of my visionary predecessor, Dr. K. Vijayaraghavan, who is also present in the audience today, and with funding support from Mr. Chris Gopalakrishnan, with the aim of powering a large-scale multidisciplinary effort in the fields of science, technology, computing, and medicine to map human brain at cellular level. The center is a unique example of how risk-taking by a public agency led to an advanced technology platform for doing big science, and that was then scaled by private and philanthropic support to produce world-class results in frontier areas of human brain sciences. Another noteworthy feature to talk about the collaborative aspect of this project is that various medical institutions in the country, such as CMC Vellore, Mediscan Systems, Kilpauk Medical College, Shri Ramchandra Institute of Higher Education and Research, helped the center to acquire over 350 brains of different types, ages for this project. To mention a little bit of science, the latest achievement by IITM Brain Center called ANCHOR, Atlas of Neurochemical Characterization of the Human Brain System, brain stem, comprises the most comprehensive multimodal 3D maps and atlases of the human brain stem to date, spanning from the prenatal period to childhood to adult brains. This is indeed a very significant accomplishment in the field of neurobiology, considering the important role played by the brain stem in controlling the vital organs and functions of the body, such as sensory and motor functions, brain processing, heart rate monitoring, thermos-regulation, and breathing. This work by IITM Brain Center, which is a multimodal framework integrating MRI, histology, and detailed chemo-architectural analysis of the human brain stem, will be perhaps the most detailed and comprehensive maps of the human brain stem, and they will be made available publicly in digital form. These maps would help in identifying specific cell populations affected in brain stem lesions, which will be critical for clinical applications. Most importantly, these data sets will provide robust ground truth resources for the development of advanced computational algorithms and their future clinical translation, which will be useful in the diagnosis and treatment of brainstem lesions and related neurological disorders. The Dharani platform, which I had mentioned earlier, attracted leading brain researchers from institutions in US, Germany, Croatia, Romania, South Africa, Australia to work closely with the IITM Brain Center. And having become visiting faculty at IITM, they made valuable contributions to the Anchor platform also. The support this brain project has been able to garner in terms of funding and collaborations with national and international institutes and researchers speaks volume about the robust technological backbone built by the IITM, which involves high quality acquisition of brains, histological processing, digitization, quality control, annotation, multimodal visualization, AI-driven analysis and modeling. This project is an excellent illustration of how complex fields like neurobiology can be approached and scaled by applying engineering methods built on advanced digital and AI technology. In the end, let me make a wild suggestion. Apart from the tremendous impact these efforts can have on the clinical side by aiding diagnosis and treatment of neurological disorders, can IITM also explore how these detailed brain atlases can add a thrust to R&D efforts in the area of neuromorphic computing? To conclude, I would just like to say that the IITM Brain Center, which has earned India a seat at the table of human brain cartography through its efforts, is in a unique position to attain global leadership by imaging more than 100 plus human brains and generate the largest repository of human brain data in the world that will serve as a reference for researchers worldwide for decades to come. I wish all the very best to the future of the center for path-breaking science and technology and eventual benefit to the mankind. Thank you and Jai Hind.
Thank you, everyone. I now request everyone to please proceed forward for the high tea that has been arranged, and will commence a special cultural event which will begin at 5 p.m.
The brainstem that connects the cerebrum and the spinal cord is one of the most crucial parts of the human brain. The brainstem includes control centers for vital function. IITM's Sudha Gopala Krishnan Brain Center is releasing the most comprehensive multimodal 3D atlases of the human brainstem to date, spanning from the prenatal period to childhood to adult brains.
So the larger mission of this center is to map human brains at cellular resolution. But not just regions of the brain, but the whole brain. They're doing it at about half a micron. The whole human brain. Visualizing it, which means you be able to see it, to digitally converting this to an image. Then overlaying all this image to create a true 3D structure of the human brain, which you can see as if you're inside the human brain.
These are the most detailed and comprehensive maps of the human brainstem made available publicly in digital form. These atlases encompass more than 200 brainstem nuclei and fiber tracts, built from hundreds of serial sections. To resolve distinct neurochemical cell types, 8 complementary immunostains were overlaid across more than 500 sections, enhancing the detailed mapping. Identifying the specific cell populations affected in brainstem lesions will be critical for clinical applications. The 3 specimens analysed that are from different age groups were acquired with due consent and institutional approvals.
The challenges were in optimizing the protocol for a larger tissue versus a smaller tissue. A lot of our adult brains, what we are processing at this center, are diseased brain. Again, that requires a different optimization.
Histological processing, digitization, quality control, and atlas annotation were done at the Brain Center in IIT Madras. All 3 specimens were fixed in 4% paraformaldehyde and subsequently cryoprotected with graded sucrose solutions to minimize freezing artifacts. Prior to cryoprotection, 3D surface scanning of each brain was performed to generate a custom cryo mold that was utilized to freeze the specimens in an OCT medium at -80 degrees Celsius.
So the first one is purely the methodological one, how you cut the brainstem. The second is that the brainstem contains a lot of fiber tracts, a lot of pathways, if you want, and many nuclei are hidden inside of these fiber tracts. The third is that the brainstem contains many nuclei, many nuclear formations or neuronal formations, more than 200. And many of these are very, very small in the human, and one has to have a lot of patience looking for them. The fourth one is that for humans at least, the brainstem is not so well studied, the human brainstem is not so well studied. So there is little, let's say, literature on the human brainstem nuclei.
Cryosectioning was carried out using the tape transfer technique and cover slipped using custom-developed robotic cover slippers. To establish a 3D reference volume, the exposed block face was imaged during sectioning using the block face imaging technique. The staining of the serial tissue sections consisted of Nissl, hematoxylin and eosin, myelin, and 8 different immunohistochemical markers. Brainstem sections were manually annotated to generate high-quality atlases. The annotations were made using a custom in-house online annotation tool, and the brain structures were divided into 5 general classes. The difficulty with the brainstem is there's so many different parts to it. We know what a lot of those things do, but between different animals and in humans, they can vary quite a bit. So knowing all those different parts, it's like one of the most complex Rubik's Cubes. The process begins with MR imaging to establish the global anatomical reference, followed by block-face imaging that captures the physical geometry of the tissue block during the sectioning process.
Raw data is actually 2D. It is serial sections of 2D, but then we reconstruct the 3D. Our own teams are putting it together in 3D by performing 3D registration reconstruction. Once we do that, we also register it to the MRI so that we get a proper 3-dimensional reference space through which we can compare across brains also. Once we found that, we actually utilize our specialized tools, some of which we actually collaborated with companies like NVIDIA to custom build for our use cases. So once the data is ready, then we use these tools and then we make it available online.
To make these high-quality atlases practically accessible for researchers globally, we have developed a web-based low-footprint viewer. By establishing precise spatial correspondence across these modalities, the viewer enables a seamless transition from gross brain structures in the MR images to features at the cellular level. This unified coordinate system allows navigation across adjacent histological sections, from NISL to the 8 immunohistochemical stains for all the annotated sections simultaneously. The online viewer offers synchronized side-by-side panes that align with identical spatial coordinates, enabling users to concurrently zoom, pan, and examine high-resolution cellular insets of NISL and immunohistochemical stains while preserving macroscopic content through co-registered MR images and block face images. Cross-referencing these complementary visual cues enables researchers to confidently identify anatomical borders of complex neuronal parts and validate macro to micro registration across imaging modalities. Using this detailed primary data, we have developed a 3D brainstem foundational model across brains using Oak Ridge National Laboratory's supercomputing facility. This model of the human brainstem is the world's first cell-level 3D brain model. Key findings include the identification of protoplasmic commissure dendrites of the hypoglossal nucleus, previously only identified in the African wild dog, and the distribution of specialized catecholaminergic neurons in the human brainstem. To our knowledge, this is the first time that the protoplasmic commissure dendrites of the hypoglossal nucleus have been identified in the human brain. The pretectal nuclei and the 25 gestational week fetal brainstem are also described in detail for the first time.
In addition to neurotypical brains, from fetus to old age, we are focusing on disease brains. And today we are releasing Anchor, which is detailed map of the brainstem of humans across lifespan. And this is a very important milestone in this mission of mapping 100 human brains.
Future developments of Anchor will include extended sets of immunohistochemical stains and applying in situ hybridization techniques as well as transcriptomic maps. We envision that these maps and atlases will have significant implications for neuroscience and neuromedicine. The brainstem that connects the cerebrum and the spinal cord is one of the most crucial parts of the human brain. The brainstem includes control centers for vital function. IITM's Sudha Gopalakrishnan Brain Center is releasing the most comprehensive multimodal 3D atlases of the human brainstem to date, spanning from the prenatal period to childhood to adult brains.
So the larger mission of the center is to map human brains at cellular resolution. But not just regions of the brain, but the whole brain. They're doing it at about half a micron. The whole human brain. Visualizing it, which means you might be able to see it, so digitally converting this to an image. Then overlaying all this image to create a true 3D structure of the human brain, which you can see as if you're inside the human brain.
These are the most detailed and comprehensive maps of the human brainstem made available publicly in digital form. These atlases encompass more than 200 brainstem nuclei and fiber tracks, built from hundreds of serial sections. To resolve distinct neurochemical cell types, 8 complementary immunostains were overlaid across more than 500 sections, enhancing the detailed mapping. Identifying the specific cell populations affected in brainstem lesions will be critical for clinical applications. The 3 specimens analysed that are from different age groups were acquired with due consent and institutional approvals.
The challenges were in optimizing the protocol for a larger tissue versus a smaller tissue. A lot of our adult brains, what we are processing at this center, are diseased brain. Again, that requires a different optimization.
Histological processing, digitization, quality control, and atlas annotation were done at the Brain Center in IIT Madras. All 3 specimens were fixed in 4% paraformaldehyde and subsequently cryoprotected with graded sucrose solutions to minimize freezing artifacts. Prior to cryoprotection, 3D surface scanning of each brain was performed to generate a custom cryo mold that was utilized to freeze the specimens in an OCT medium at -80 degrees Celsius.
So the first one is purely the methodological one, how you cut the brainstem. The second is that the brainstem contains a lot of fiber tracts, a lot of pathways, if you want, and many nuclei are hidden inside of these fiber tracts. The third is that the brainstem contains many nuclei, many nuclear formations or neuronal formations, more than 200. And many of these are very, very small in the human, and one has to have a lot of patience looking for them. The fourth one is that for humans at least, the brainstem is not so well studied, the human brainstem is not so well studied. So there is little, let's say, literature on the human brainstem nuclei.
Cryosectioning was carried out using the tape transfer technique and cover slipped using custom-developed robotic cover slippers. To establish a 3D reference volume, the exposed block face was imaged during sectioning using the block face imaging technique. The staining of the serial tissue sections consisted of Nissl, hematoxylin and eosin, myelin, and 8 different immunohistochemical markers. Brainstem sections were manually annotated to generate high-quality atlases. The annotations were made using a custom in-house online annotation tool, and the brain structures were divided into 5 general classes. The difficulty with the brainstem is there's so many different parts to it. We know what a lot of those things do, but between different animals and in humans, they can vary quite a bit. So knowing all those different parts, it's like one of the most complex Rubik's cubes. The process begins with MR imaging to establish the global anatomical reference, followed by block-face imaging that captures the physical geometry of the tissue block during the sectioning process.
Raw data is actually 2D. It is serial sections of 2D, but then we reconstruct the 3D. Our own teams are putting it together in 3D by performing 3D registration reconstruction. Once we do that, we also register it to the MRI so that we get a proper 3-dimensional reference space through which we can compare across brains also. Once we found that, we actually utilize our specialized tools some of which we actually collaborated with companies like NVIDIA to custom build for our use cases. So once the data is ready, then we use these tools and then we make it available online.
To make these high-quality atlases practically accessible for researchers globally, we have developed a web-based low-footprint viewer. By establishing precise spatial correspondence across these modalities, the viewer enables a seamless transition from gross brain structures in the MR images to features at the cellular level. This unified coordinate system allows navigation across adjacent histological sections, from NISL to the 8 immunohistochemical stains for all the annotated sections simultaneously. The online viewer offers synchronized side-by-side panes that align with identical spatial coordinates, enabling users to concurrently zoom, pan, and examine high-resolution cellular insets of NISL and immunohistochemical stains while preserving macroscopic content through co-registered MR images and block face images. Cross-referencing these complementary visual cues enables researchers to confidently identify anatomical borders of complex neuronal parts and validate macro to micro registration across imaging modalities. Using this detailed primary data, we have developed a 3D brainstem foundational model across brains using Oak Ridge National Laboratory's supercomputing facility. This model of the human brainstem is the world's first cell-level 3D brain model. Key findings include the identification of protoplasmic commissure dendrites of the hypoglossal nucleus, previously only identified in the African wild dog, and the distribution of specialized catecholaminergic neurons in the human brainstem. To our knowledge, this is the first time that the protoplasmic commissure dendrites of the hypoglossal nucleus have been identified in the human brain. The pretectal nuclei in the 25 gestational week fetal brainstem are also described in detail for the first time.
In addition to neurotypical brains, from fetus to old age, we are focusing on disease brains. And today we are releasing Anchor, which is detailed map of the brainstem of humans across lifespan. And this is a very important milestone in this mission of mapping 100 human brains.
Future developments of Anchor will include extended sets of immunohistochemical stains and applying in situ hybridization techniques as well as transcriptomic maps. We envision that these maps and atlases will have significant implications for neuroscience and neuromedicine.
Yeah. Conference. And when somebody was mentioning, oh, we should have a cultural program, right? I mean, generally what we do is we sort of tend to showcase, sort of a deep music, dance, and sometimes a performance art. But this time, being a neuroscience meeting, I had spoken to Rajappa here, who's a good friend of mine, and said, can we do something different instead of just a performance? They're going to attempt something which is to almost try to trace the pathway of why the mind behaves in a certain way when you hear music in a certain way. Of course, as all of you know, when I say pathway, it is the emotional pathway. Shubha is already looking at me like, right? Because, that's the ultimate mystery. We don't know why certain things happen to us in a certain way. So, it is not only a showcase of the culture of the land, it's also to make us think how something so subtle can have an extraordinarily non-linear impact where we can go from joy to tears, and sometimes it is joyous tears. Why does it happen? So, here is a troupe of some extraordinarily talented youngsters who, interestingly, all of them, most of them are in the technical field. That's very interesting, right? What really happens as when you make these choices, right? So I will leave you to them and let's enjoy the next 1 hour.
PPT can you? Thank you, Mohan sir. Thank you for the introduction and the beautiful context that you had set today. A very good evening and a warm welcome to all the respected delegates and the members of the prestigious IIT Madras who are here to be a part of such a historical event. A special welcome to all the delegates from different parts of the world. Welcome to India and welcome to Chennai. I'm sure you're already liking our country and this beautiful city. And of course, needless to say, the heat. Welcome once again. My name is Mayur Dave and I'm an Indian classical vocalist. We also have 4 other amazing musicians with us today who are going to be throwing some light on a few musical aspects and also entertain us with their enthralling music. Join me in welcoming Rajappa on the mridangam, Arvind Dave on the kanjira and the clapbox, Sai Govind on the mandolin, last but not the least, Sai Aditya on the keys and the harmonium. Music, mind and emotions, as you see on the screen. As we take you through the next probably 60-70 minutes or so, I'm sure another presentation, another talk, another panel discussion is not what you want to see on a Saturday evening. So, but of course, let me also set expectations. We will do a little bit of talking, but we promise we'll definitely substantiate with a lot of music. Music, mind and emotions. A topic based on which our musical presentation is going to be today. Let me open up with a question. Very, very basic for an esteemed gathering like this. Can some of you just list down a few emotions? Anybody? Oh, we have the senior most delegate, Hari, who is in grade 5. Yes, Hari. Sir, can you pass on the mic to him? Mohan sir, he is the future torchbearer of IIT Madras.
Fear.
Fear. You are so brave. Why do you call fear as the first emotion? But that's okay. One more.
Happiness.
Happiness. Excellent. Few more, please.
Anguish.
You need a mic? Somebody there? Anger. Okay. Sorry? Sorrow. Okay. Sorrow, sadness, misery.
Joy.
Joy. Happiness, joy. Very nice. Curiosity. Wow. Okay. Excellent. It can't be only something happy or sad. There are so many emotions that we all have, right? Few more.
Confusion.
Sorry?
Confusion.
Confusion. Wow. Excellent. Some more. At least a couple of more, please. Envy. Yeah, I mean, yes, why not? Jealousy, envious. Last one. Anybody? One last. Yes, sir. Sorry. Gratitude. Wow, what a point. Yeah, that can be an emotion. Yes, why not? Excellent. Now that brings me to my next big question. What, in your opinion, is the impact of music on any of these emotions? First of all, do you agree music may have an impact on emotions? Yes? Anybody no? Anybody no? Okay, excellent. So we all agree it is a yes. Music has an impact on human emotions. Excellent. So, what is the impact of music on emotions? And you will have to substantiate your answer. Why? Now takers, please, again. Hari is halfway through. You have to tell me why. What is the impact? I'll repeat my question. What is the impact of music on emotions? Why? Depends on the mic, please. I'll anyway tell what he says. Yeah, he's getting the mic.
It depends on the type of music.
Depends on the type of music. Wow. Can you elaborate more?
Because different types of music bring different types of emotions.
Different types of music will evoke different types of emotions. What an answer. Anybody who's differing with this? Yes, no? Yes, sir.
So, human brain is not evolved to understand Tamil, Kannada, Telugu, English, Russian, Japanese.
Absolutely.
So, music is universal.
Absolutely.
Will stimulate every neuron in the brain.
Superb. Thank you. That gets us back to the Neuroscience Symposium. Thank you, sir. Thank you so much. So now, yeah, that's wonderful answers. So from here we move on to the next one, where we say music on the whole, it is a very vast subject to cover, and especially when we are going to be talking about music and mind and emotions, I'm sure we can do multiple PhD with that. That's the kind of topic that this is. So we will today just spend some time trying to understand and also relate to these mind emotions kind of a thing. Music the Indian way. Now music, Vedas, as many of you would have heard, 4 Vedas based on which that's the guiding principle for the Indian civilization or the Indian tradition. Music has its roots from the Samaveda, which is the 3rd of the 4 Vedas. Let us now listen to a small excerpt from the Samaveda, which may be able to build a context. So, sorry, one second. A small portion of the Samaveda, Samaveda is very vast, just a small portion of the Samaveda that we will listen to, which may help us to connect as to understand why music, Indian classical music, sounds the way it actually sounds today, right? Just a small example. Does it sound like music? Did it sound like music? Not fully, but to some extent. So this doesn't sound like music. Music has its roots from this. Samaveda is the basis for the Indian classical music, known as Sangeeta. The roots of Sangeeta are firmly founded in the Samagana. Music in the ancient Vedic times was sung during the performance of yajnas, or the sacred sacrificial rituals, with Agni, fire, as the basis of the whole process. And it was here that the Samagana was born. Samagana employed only 3 notes, Udata, Anudata and Swarita. That's why the piece that you heard now seemed to be incomplete, because human now in today's world is used to a wider range of music, but what we just heard sounds a little incomplete. No, so that's the source from which the 7 notes have come. So these three notes, the Udata, Anudata and Swarita, later subsequently broke into wider notes, number of notes as the 7 notes that we all know, which is common to all forms of music across the world. What we call as Swara in the Indian classical music system, what we call as notes in the other forms of music. So now that we have started talking about raga, swaras. Now what is a raga? The whole Indian classical music is based on something called a raga. A raga can define the entire diaspora of the Indian classical music system. So now let me also take this opportunity to tell you, Indian classical music is present both in the northern part of India and the southern part of India, if I can just call it as two different parts of. So the northern part of Indian classical music is called the Hindustani classical music, and the South Indian music is called the Carnatic classical music. Many of you would have heard these terms. So now, what is a raga? As I said, a raga defines the Indian classical music, but what is a raga? So we spoke about 7 notes, right? The 7 notes in the Indian classical music, the Bharatiya Shastriya Sangeet, are termed as Sa, Ri, Ga, Ma, Pa, Dha and Ni. Again comes the Sa, which is basically the very first note, again getting repeated. So that becomes the next octave. Now, a completely varied permutation and combination of these swaras form a raga. To put it in a very simple definition, any kind of combination of these 7 notes can give birth to a raga. It can have all 7 notes, it can have 6 notes, it can have 5 notes. The other condition for a raga is also, the definition is also based on ascending and descending notes. A raga definitely will have ascending notes and descending notes, right? What we call as arohana and avarohana in the classical music system. Sarigama padani, sani dapa magari, for example. So it can be 7 ascending and 7 descending. It can be 5 ascending and 7 descending, or any kind of combination like this. It's possible. Yes, that is possible. These two define a raga. Now, are these plain notes going to define something? Sa, re, ga, ma, pa, dha, ni, sa. Okay, it sounds a little musical, but what out of this? Sounds very symmetrical and very plain. Doesn't make sense, right? No. So there are a lot of things that happen with these 7 or 6 or 5 or all these swaras, which basically gives birth to music, right? A raga is defined with a lot of other things, and one of that is something called a gamaka. The very next point that we're going to move to is about gamaka, and before that, so we were just talking about sorry. Ragas, mind and emotions. So as you can see on the slide very beautifully, so one of the most important point in the Indian classical system that we kind of follow is also about season, time. There are ragas. By now I'm sure you've got some basic understanding about a raga. So there are ragas which are very time specific. Does that mean I can't listen to that particular raga at a different time of the day? No, you can. But human mind, human body is completely aligned to different times of the day for various reasons. From a neuroscience point of view, from a brain point of view, I don't have to tell you, you know it all. But the beauty is that particular raga at that particular point in time of the day helps a human being align to it very easily. So it makes it more meditative for the listener, or it can make it more focused for the listener, or it can be different things, different emotions throughout the day. So that is something that is a great impact of Indian classical music on the human brain. I wouldn't confine it only to the human brain. It can even be emotions of animals. Why not? We have seen that. Elephants and cows reacting to music, peacocks reacting to music, right? So This is a quick slide just to touch upon that. Without much ado, let me move on to the next one, where we are going to be touching upon something called gamakas. Gamakas are basically notes oscillating, I can say, in a very simple language, but I'm going to pass it on to our mandolin player for the day to take it over from here, who can demonstrate and explain how this works. Over to Sai Govind for this.
A very good evening to one and all present here. So before we get into gamakas, I would like to introduce this instrument that I am holding in my hand. So this is a 5-string electric mandolin that we have here. So this instrument originated in Italy a couple of 100 years before. So it was an acoustic instrument. Just about 50 to 60 years ago, a great musician called Mandolin Srinivas completely transformed the instrument by making several adjustments. You can see the adjustments on the slide. So one of the main adjustments that he did was, the instrument was a hollow acoustic instrument. He made it electric. So why did he make it electric? He made it electric to enable one of the very fundamental aspects of Indian music. So making it electric made him play the gamaka. So gamaka is the bedrock of all Indian music. It's the very foundation of Indian music. So a very simple definition of a gamaka can be deviation of a note from its original position. So the note deviates from its original position. Gamakas can be very simple, or they can go on to be very complex, involving multiple notes or even notes between notes. So this is a neuro symposium. So one of the natural questions that arises is, how does the brain perceive such complex pitch movements? They're all pitch movements, right? So I got a study, a research paper from Mr. Ananthanarayanan Krishnan and his colleagues at Purdue University. So this was published in the journal Brain and Language in the year 2014. I would like to place a disclaimer that this study is not about Carnatic music or gamakas. It was actually to investigate how the auditory cortex processes dynamic pitch contours. So for our purposes, we are trying to extrapolate the findings and see what we can find from that. So I would like to give an example. We have a board in front of us. So we draw 2 points there. So our brain perceives it as 2 different entities. So the same 2 points we join with a line. No matter the nature of the line, now we look at it, our brain perceives it as just one entity. So this concept comes to music as well. So for example, let me play 2 notes. So I am playing 2 notes. So now we perceive it as 2 entities. I am going to join the notes with a straight line now. It's just one entity. So our brain not only processes the beginning point and the ending point, but it also takes the path into account. So it's not just a path or the direction into account, but there is also contour which is taken into account. So there are different contours, as you can see in the slide above. So I'm going to take 2 notes, ga and pa. So first contour, it's a fast sine wave like movement. It's a slow sine wave like movement. It's just a straight movement which is slow. This is a fast straight movement. So all of these represents different contours that our brain can produce. And when we plot all the contours on a time plot, we also get the acceleration into account. So I can accelerate in the beginning of the movement, or I can accelerate towards the end. So all of these are processable by our brain, which is what we can see in the study there, where pitch is a multidimensional perceptual attribute that relies on several acoustic dimensions, one of which we saw is the contour. So our great composers of the past have leveraged this, and they have used so many of the combinations of these gamakas into their compositions. So I'll just play some simple ones for you to listen. So notice there are about 4 to 5 notes involved in each. This is the world of gamakas, which form the foundation of our great tradition of music. Thank you.
Thanks, Govind. Thank you so much for those insightful points. Now, is it all about melody that makes music? I'm sure it's a no for an answer, right? So it is not about only melody, because the other most integral part of any form of classical music or any form of music is going to be something based on rhythm, tempo, right? Which we can draw an equivalent to the heartbeat. So now the rhythm portion of any form of music is going to be adding so much of value to it. Now let's try to understand from an Indian classical point of view, and today we have a couple of them who are going to be showcasing us and talking about 2 different instruments predominantly used in the southern classical music in India, which are the mridangam and the kanjira. So I will now pass it on to Rajappa and Arvind to quickly touch upon these points.
Good evening, everyone. So far, we were exploring everything about ragas, gamakas, swaras, so on and so forth with respect to the musical notations aspect of Indian classical music. Let me now take this opportunity to talk about 2 special percussion South Indian instruments, which are mridangam and then the kanjira. Along with them, we also have the clap box, which beautifully bridges traditional and contemporary rhythmic traditions. India has a rich variety of percussion instruments, but one of the unique features of South Indian rhythm is its extraordinary combinations of mathematics, creativity, and improvisation. The possibilities of rhythmic patterns are almost limitless. Before we explore some of these patterns, I would like to share 2 simple thoughts about rhythm. First one is, rhythm is life. There is a reason for me to say this, and the reasons are as follows. Our heartbeat is a rhythm. Our breathing has a rhythm. Even our daily cycles of movement and rest are rhythmic in nature. And the second one is a famous saying from the Indian classical music, Shruti Mata Laya Pita. It means melody is the mother and rhythm is the father. Shruti refers to the musical notes, swaras and ragas, whereas Laya refers to the rhythm and the flow of time. If I may put it playfully, melody represents qualities we often associate with nurturing, emotions, beauty, expression and connection, whereas rhythm, like our father, represents structure, discipline, balance, precision and presence. When melody and rhythm come together, they create what we call as music. Just as Indian music ragas and swaras, the rhythmic system also has its own vocabulary. And like aksharas, nadai, matras, korvai, so on and so forth. To understand this, let us look at one of the most popular rhythmic cycles in Carnatic music, which is the Aditala. Aditala consists of 8 beats or aksharas for one cycle. So to perform Aditala, it goes like this. 1, 2, 3, 4, 5, 6, 7, 8. So this completes a cycle, one cycle of Aditala. So one cycle of Aditala has got 8 beats or 8 aksharas. This can also be looked at from a different point of view, that is, each beat of Aditala has got 4 subbeats into it, which we call as Madras. And it goes like this. I will repeat one cycle of Aditala with aksharas. 1, 2, 3, 4, 5, 6, 7, 8. Now, if we perform the same cycle, one cycle of Aditala with subbeats or Matras, it becomes like this. 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, ta. So, in one cycle of Aditala, there are 8 beats and 32 subbeats. So, in one cycle of Aditala, we have 8 aksharas and 32 matras. Within this framework, musicians create countless rhythmic possibilities. One such form is what we call as korvai. A korvai is a structured rhythmic composition that is traditionally performed 3 times in musical concerts. It combines mathematical accuracy with artistic expressions. A korvai generally has 2 aspects to it. One is the purvangam, the first part of the korvai, and the uttarangam, the concluding part of the korvai. We will now see how a simple korvai is performed. First, we will recite the korvai that is composed in Aditala, and then we will look at the numerical patterns involved in the korvai, and also we will see how it feels if the same korvai is performed with our instruments. So we'll now recite the korvai. Tata dum. Tarikata tata dum, tarikata tata dum, ta di ki ta dum, ta di ki ta dum, kedaka tarikata dum, kedaka tarikata dum, kedaka tarikata dum ta. So this is something we call as korvai. This has to be performed 3 times, and the notations which I use right now are called as jatis. Let us now look at the same korvai from a numerical mathematical standpoint. 1, 2, 3, 1, 2, 1, 2, 3, 1, 2, 3, 4, 1, 2, 3, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, ta. So this is the mathematical composition involved in this korvai. Let us now see how the same korvai feels to us if performed using this instrument.
Notice something. The melody instruments also joined the percussion instruments towards the end because they complement each other in the Indian classical music system. Also because the patterns, the mathematics are all common between the melody and the percussion instruments. So that is another very unique aspect of the Indian classical music system. So now without much ado, let's start exploring a few ragas with some musical presentations. So the very first raga that we are going to be taking up for this evening is a raga called Kalyani in the Carnatic system, which is also called as Rag Yaman in the Hindustani system. Now this is again a raga which is full. When I say full, it has all the 7 notes. Sa, Re, Ga, Ma, Pa, Dha, Ni, and the same way back. That is Raga Kalyani or Yaman. As you can see on the slide, Kalyani is a raga which is the time and the season of this raga, time of this raga for the day is basically evening. That's the reason we took this raga for this evening. So a raga which calms the mind, a raga which evokes devotion, evokes spirituality, a raga which can create a lot of inner peace, a raga which can inspire a lot of positivity, a raga which leaves a lasting impact. So we will try to take you through this raga in various forms. Let's do that. आज जाने की जिद ना करो, आज जाने की जिद ना करो, यूं ही पहलू में बैठे रहो, यूं ही पहलू में बैठे रहो, आज जाने की जिद ना करो, हाय मर जाएंगे हम तो लुट जाएंगे, ऐसी बातें किया ना करो, आज जाने की जिद ना करो, तुम ही सोचो जरा क्यों न रोके तुम्हें, जान जाती है जब उठ के जाते हो तुम, हमको अपनी कसम जाने जा, बात इतनी मेरी मान लो, आज जाने की जिद ना करो, आज जाने की जिद ना करो. This was a ghazal which is predominantly sung in the northern part of India, comes from the Hindustani classical music. We will now also listen to a very bright composition, an instrumental composition from the southern part of India, the Carnatic classical music system. So we will have a presentation with the Carnatic Kalyani. What we heard just now is Raga Yaman, but we will also listen to Raga Kalyani now. Thank you. Thanks to all four of them. That was thunderous. So a raga which can be very serene, a raga which can even be thunderous. That is Raga Kalyani or Yaman for all of you. Moving on to the next raga. So this is an early morning raga. It's called Maya Malava Gaula in Carnatic, which is called as Rag Bhairav in the Hindustani music system. A raga which is extremely intense, I would say, as you can see the slides also. A raga which can really instill focus in somebody. So imagine if it's going to be 5 a.m. in the morning and you wake up and there is a very meditative music going on, and it really gets us into a meditative state, isn't it? That is Rag Bhairav, Rag Maya Malava Gaula. It can instill focus, a lot of serenity, a lot of meditative. It can really bring somebody to a meditative state. Create a feeling of serenity as you see that. As you start the day, set the tone for the day and keep going. It's very majestic as well, and not just about only meditation, it's very majestic. So very interesting fact that I would also want to mention here, in the Carnatic music system specifically, when somebody starts learning this form of classical music, the very first raga that is taught to the student, irrespective of the age of the student, is this raga called Maya Malava Gaula. A raga which is full again in nature, which has all the 7 notes. Sa, re, ga, ma, pa, dha, ni, sa, sa, ni, dha, pa, ma, ga, re, sa. That is Maya Malava Gaula. Just to give you an example of how these basic lessons start for a beginner, we will just try to spend a couple of minutes to show you how these basic lessons sound. Sari 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 gama sari ga sari ga sari sari gama sari gama sari gama pa sari sari gama pa da sari gama ri gama pa sari gama sa ga ri ma sari gama sa ma ga ri sari gama pa ma ga ri sari gama pa ma da pa ni da pa ma sa ni da pa ma ga ri sa pa da ni sa sa ni da pa sa ni da sa ni da sa ni da pa sa ni da sa ni da pa ma sa ni da pa ma ga sa ni da pa ma ga ri sa ni da pa ma sa ni da pa sa da ni pa sa ni da pa sa pa da ni ma pa da ni ma pa ga ma ri ga ma pa sa ni da pa sa ri ga ma pa da ni sa ma ga ri sa. That's something which is very basic when somebody starts as a beginner. Maybe like our senior delegate, Mr. Hari. Hari, have you learned something like this? You remember this? Have you learned something like this? No? Maybe you should start. Yeah. So that's a very basic beginner lesson. So after years of learning, years of intense practice and trying to hone that skill more and more with so much of practice, somebody reaches at a stage where the same raga sounds really intense and very majestic. Let's now try to listen to something like that, which is the greater form of this great raga, the majestic raga Maya Malava Gaula. Over to the boys. Wasn't that serene? Imagine this kind of music early in the morning at about 4:30, 5 o'clock sunrise time. That's the beauty and majesty of a raga called Maya Malava Gaula or Bhairav. Moving on to the next one. In the interest of time, we are going to skip one. We'll move here. A very unique raga. As you can see, I will even call out a couple of unique aspects of this raga. First of all, the ascending and descending notes that we were talking about some time back. The Arohana and Avarohana. So it has 5 ascending notes and 7 descending notes. This is a raga called Desh in both the systems very beautifully, both the Hindustani and the Carnatic system. Raga Desh. Now, the very word called Desh in the Indian language, Desh means country. In very simple terms, Desh means country. Simple. Now, a raga, a scale, imagine a raga or a scale which can represent a country. I'm sure all my Indian brothers and sisters who are here will agree with me. If you listen to something in Desh, immediately what comes to your mind? India, patriotism, Independence Day, Republic Day. So I take this opportunity to showcase this great, beautiful raga to all our delegates from all over the world. So this raga is very personal to all of us because it represents our country. A raga which can represent a country, it is a very unique thing across the world, I'm sure. There may be some other country which has a raga or a scale which represents, but which I have not heard of. So that is a very unique thing about it. So this raga also is a very warm raga which also evokes a kind of sense of nostalgia. Very warm by nature. Immediately brings something like a rain or a cool breeze to the mind. Imagine how beautifully these kind of ragas can have impact on the human brain or even our emotions. The moment you listen to it, how beautiful that sounds. So we are going to take this opportunity as a team. We are definitely going to take this opportunity to sing the national song of India, which is Vande Mataram. Most of you would have heard, most of you will know, most of you would have heard, in the interest of people who have not heard. And if people want to join, you are most welcome. Absolutely, absolutely welcome. So we will sing Vande Mataram and then we will move on to a very, very peppy piece in the same raga, just to showcase how a raga can really give you the sense of patriotism one side, on the other side can really make somebody dance. We are going to see both. So first we will start with Vande Mataram, and anybody here who's willing to join, please join. Don't worry, you don't have microphones, so don't worry, you will go off Shruti, you may not sing properly, just sing. If you know it, please join us. Right? Vande Mataram, Vande Mataram, Vande Mataram, Vande Mataram, Sujala, Shubhala, Malaya Shitala, Sashyamana Mataram, Vande Mataram, Shubhra Jyotsna Pulakita Yamini, Mulla Kusumita Drumadala Shobhini, Subhashini Sumadura Bhashini, Sukha सदां वरदां मातरं वन्दे मातरं वन्दे मातरं वन्दे मातरम्. Thank you. So we will now listen to a very peppy piece in the same raga Desh. I'm sure you all will enjoy this. Over to the boys. Rag Desh for all of us. Thank you. So, we're slowly moving to the last segment of our musical presentation. We really hope in the last about 60 odd minutes, we were able to share some useful insights about the Indian classical music system. And we are really happy to have shared that with all of you here. Last but not the least, we would like to present a song called Maitrim Bhajata. I'm sure some of you would have heard it, some of you know it. Maitrim Bhajata. The theme of which you can see it on the screen. Very apt for today's world, right? So the very theme of this whole song is all just about world peace. So in these trying times with so much of chaos across the world, in the form of war, in the form of scarcity of resources, in the form of political and religious conflicts, whatnot. Too many things are happening across the world. Let's all come together, help each other, love each other, and spread the love. Let there be only one religion, the religion of love. Let there be only one language, the language of the heart. Let peace, happiness, and prosperity be to all in all the worlds. Maitrim Bhajata. Maitrim Bhajata Akhila Hrijetri. Maitrim Bhajata Akhila Hrijetri. Maitrim Bhajata Akhila Hrijetri. Atmavadeva Paranapi Pashyata. Atmavadeva Paranapi Pashyata. Yuddham Tyajata Spardham Tyajata. Yuddham Tyajata Spardham Tyajata. Tyajata Pareshu Akramamakramanam. Tyajata Pareshu Akramamakramanam. Maitrim Bhajata Akhila Hrijetri. Maitrim Bhajata Akhila Hrijetri. कामदुघास्ते जननी पृथ्वी कामदुघास्ते जनको देवः सकल दयालु जनको देवः सकल दयालु दाम्यत दत्त दयत्वं जनता दाम्यत दत्त जयत्वं जनता श्रेयो भूयात् सकल जनानां 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 Shreyo Bhuyat Sakala Jananam. That's the very crux of this song. Let peace, happiness, and prosperity be to all in all the worlds across the universe. Thank you so much. Thanks to all of you who are present here on behalf of our entire team. I really express our gratitude to all of you to have given this great opportunity to all of us in such a historical and a prestigious event like BRICS as part of the initiatives from IIT Madras. IIT Madras is, as you all know, is one of the most prestigious institutions in our country, and we definitely are now very grateful to have got this opportunity, because we have literally grown up always listening to stories about IIT Madras. We used to come here, and a couple of our family members have studied here. It's such a great opportunity, and we are very grateful, very, very grateful to Mohan sir. Sir, thank you so much for this great opportunity. Thank you once again. Have a great evening and have a great rest of the stay in our country. To all the foreign delegates who have come to attend the symposium, thank you so much. Thank you.
Thank you so much. Today morning we started something with mindfulness, and I think this is something we are ending with more soothing. So now I request Professor Mohana Shankar to please come and felicitate them.
Making an announcement on where people should board the bus? Sorry.
Sorry. Take it. I'll take the last option.
I request all the delegates to please head downstairs. There is a bus waiting for you, so you can proceed towards the ITC for your dinner. Also, I request external participants and SGBC staff to kindly be here and enjoy the gala dinner.